one, but I don't know if I can get it all the way down. Or the whole, it would, yes. Let me just read this. It's not a primary sharing the PowerPoint presentation. There isn't a lot of memory. And I think this camera view, so the only thing that will have a PowerPoint is a panel discussion. But the camera can see that. Yeah, even if we had to see, we could at least see the speaker. We can see the panel whenever we do it this afternoon. So if that's what we're left with, I don't. Yeah,
Just need to get that in.
yeah. Like people at home, they're in PowerPoint part, but they're not like, well, actually have your camera that you can see.
Verify that you got audio on both sides.
I did yesterday. I logged in about to jump in on the YouTube side. There, I don't know. Yesterday was. Okay. I really get done.
And sorry, if the, you know, program. How much do you think, you know? Spend all day, you guys,
Yeah.
it's almost all IP. Operations and all that.
Yeah, it's no problem at all.
Set the monument voice on What, not even exactly.
Yeah, I think. It's just, oh, whatever. Yeah. Right. Test. 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 Test.
Yeah, coming along. Are you still? Short time. Thank you. Like what? Everybody.
There will be that you can turn on, so there won't be notes. But you will be able to see in theory.
Gosh.
I was learning. Standing around something. Yeah. Actually, that guy, I think. Yeah, so maybe you're, is that it? Stand by. Okay.
Whenever I.
Yeah.
For now, it's right here. So. Still like I would.
Marks.
Like you're.
Lots of moving slightly.
Yeah, they didn't know.
Stand by here. About the first time. Yeah. Yeah.
Yeah. Yeah. Yeah. I heard. It's like mess things. Good luck.
They're roughly off. Wait, myself.
Maybe. I think it's kind of.
Satisfaction.
What do I gotta check it out. I was thinking about it.
Good morning, everyone. Just to remind you, we'll get started here in a little bit. But downstairs, there are some refreshments, so coffee, juice, water, some snacks if you need something. If you do have a presentation, bring it up to the front. That way I can load it up to our computer. That way we can kind of get everything going once we start. I am missing one presentation for this morning, Texas Reservoir Ecosystem Networks. So Malcolm, if you're around, do you have your stuff? Okay. Yes.
Good morning. We are going to get started. So I'm Ben Pierce. I'm Professor Emeritus of Biology at Southwestern University and part of the planning group for this symposium. It's my honor this morning to introduce the president of Southwestern University, Dr. Laura Scandera-Trombley. Dr. Trombley is the 16th president of Southwestern. She came here in 2020 in the middle of the COVID pandemic. What a way to start a new job. She's prior president at Pitzer College, at the Huntington Library and Museum, and at the University of Bridgeport. She's a great champion for the liberal arts and has been a strong advocate for environmental sustainability. She's a preeminent Mark Twain scholar and has written several books about Mark Twain, particularly about the women in Twain's life and the influence that they had on his work. She's also an avid outdoorsman. She's hiked all 217 miles of the John Muir Trail. And just a couple of years ago, as president, she took a trip to Antarctica over the winter break. So please join me in welcoming Dr. Trombley to Texas Conservation Symposium.
Well, good. I'm back. So good morning. It's wonderful to see you here today. It's a beautiful day in Georgetown, and looking at your faces, I kind of feel as though we should all take a hike. I have a feeling that you may be pretty regularly enjoying the outdoors here in Austin. And it's true, I did arrive in the middle of a pandemic to a campus that was completely empty. And I arrived with my son and my bulldog. He was part of the class of 2020 that didn't have a commencement ceremony. And so he came with me, and for 2 months, it was just the two of us on the Southwestern campus. And that's when I learned just how much flora and fauna you have here in this part of Texas, because everybody came around, the deer, the armadillos, the foxes. I realized that this is just an incredible resource. So I want to commend you for having this conference, particularly Ben Pierce, who has been a leading light on our campus, teaching generations of students, and who I think really has promoted sustainability, biodiversity, and conservation on the Southwestern campus. A little bit about Southwestern. We are a university that runs on 100% renewable energy. We're in the process of building 3 new buildings on campus, 2 new residence halls and a welcome center. They will all be gold LEED rated. We started an outdoor rec program a few years ago, and I'll admit in part because I thought it was important for students to stop looking at their hands and to start looking at the sky. And we take our students to go kayaking, backpacking. Last year, we brought a group of 12 to summit Mount Kilimanjaro. And I had trustees ask me why, and I said, why not? They're 19. They should go do something really exciting that will follow them for the rest of their lives. Also, we are very fortunate. We are the largest private university landholder in Texas, more than Baylor, just saying. And we have 560 acres that is contiguous to our campus. It isn't raw land. It was actually a dairy farm for many, many years. And we have owned that property for about 50 years now. And we are now talking about creating what would be a very much sustainable, environmentally friendly campus on that property, where we have already set aside 80 of those acres that will never be developed. Those are going to be nature trails and floodplains. Again, this is a beautiful part of Texas, and I watch the development that's happening here, and I feel as though part of our responsibility as a liberal arts institution, where we try and model what we think is ethical behavior, we have to protect that land and save that land for future generations, as well as make that even more so part of the Georgetown community property that we like to share. So it is particularly fitting then that this symposium places an emphasis on student presentations, encouraging emerging voices in conservation. And your voices are needed more so today than ever before. I come from Los Angeles, and I have spent the last week in tears watching the city that I love burn to the ground. And I just found out last night that one of my roommates in college came back to her home to find it was in ashes. And so we have to really think very carefully, particularly as we see all of this development coming to this area. It is a very exciting time. I think, pretty much keep my hours shopping at HEB to 2 o'clock in the morning because that's the only time I can find a parking spot. We have so many new people coming to this area, and we have to be very, very thoughtful about how we want to continue the development here. So we keep what is most beautiful and precious to us. So I want to thank you for your commitment. I appreciate you coming. I know that this conference keeps getting bigger and bigger and that we have lots of people who are going to be online. So we are now a hybrid conference, and I hope to see that this continues to grow. And I want to thank you for your commitment, for taking the time to be here today. I wish you all the very, very best and happy trails. Thank you so much.
So Texas Conservation Symposium is co-hosted by Southwestern University and the Williamson County Conservation Foundation. I'm going to ask Josh Renner, who is the program director, environmental program director for the Conservation Foundation, to come and give us a few instructions and logistics.
Thank you, Dr. Pierce, and thank you again, President Trombley. The support from Southwestern has always been astounding, and we certainly couldn't do it without Southwestern and staff. So just some housekeeping things before we get started. This is a hybrid meeting, so if you are giving a presentation, if you're not speaking directly into the mic, so even though you can hear me right here, no idea what I'm saying. So they're just watching me wander around. We do have refreshments downstairs, coffee, juice, water, some foodstuffs. We'll have that this morning, after lunch today, and then tomorrow morning. So if you're planning on, hey, when am I going to eat? Outside of lunch, we will feed you, but lunch is on your own. We do have presentation remote. So if you're up here, we can get that rocking for you. If you do have a presentation that you haven't uploaded and you're presenting today, find me or staff up here at the front during the breaks or during lunch, and we'll upload that for you. So yeah, with that, we can go ahead and get started, Dr. Pierce.
Yeah, let me add my words of welcome also to the 11th annual Texas Conservation Symposium. This symposium is very diverse. We have a wide range of talks and posters, everything from geology, groundwater, conservation management. We have talks and poster presentations on trees, ants, freshwater mussels, cave spiders, monarch butterflies, fish, turtles, lizards, snakes, frogs, salamanders, birds, bats, peccaries, rabbits, and more. So there should be something for everybody over the next 2 days. I want to sort of highlight a couple of special features of the symposium that we have coming up. This afternoon, starting at 2:45, we have a series of talks and then a panel discussion on current issues in bat conservation, a very timely topic. This evening, you're all invited to attend a reception from 5:30 to 7:30 at the City Post Chop House in downtown Georgetown, a very elegant venue. Hope you'll all try to be there for that. Tomorrow at 9 a.m., we have our keynote speaker, our keynote address from Dr. Timothy Bonner from Texas State University. He's going to share with us some conservation insights from his 30 years of field research in Texas. We have a number of sponsors for this meeting. This meeting is free, doesn't cost you anything, and that's a direct result of the sponsors that we have who have financially supported the symposium. So we want to thank them, and some of them have tables back here, so I would invite you to visit the tables of the sponsors sometime while you're here over the symposium. As Dr. Trombley mentioned, we have a number of student presentations, and we give awards for both the best student oral presentations and the best poster presentations, and we'll announce the winners of those awards at the end of the day on Friday. We had an overwhelming response to our call for abstracts this year, the most presentations by far we've ever had before. We have 36 oral presentations and 25 poster presentations that we'll get a chance to look at over the next couple of days. So again, welcome to the Texas Conservation Symposium. So just some instructions for the presenters. Josh talked a little bit about this. If you're going to present, be sure to get your presentation to Josh or to Heather up here before the session in which you're going to be presenting. We're going, oh, and be sure to stand behind the mic. I don't like being tethered to the podium either, but as Josh said, our online people will not hear you if you're not standing up here talking to the mic. So we gotta stay in front of the microphone during your presentation. The presentations, most of the presentations are 15 minutes. That includes both the presentation and questions and answers. So you should plan to talk for 10 to 12 minutes, leaving 3 to 5 minutes for Q&A. When I'll be timing the presentations, when you get to 10 minutes, I'll hold up 5 fingers indicating you got 5 more minutes. When you get down to 13 minutes, I'll hold up 2 fingers. And then when it gets 15 minutes, I'll stand up and ask you to stop. And there's a device up here that allows you to advance the slides and has a pointer. Okay, so let's begin with our first presentation this morning. Our first oral presentation is by Aaron Hitchcock, and it's entitled Managing the Benefits and Challenges of Regional Habitat Conservation Plans in Central Texas.
Good morning, everyone. Hopefully everyone can hear me. Very excited to help kick things off today. Just a little bit of background on myself. I've been supporting the development and implementation of habitat conservation plans for about 15 years now. And currently, I am supporting the development of the Bell and Coryell County Regional Habitat Conservation Plan as the project manager for the technical team. And before I forget, I just want to mention that there are other key members of our technical team here today, Jared Haney with ICF, who you will hear from later, and Cambrian Environmental. Where am I pointing this? Just a quick snapshot of what I'm going to talk about today, provide a very brief overview of habitat conservation plans, we call HCPs, talk about regional HCPs, their benefits, their challenges, how we can best manage the benefits and challenges. And then I'm going to talk about the Bell and Coryell County Regional Habitat Conservation Plan, which we call the BellCor RHCP. So just, a little bit about HCPs for those of you who might not be familiar. And if you need more information, you can feel free to find me at the break or later today. Under the Endangered Species Act, non-federal project proponents must obtain incidental take permits for impacts to federally listed species. And in order to obtain incidental take permits, a habitat conservation plan must be developed. And this requirement can be met through the development of a regional HCP. So what are regional HCPs? They are a tool for addressing widespread development and population growth related impacts to federally listed species. They are landscape scale plans initiated by state, county, local municipalities, and other entities. Water agencies, that's becoming much more common given that there were several freshwater mussels that were listed last year. These are programmatic in nature, covering multiple projects or activities over decades, very long periods of time. And they provide large-scale conservation benefits for federally listed species, but oftentimes these species act as umbrella species for other non-listed but sensitive species and habitats within the plan area. So as with any large-scale long-term plan, there are benefits and there are challenges. first walk through some of the great benefits of these plans. They provide comprehensive conservation, covering large areas, multiple species, really providing this holistic approach to species and habitat conservation. And depending on the conservation approach that's taken, they may create or conserve interconnected habitats that enhance biodiversity and ecosystem health. The second one is a big one, streamline permitting as compared to project by project permitting. These regional habitat conservation plans reduce timelines, they reduce costs, the uncertainty, and just really create a more predictable regulatory environment for project proponents, developers in particular. Economic efficiency is another big one. These regional HCPs allow the plan participants to really pool their resources together to be able to conserve larger, to essentially pay for larger conservation efforts that they would otherwise not be able to afford. So it just really provides a greater economy of scale in these efforts. And then finally, adaptive management, which is really a requirement of these plans, but very important because it allows for the plan adjustments based on new scientific information from monitoring, and ensures the flexibility and the long-term effectiveness of the plan. Now, just a look at some of the challenges with regional HCPs. So they do have high costs and time commitments. Developing and implementing HCPs, regional HCPs, they can be expensive and time consuming, and they do require a big commitment from all key stakeholders. They are complex coordination efforts involving federal, state, local agencies, as well as advisory groups. And in Texas in particular, regional HCPs are required to have a biological advisory team and a citizen advisory committee as per the Texas Parks and Wildlife Code. And I mean, it's not a negative thing at all, but it adds complexity to the process because, more people commenting and weighing in on, the approach and the strategies going into that plan. Uncertainty and risk is definitely a big challenge with these, again, large-scale, long-term plans. Being able to predict future environmental changes and the impacts on species and habitats really leads to, can lead to uncertainty in the effectiveness of the conservation strategy. And then finally, balancing development and conservation. It's just really important to ensure that the covered activities do not undermine the conservation goals. And then this really takes, careful planning and just ongoing management, but it can be a real challenge with these plans. So, here are some ways that I have found to best manage the benefits and challenges of these plans. There's no real specific order here, but I think the first one is very important. Effective communication and engagement with stakeholders and agencies and the public. This really starts in the early planning, or should start in the early planning stages with the formalization of a plan or a process of how the plan participants are going to communicate and work together, how they're going to communicate with the agencies, how they're going to communicate and engage with the public. This should all be laid out early in the process. And that's not to say that the approach cannot change or pivot should there be some sort of need down the line, but it's just really important to have a plan for that and something to look to be consistent throughout the process. The second one is also, I've found very important, just understanding the unique challenges or obstacles specific to your plan area and incorporating targeted strategies to address and monitor these issues throughout implementation. this can mean different things depending on your plan. It could be political obstacles, it could be environmental obstacles. You need to know what those challenges are and be able to address them early on before they become problems. The third one, ensure sound funding strategies and efficient use of resources. This is one of those that there's not a one size fits all for each plan. There are obviously standard funding strategies that are utilized. Development fees or participation fees are common. Contributions from the plan participants or stakeholders is common, but there's a lot of other creative ways to provide funding for a plan. And so again, just really understanding the needs and just making sure to plan accordingly. And finally, I think the last one is pretty obvious, but can be very challenging. Developing clear and achievable goals and implementable measures. This can be a challenge because oftentimes we have species that have recovery plans and specific goals, and the agencies, want those goals to be reflected in the plan. And for whatever reason, sometimes that is not feasible. Or the agency may just have, expectations for goals or a strategy for a certain species. The key here is just really for the plan participants and the agencies to have those thoughtful discussions and to work those issues out so that the plan participants don't end up agreeing to measures that they know that they cannot implement down the line. It is not good for anyone, the plan or the species. This is a really key area. So just a quick snapshot of the currently approved regional HCPs in Central Texas. I'm not going to read through the list here, but there are 6 currently. I just want to mention that I've included Edwards Aquifer HCP, which technically meets the definition, Fish and Wildlife Service definition as a regional HCP, but it doesn't meet the definition under the Texas Parks and Wildlife Code. That's for a whole nother talk. But for the purpose of this conversation, I think these are the 6 regional HCPs that we have right now. So now on to the Belcour RHCP, which is currently in development. We are in our 2nd year of development. The incidental take permit is expected early in 2028. So it is a collaborative effort between 13 entities. You can see them all listed at the bottom. Now, this is a messy slide, but I think it's sort of representative of just how complex this effort is. You have 2 counties, 6 cities, a village, and a planning organization. The expected plan coverage is going to include all of Bell and Coryell County, excluding Fort Cavazos, so all non-federal lands. We are looking at including 10 species, including karst invertebrates, freshwater mussels, a bird, salamander, bat, and butterfly. And the covered activities are going to include development activities, public facility, construction and operations and maintenance, and select water management activities, including surface water and groundwater activities. And one of the big goals of this plan is to have this large-scale coordinated regional conservation effort where this plan can work with other adjacent entities like Fort Cavazos and Williamson County, RHCP, and work with them on kind of conservation for those species that the different efforts have in common, for the benefit of the region and those species. So now just kind of a little bit more about the Belcour RHCP. So these, these are really kind of again, we're in year 2, kind of keep that in perspective. If I came back, next year, it might be, more lessons to be learned. And, we're learning, kind of new things every day and just trying to gather as much information as possible to develop our strategy. But currently, the key benefits that we see for this plan are, number one, streamlined permitting. This is really this was the impetus for this plan, was to be able to simplify the compliance and permitting for some of those complex species that occur in Bell and Corral County, the aquatic and terrestrial karst species that can be very time consuming and expensive to comply with. Large scale conservation. So again, we have 2 counties, just a huge potential to conserve large tracts of land. And the potential to undergo these collaborative efforts, like I mentioned, with Fort Cavazos and Williamson County RHCP, just really for the benefit of the region, just, working with them to try to coordinate on large-scale species conservation strategies. Some of our key challenges, environmental uncertainty. So water availability, that's huge, related to drought and competing uses. Climate change, always a big uncertainty. Unsurveyed areas in these 2 counties, we do have, a number of data gaps that create challenges in terms of understanding, are these species here? Are they going to show up? Will they be impacted? And stakeholder coordination, as I mentioned, 13 entities, that definitely creates some complications in terms of, keeping everyone aligned. There are different agencies with different areas of focus, and then cost. Again, I already mentioned this, that there is that high cost, especially that initial high cost for development and implementation. And so some of our key strategies, I guess number one, just really based on, having a good scientific foundation. We're conducting hydrologic modeling, including climate change scenarios. We are developing a robust monitoring and adaptive management strategy. And, the third one, this is kind of huge. We're really trying to learn from other HCPs in terms of looking at, how they expected to do things versus how, what information they're collecting from their monitoring, what they're seeing in, year 5, year 10, to learn from that. The operating framework, I think I talked about this before, but, we created an operating framework that really outlined the process for how the plan participants would work together, how they would work with other stakeholders, with the agencies, with the public. And that just created a really clear process for everyone, all the way through to decision making. And then funding, we, like many HCPs, this plan has taken advantage of the Section 6 funding. And then we're also just, again, looking at other examples for other creative funding ideas, looking at Williamson County HCP and their kind of, their tax benefit financing options and some other options maybe to work with some federal funding programs. That's all I got.
So unfortunately, we don't have time for questions, but catch Aaron during the break if you have some questions about the presentation. So our next presentation is from Valerie Collins, Balancing Development and Conservation, the UTSA Habitat Conservation Plan for Karst Ecosystems.
I'm going to try to go through this quickly. So we have time for questions because I have Lonnie May from UTSA here and Dante Fonolio from the San Antonio Zoo. Is it? There we go. Here's the agenda of topics. I'm going to try to go through this in 10 minutes, so I'm going to whiz through it. I'm also not going to go into all the details about an HCP because I think most everybody here knows what an HCP is. So I'll kind of go through those kind of quickly, hit the high level topics. That's us. HCP overview. So this is this is a unique HCP. So how this HCP differs from the other HCPs that you typically have heard about is, number one, it's a university. It's a university's HCP. That's a little bit different. Number two, it's incorporating research at a level that other HCPs normally don't, because a university is a research facility. And so we want to try to incorporate that as much as possible into this HCP. And number three, it involves more collaboration for that research and the implementation than probably a lot of HCPs involve. But here is the area where the key area in the UTSA HCP. And this is where you have critical habitat unit 9 on the Loop 1604 campus of UTSA, University of Texas at San Antonio. There are 105 acres of critical habitat unit 9. It's in the UTSA, aptly named UTSA KFR. This HCP covers Cichurina madla and Rhydione exilis. We've got those species on this campus, but it also covers other species within the KFR and golden cheek warbler. We've got a number of caves on campus, named caves, long-known caves, Mastodon Pit, Lost Mine Trail Cave, features F50 and 43 on the east side of UTSA main campus. Those are the ones that have been on the books for a long time, but we did a lot of field work for this and updated it. So this HCP is going to cover development of the campus. We're also now incorporating, since there was the merger of the UTSA University with UT Health, we're going to be adding that into the HCP. That's an update that we're making, but it's going to cover all the maintenance, construction, and monitoring and management of the preserve that we're creating. So here is the preserve. It's roughly the preserve. We still have to go through, we have to take this through our just conversations with Fish and Wildlife. We're not done with that yet. So this is all draft. I want to make sure that everybody understands that. But this is kind of a rough depiction of what the preserve would look like. It is not final for those of you taking photos. These are the known features right now that are known to be occupied. We actually have more than that now. This is the geological assessment that we started with, and we are fully evaluating all the features on the entire campus, all the known features from the survey. And we are finishing up all of the survey work, the karst invertebrate survey work. We have new locations for Cichurina madla and new locations for Rhydione exilis, which is pretty exciting. What's unique about that is, since we have new locations in karst features that are developed all around, it's going to provide unique opportunities to evaluate what does that mean for those species in those features, and do some research that will help us find some clues toward recovery. We also did geophysical surveys, and we did a warbler survey in 2024. So we have, for the excavation and evaluation of additional features, we had 17 evaluated karst features and 3 caves. In addition, 10 closed depressions, 4 solution cavities, and 3 sinkholes that we excavated and evaluated. 5 required mechanical excavation, and then we also evaluated all the rest of the features in the GA. So we had mechanical excavation. We had to go through TCEQ. This is on the recharge zone. We had to get the full permissions to do that within the WPAP limits. So we established biological goals in our HCP. We want to permanently protect at least 3 caves known to contain listed species in a preserve that is 40+ acres in size. And this involves the management and monitoring of those features within the preserve. There's also a stream that goes through the preserve, so this discusses the protection of the riparian areas. And here are the institutional goals of the university. Maintain and enhance the biological resources, native biodiversity on the university lands, so that these resources can be utilized by future generations of students and faculty researchers. And I'll have you know, we worked very closely with the university. We are still working very closely with the university in developing these goals. These are not things that we just came up with as consultants. So it's very important that the university is getting out of this what they want to get out of it to accomplish their goals as well as accomplishing the species goals. Institutional goal 2, prepare a conservation program that incorporates sustainable land use planning policies and practices. 3, implement cost-effective conservation measures that efficiently invest the university's assets. And 4, define the university's legal responsibilities toward biological resources so that the university can develop its lands and operate an environmentally and fiscally responsible manner during the next 50 years. I don't know if that can be removed from future presentations, but you might want to consider it. Karst research and academics. So there, as a part of this HCP, we're setting up a karst lab. The idea is that this lab at the university is going to be able to employ graduate students, undergraduate students to actually teach them about conservation of caves, actually funnel potential employees into your firm. I mean, actually, you may actually be able to hire somebody that knows how to manage a cave preserve. Imagine that. And so, that's fantastic. And then, we also want to provide opportunities for research in these features as allowed, and that's obviously being discussed with the U.S. Fish and Wildlife Service. But we've been collaborating with professors on campus. We held a session. Lonnie can answer questions about that, how we've collaborated with the professors and how easy or difficult that has been. But we have collaborated with them, talked to them about potential research that they might want to be able to do. And we're going to be working with the service on that and with those professors in order to make sure that the people who are at the university are passionate about it and will carry it on and implement it. Here's some of the ideas that they came up with. Microbiology research, a geophysics lab, genetics research, senior seminars discussing the applicability of all of this, captive breeding with the zoo, population seeding of species, and partnerships with other institutions. The remnant caves across the UTSA KFR could also be used for research opportunities. If there are other remnant caves out there, those could be researched by the folks who are learning about that at UTSA. And then scientific information collected through the urban karst laboratory setup will be made available obviously to the Fish and Wildlife and to Parks and Wildlife for recovery purposes. So the UTSA professors, as I said, they presented comprehensive ideas, and then we're collaborating with the San Antonio Zoo. Dante's here. Dante Fenolio can answer any questions that you might have about potential collaboration with the zoo and their captive breeding programs. And then this is just the status and ideas for implementation. I know one of the things that we've been discussing. Oh, that's not correct. One of the things that we've been discussing is there is a problem right now with the SCP, thank you, with the SCP HCP not having enough credits. And so we have a credit shortage in Bexar County, and it's possible that UTSA will generate an overabundance of credits, more credits than they need for their development. And so if it's possible for us to generate additional mitigation through this UTSA HCP, then those credits may be purchasable through the SCP-HCP. So that's another idea we're exploring. But that's pretty much the whole presentation. And I've got Lonnie here and Dante to answer any questions you might have.
Yeah, we've got some time for questions. And if you want to repeat the questions into the mic, then the people online can hear. So the questions?
Yeah. No, they're not really that big. So the question was, are there any bats in these caves? Yeah. So the wooded area, it's more about the geology. And so if the geology in the area is cave bearing or potentially cave bearing based on what we know about that geologic unit, then we did those surveys. And so, there are karst stones that are delineated by professors that have been doing this for a very long time that indicate where we should be doing surveys, and that's where we did the surveys. Lots of research on that preceding this.
There are some surrounding areas around the campus that do have that. As a student, you may not have noticed, but there are several caves within the main part of the campus. There's one right on the end of the North Paseo building that's extremely large. And there are some that are near housing. So it's interesting that students walk by those every day and they don't know that they're right there.
Yeah, so there was a comprehensive geological assessment done on the property that's been resurveyed and resurveyed and resurveyed because it's recharge zone out there. So there's that as well. So, any back there? Actually, you can look up Stanford University's HCP. Lonnie and I just got back from visiting Stanford University. UTSA is collaborating with Stanford University to help with the success of their program. I think it's great to have those collaborations. We also just got back from the National HCP Conference, so more collaboration there. It's great to have, especially Lonnie's incredibly passionate about this project. It's so good to have people you're working with that are very passionate about making sure that it happens. There's a collaboration with Stanford, and they have a fantastic website, by the way. I encourage you to take a look at it. The Stanford HCP is online. All of their documents are online too. It's a great program. Mm-hmm. Any other questions? Anything for Dante? Donnie, I think there's 30 seconds if you want to come up and just talk about the captive breeding program you have and some of the, there have been other people making requests of you.
All right. Just short and skinny of it is one of the nice things that a zoo or an aquarium can bring to a conservation collaboration are opportunities to work with critically endangered species in the lab. At San Antonio Zoo's Center for Conservation Research, we work with subterranean species specifically. We were the first ever to breed things like Georgia blind salamander, Doherty plain cave crayfish. We're working with some other cave crayfish, but this is an opportunity where we might be able to coordinate students coming into zoo labs to gain some hands-on experience with captive work with critically endangered species.
All right, thank you very much. So our next presentation is by Malcolm McLeod. He's going to talk about the Texas Reservoir Ecosystem Networks database.
Good morning. My name is Malcolm McLeod. I'm a PhD candidate at Baylor University, Department of Biology. and with the Center for Reservoir and Aquatic Systems Research. And we've been working on doing a statewide inventory of Texas reservoirs funded by the Texas Commission on Environmental Quality. And because there is over 200 major reservoirs, they're really important water resources. Of course, here, 188 of those are major water supplies sources for both municipalities, also agriculture, critical for recreation. And although they're human constructed systems, we know that so many non-human species rely on them for habitat and other services. And there's a lot of data on reservoirs, but from a lot of different sources. So there's a need for consolidating this data so that water resource managers can be better informed in managing them for the challenges that we are facing. So some of these challenges are related to water quality. These photos are both from our local lake up in Waco, but reflect what we see across the state, some of these challenges. So with water quality, we have eutrophication, where we have really high levels of nutrients, which increase the trophic state index, which basically just means that we have high levels of nutrients in the summertime, especially phytoplankton blooms, which can deplete oxygen, cause fish kills, and other water quality issues, which are also amplified by water quantity issues. So throughout the state, we have had a reduced supply in our reservoirs, our reduced capacity, and this is just made worse by the increased severity and frequency of droughts. So here's one from our most recent drought up in Waco. So our project vision is to create a user-friendly database that consolidates physical, chemical, and biological data for Texas reservoirs. And we're doing this in 4 parts. So our trend, our Texas Reservoir Ecosystems Network database, includes a trophic state classification. It also has physical data, water quality data, and then the watershed geospatial data. And this all leads so that we can do our trend analysis. So this is where we, in having all of this data consolidated, can start doing some further data analysis and some recommendations, such as the environmental criteria for least disturbed conditions, and then also looking at some watershed to reservoir interactions to identify trophic state drivers. So which kind of data? Well, in the first part is where we have trophic state variables, also looking at how trophic state index has changed over time. And then that's from the Texas Commission for Environmental Quality, and then we geo-reference that with data from the National Hydrography data set, get some physical data like surface area, shoreline length, which leads to our next section where we have physical data, mainly from the Texas Water Development Board, but also some from the Army Corps of Engineers. So conservation, flood pool, elevations, capacity, then water level over time. So seeing how water level has changed. And then going to water quality in part 3. And here we're focused both temporally, so looking for the historic period, but then also spatially. So many of these reservoirs have multiple stations, and we have data from a lot of different sources, both at the surface and then for some variables at multiple depths, such as with oxygen, other chemical data, nutrients, and then biological data, primarily focused on phytoplankton productivity, but also fish. And then this going to part 4 is our watershed data, geospatial data. So again, with the National Hydrography data set, we've got our flow lines, but then also the National Land Cover database, and also looking at discharge from EPA's NPDES. And then Model My Watershed has modeled variables. So we're going to be combining all of these and have been doing this by first identifying which are the priority reservoirs. This was based off of TCEQ's Trophic State Report, which they do every 2 years. And 141 reservoirs were identified as the priority ones we're going to have the most data on. And then we develop a code to scrape specifically the TWDB websites where they have historic reservoir plates. That's where we get our time series data, capacity curves, a lot of data from the water data pages. And then we combining different data sources within R using the segment ID, standardizing our naming conventions. This is where our meticulous metadata really comes into play and the need for quality assurance that our team does. Delineating watersheds within GIS so that then we can download that data for the entire area for each of these watersheds. So some of these challenges that we face though in doing this, first is reconciling inconsistent data sources between reservoirs. So some reservoirs, large ones, deemed important, have a lot of data collected on them, and then some very small ones have very limited data. And besides different variables being collected, oftentimes, since we're working with a lot of different data sources, we might have the same variable from different sources. I give the example of drainage area there, or we can get from historic reservoir plates, from TDWV, or from StreamCat, and they might differ a little bit. And lastly, also applying functional approaches, since we're working with such big data, to non-standardized data. I'll give an example of that. And I know it's real small, but this is an example of a reservoir plate. So these are great because we're able to see historic data, right, for each of these reservoirs, but it's kind of non-standard. We can see, a drainage area, for example, but in this example, there's a proposed, there's an existing, there's some other nuances where we kind of have to, it just emphasizes the importance of our quality assurance. But once we have consolidated, we can start visualizing trend. So first, what we can see is, let's say with existing variables that we are compiling on our right over here, and we could plug in any two variables that you want, but here, just for an example, we have our drainage area on our Y, or surface area on our X, on a log scale, and being able to see what's the drainage area to surface area ratio for all of these lakes. But then we can also calculate some variables based off of what we have. So for example, on the other plot, we have mean depth. So we had the volume and the surface area for all of these. So we're able to calculate mean depth from that and then visualize what's the spatial trend across the state. And this is just an example of so much more that we can do. And we have, I'll be giving some ideas of what the future directions are, but just another idea of how we're incorporating some of this data is combining existing variables with some calculated variables. And here I'm going to highlight the trophic state variables. So first, for example, we have our calculated variable of mean depth, Secchi disk depth on our Y, and we can start seeing some patterns, right? And see that in the deepest lakes, for the most part, with one exception, in the deepest lakes, we have the highest Secchi depth, so the most transparency. Shallow lakes are less transparent. And start applying it to more variables. So up on the top, we have all of our three trophic state, and the variables going into the trophic state index: total phosphorus, Secchi, chlorophyll A. And then on the bottom, the trophic state index derived from each of those. So I'll just highlight, for example, with the total phosphorus one, we might not see it doesn't seem like there's any kind of relationship between mean depth and total phosphorus until then we look at the trophic state index derived from that variable, and we can see that indeed the deepest lakes have the lower trophic state index. And this is just, again, examples. There's so much more that we have been looking at and will be looking at. So that leads us to our future directions. And one of the big goals from this is to be able to inform the development of an index of biotic integrity. There's other states, places that have developed these, but we want this to be used for developing one specific to Texas reservoirs. We've also been how to use this database to identify reservoirs most at risk of impairment, also those that have suffered the most degradation over time, and calculating reduction in water supply statewide. So some work has been done showing that water supply has reduced over time, and we think that this resource can get to some more of the nuance and be able to quantify that better. But there's also some questions I have for you, and then I'll open it up for all of us. And that is, And, there's a lot more I didn't say, but from what I presented here, how can this statewide inventory aid your conservation efforts? Also would like to hear what missing component did you see that would make this tool more valuable for water resource managers? Since we're still in the process of compiling all of this, and any other thoughts you might have either now, or you can also connect with me later. There's my email, and I will open it up for questions and comments, and if you want to respond to any of that.
Questions?
Yeah. Yeah, so the question was that out west, they are planning to remove a lot of reservoirs, or some dams, right? And that if this could be used to evaluate whether they're useful. Hmm. Yeah, so I think that in part, assessing how useful it is depends on to who. So there's the economic valuation, sure. For example, is it useful for farmers? But then there's also the, which, I think we do have data on this of, okay, what was the cost of producing? There's also a lot of other ways we can value, is it useful? But if we see that there's, especially looking at watershed variables, I think that we could see, okay, what's the value that's being derived from this based off of, how much has the water level decreased, and is it really worth keeping that impoundment? I think that there is both mainly physical data that we could use to assess that, but also the economic aspect. The question being that in the observed reduced supply of reservoirs, have we identified any role of private landowners on them contributing to that by pumping out of the reservoir? We haven't specifically looked at how much water is being pumped out by private landowners. A lot of these are pretty regulated, but it's from a lot of different levels. So it could be from a river authority, or it could be the Army Corps. So a lot of the pumping is specifically to, through a permitting system. So, I think that with those that are permitted, that's something that could be added to be able to quantify how much is being pumped and where's that being allocated. Another thing we're interested though is that there's some private and some not point discharge sources, right? So not just pumping out, but how much wastewater is getting input, and is sure it might be permitted, but it might also be having a disproportionate effect. So I think that's other data that could be looked at for sure.
One more question.
One more. On data optimization? Standardization. Yeah, that's a great question. So the question being, is there criteria for standardizing the data? And when we get this data, for example, a lot of even just what I showed, we were getting some of the data in square kilometers, some of it's in acres. And so how do we decide which to use? we tend to go towards metric just because, I mean, we are looking at making this, you know, peer-reviewed research and putting it out in scientific journals. And so that's kind of our criteria, because different state agencies, different municipalities, and the federal government all have different ways that they you know, they might gather the data the same way, but the way it's kept is different. And so that's something we have to pay a lot of attention to, is making sure not just units are the same, but that even when we're talking about, you know, drainage, watershed, different terms, and making sure that we are, that it is the same. And so then that's where we go, and we have our team, and we have meetings where we say, okay, this is what we're going to name this specific variable, right? We're going to go with drainage area. And that's just, a lot of it is from the TCQ, what they were already using, but other ones that we've had to make some decisions based off of what we've seen in literature.
So our next presentation is by Sean McCracken from Bites to Biodiversity, Advancing Wildlife Conservation in South Texas.
Good morning, everyone. Wow, a lot of familiar faces that I haven't seen in a long time, so that's awesome. So the purpose of my talk this morning is really going to be to introduce you to the research projects that my team is working on in South Texas, okay? And it's about the wildlife conservation that we're doing and the conservation technology that we're applying to that. And I'll have several of my team members here today who are going to give you more details about some of the aspects of these different projects, okay? Let's see if I can, there we go. Yeah. So 4 main projects here in Texas. I'm doing similar work to these projects in the Pacific equatorial forests of Ecuador as well. So that's where some of my kind of comparison research methods come from for these projects. You can see we're pretty Texas-centric here. The first project is this assessment of the Ingleside sand sheet pond habitat that you'll get some more details on later today. But we're basically looking at all the species that occupy this habitat or use this habitat, except for invertebrates. And we're focusing on SGCNs primarily, but right, we're documenting everything. Next is going to be these 3 herp species in Texas. These are all state threatened species, so the Texas scarlet snake, the Texas tortoise, and the Texas diamondback terrapin. So let's start with the Ingleside sand sheet. We've been monitoring 36 ponds along the Ingleside sand sheet. This is in the coastal bend for about 3 years now. And the overall kind of, you know, objectives of this project is we're mapping all these ponds. These are really unique ponds. I'm going to save the details for Bria's talk next. And then we're compiling these species lists. And then we're going to generate these biodiversity metrics, right, and try to get an idea of what are the drivers, you know, that are influencing these communities, whether, you know, positive or negative. And then lastly, we'll do a functional diversity analysis, again, focusing on the SGCNs. This is, you know, kind of a new approach or newer approach in ecology to get a more comprehensive idea, right, of what's going on in these communities. I have a recently graduated master's student who took a first stab at this. First, I want to let you know, I do mostly canopy research in Ecuador. So when I proposed this project back in Texas, I wanted to look at the canopy naturally around these ponds. So we placed our camera traps and our automated recording devices and, you know, little microclimate monitors all in tree canopies surrounding these ponds. To date, we have, it's about, I think it's 169 species that we've documented from camera trap data. Remember, this is from the canopy. So you can see, right, we're getting birds, that's a rough green snake in the bottom middle there that you can't really see, you know, but kind of species across different taxa. We also, I forgot to mention, we now have about 1.9 million minutes of acoustic data that we're analyzing, as well as I think we're at 24,000 camera trap days so far. We use Wildlife Insights, so this is one of the conservation tools that we use for our camera trap data. It has pretty good machine learning models that do a fair job at identifying some species. They do a pretty good job. It does do a pretty good job of putting a nice red box around animals. So it makes it a lot easier to deal with, right, those blank camera trap photos. And then we use both kind of canned or pre-built convolutional neural network models or custom models that we're beginning to build now on the Arbomon platform or the BirdNet Analyzer platform to look at our acoustic data. And Albany Moon, who recently graduated with her master's degree from my lab, like I said, took a first stab at this. From the camera trap data, you can see mostly were birds. Remember, these camera traps are up in the canopy, right? So that's why we probably have mostly birds, but a fair representation of mammals and reptiles and a couple amphibians as well. She also did a detection technique analysis. She used a subset of our data. We obviously, we're good at keeping up with camera trap data. The acoustic data is a lot more time consuming, but you can see from our camera trap results, we have 10 unique species. And from our acoustic results, right, we have 73 unique species. So this really emphasizes what we kind of already know is the more tools we put in place to assess biodiversity, right, the more species we're going to detect. This is plug for Bria's talk next. She's going to tell you a lot more detail about the Ingleside sand sheet geological formation and the pond wetland habitat, specifically about her plant surveys, okay? Next was the Texas scarlet snake. I'm not going to say much about this because Tyler Bowling, Dr. Tyler Bowling, a postdoc in my lab, will be giving you the full details on what we got going on with Texas scarlet snake. I try not to say it in front of Tyler, but we hope this works. But we're doing species distribution modeling and trying to figure out how to actually observe the species in the wild. And then next, probably our biggest project is with Texas tortoises. And this is a really huge collaborative project. Jackie Tlemat, a PhD candidate in my lab, has really spearheaded a lot of this. We have a lot of partners working on this. I'm not going to read off all of these. These are kind of all the sponsored projects that we have and all the objectives of these projects, but they're all going into this last one, development of forfeiture and relocation program for the Texas tortoise. There are a lot of waif tortoises, lost tortoises. They're with rehabbers. They're with zoos. They get confiscated by U.S. Fish and Wildlife Service, by game wardens, and we don't know what to do with them. So we've done the population genomics. We're doing population genomics with collaborators at Arizona State and University of Alabama. We have the whole genome for the tortoise, and we're finishing up all the pop genomics work on that. Disease ecology is a big part of this. Jackie's done a lot of, or most all of the disease ecology work actually. But she won't be speaking about that today. I'll give you a little snippet of it. But we need to figure out how to get these tortoises back on the landscape so that they're not just stuck with rehabbers. And so that's what all of this is working towards, as well as outreach to tell people it's not okay when you're headed back from the beach for your kid to pick up the tortoise that's crossing the road and bring him home, okay? This is a plug for Jackie's talk. She's been developing machine learning models to detect tortoises in these large camera trap data sets. She'll be at 2:15 today. She's done a fantastic job of getting blood samples from across the range in Texas. Mexico's really challenging. I've been working on it. Not time to go into the details. We'll just leave it at it's really challenging. Some of just a quick glance at Jackie's disease work. So one of the issues is upper respiratory tract disease that is caused by this bacteria in the genus Mycoplasma. You can see these are fairly ubiquitous within where we've sampled in Texas. She's also looking at a fungal pathogen that causes necrotizing scute disease. This is really fascinating work. I'm sad we don't have time to go into this one some more today. But back to all of this goes into figuring out, how to relocate and translocate tortoises. And this is even more important with development that's happening in Texas, where organizations like like FERC is now requiring when they develop LNG sites to move the tortoises. Okay. Plug, sorry, I don't know why that's a little skewed there, but I do have a PhD opportunity starting in fall 2025 with Texas tortoises. So if you or anyone you know is interested, hit me up about that. Lastly, a little bit, we're working on Texas diamondback terrapins. I have never done any work in the ocean, but since I'm now at A&M Corpus Christi, I'm in the ocean because everyone else there is in the ocean. So we have projects that we're looking at we're trying to compile the historic distribution, but also identify the current distribution and where current populations are of terrapins. The species has been hit really hard. I can tell you, we're finding surprisingly few populations of terrapins. We're trying to get an idea of what their nesting habitat is, as well as we're doing some movement ecology work. Sorry, this map doesn't show up very well, but basically we're mapping all potential nesting habitat, and then we go out and we're doing eDNA surveys as well as head count surveys. We've been doing trapping, but trapping seems to be really inefficient. So we're hoping that these other methods pan out well for us. And then lastly, like I mentioned, we're starting to put GPS trackers on terrapins. We don't get a lot of data because they don't spend a lot of time at the surface. I think, I'm now on my third model of tracker, and we're starting to get some data. And what we're getting is really interesting. We're getting an idea of when they're coming out of brumation, how far they're moving. This is from one terrapin that first turned up in the Nueces River. So that does it for my talk today. Right, I'm happy to take any questions y'all may have, and please stick around for my students and postdoc who will give you some more details on these. And again, right, scan this if you're interested in a PhD on tortoises. You gotta love tortoises.
Time for a few questions.
All right, drop the mic, I'm out of here.
Thank you, Sean. Okay, the next presentation to follow up on that is Bria Marty, Unique Land, Unique Life, Rare Flora and Dynamic Diversity of the Ingleside Sandsheet Wetland Habitat.
Okay. Can you all hear me okay? Hear me all right? Hi, I'm Bria Marty. Thank you so much for coming to my talk. Thank you, Sean, for the introduction. I'm excited to talk to y'all about a couple aspects of my doctoral dissertation today. And yes, he already read the title, so I'll go ahead. A quick overview. So we're going to go over just a little bit of the background of the Ingleside Sandsheet, the significance of the Ingleside Sandsheet. I'm going to explain some of my research goals and project plans. And I'm going to share a little snapshot of some findings I have, as well as some rare plants I have found. So this is a snapshot from Corpus Christi, actually, in Flower Bluff. This land has a lot of deep sand communities, so very interesting vegetation growing there. But how did that happen, right? So we've got Texas. We know Texas. If you zoom in real far on the coast there, we have this really interesting stretch of land that was formed during the Pleistocene from various sources, either deltaic, coastal activity.
lots of ancient activity with newer sands overlaid up top of all of those features, which has resulted in a super unique landscape. So there is the Ingleside sand sheet, boom, right in the middle of all of that. And what this has done is created a very heterogeneous landscape. So we have a lot of high areas and low areas. Those low areas have filled with water and created depressional ponds. These are inland freshwater coastal wetlands. And there's also lots of lumpy, bumpy areas that we call pimple mounds there. This region is really comparable to the prairie pothole wetlands up in the Midwest, but these formed very differently. These formed from those coastal activities, wind activity, lots of deposits, whereas the prairie potholes were formed from glacial activity. So down below there, you can see a nice picture of one of the ponds that I took very, very early in the morning at Powderhorn Wildlife Management Area, which is in the very north part of my study region. So the Ingleside sand sheet is extremely important. I know probably none of y'all have actually heard of it. It is very, very understudied, but it is extremely important for a lot of animals we have in this area as well as plants. So just to name a few, including but not limited to, it is a biodiversity hotspot. It is located within a large biodiversity hotspot. That is the West Gulf Coastal Prairie, I believe, or coastal plains. It also is called the West Gulf Coast Grasslands. So it lies within that. So it is in a biodiversity hotspot. So lots of plants, lots of animals. This region also has a lot of vital habitat for SGCN. Wetlands are normally very highly biodiverse, lots of plant animals. This habitat is extremely important. SGCN is species of greatest conservation need, in case you need a reminder. And these wetlands in particular are extremely important for flood attenuation on the coast. So because wetlands act as a big sponge, they can help slow down water, they can help control where that goes. And in particular, these are very important for the region. And a big part of this is that they are understudied. We just don't know a whole lot about them, and we need to know more in order to conserve these wetlands. Here are a few SGCN that we have been focusing on. In particular, we do have a research project that's looking at this black-spotted newt here, which is classified as vulnerable. Now, with Albany's research, she's looking at a lot of the birds and stuff. One of the known SGCNs that uses the habitat, especially in Aransas National Wildlife Refuge, is the whooping crane. And then on the right there is a Texas quillwort, which is a very rare fern relative that was recently discovered in 2011 and has only been found in 2 counties so far, including that exact pond in the background I found that plant at. We'll get more into that in a little bit. So a couple of my project goals, this is not my entire dissertation, but this is part of it, a big part of it. So some of my goals are to establish this baseline data on floral diversity across space and season. So because we don't really know what's there, I'm going out and surveying the plants and checking that out and seeing if there's any differences, because the sand sheet is kind of a long and narrow strip that goes from southwest to northeast. I'm going to see if there's any differences there, as well as across seasons with the growing seasons. And then I'm also going to look at how human activity impacts the floral communities as well as management, because where these ponds are located are on several different types of land that are owned by different people and have different management techniques. So I'm going to look and see if that has an influence on the floral communities. Here's a really cool community that is imperiled. So habitat loss, as well as there is a beetle that attacks the red bay in particular. But this Texas Coastal Bend Live Oak Red Bay community is a threatened community that is listed, it is classified as S1G1, which means it's imperiled at state and global levels. That is found out in the sand sheet. These are my photos of being out there and looking at these communities. And then here's the Texas quillwort. So this plant is again ancient. It reproduces with spores. It is part of the genus Isotes, which is the only remaining genus in the family Isotaceae. And I found it in Rockport with the state botanist who first found it for me and showed it to me. And I was like, oh my goodness, this plant is very nondescript and very small. How am I going to find it on my own? But I did actually in March of 2024 out in a Powderhorn Ranch, Powderhorn Wildlife Management Area. I found it out there, a few individuals. And again, this one is also imperiled S1G1. It's a very, very cool species that I have gotten very into through this project. Zooming back to the Ingleside sand sheet. So here are my general regions that I am studying at. So I've got Corpus Christi, South Rockport, and North Rockport. So I'm distinguishing those because they are on opposite sides of that bridge going across there. Ostwell, which is Aransas National Wildlife Refuge, and Port O'Connor, which is where Powderhorn Wildlife Management Area. And of those 36 ponds that we're studying, I have 25 ponds included in my study that I'm looking at over 2 years. So that's 2 spring seasons and 2 fall seasons. It's going to be for the first chapter of my dissertation, the big floral chapter. I'm really looking at some basic things, including richness and diversity. I'm trying to see if there's spatial differences, seasonal differences, environmental drivers behind either of those things. And this is generally how I do my sampling. So at each of those 25 ponds, I have 20 quadrats that I lay out in this kind of spiral fashion where I'm able to capture each of these zones, because wetlands, you know, these ponds in particular have very distinct floral communities based on that micro elevation. And you can see me looking very, very closely, probably at a particular small plant that I'm trying to identify. But up there, you can see up here, the low prairie, this is the farthest away from the pond, where you have a lot of plants that cannot tolerate as much water. And then you've got the wet meadow, which is more grasses. It's typically kind of muddy. It's got a lot of sedges. And then the shallow marsh and open water area, where you have a lot more hydrophytes that really like water. For my other chapter, I'm looking at management and how human activity and disturbance impacts these plant communities. So we know that the human impacts around the area are driving habitat loss through urbanization, rapid urbanization down there, agriculture, climate change, of course, rising sea levels, as well as drought is a big one. But these ponds are all managed differently. Some of them have grazing, some of them undergo prescribed burns. Some of them allow recreational activities where people can go out and hunt or bird and hike and things like that. So I'm interested to see if there's an influence in the plant communities depending on how close they are to those disturbances, or if differences in management regime will show differences in plant communities. This is at Powderhorn Wildlife Management Area as I was coming in one day, and I had to get the cows out of the way and move along. Quick biodiversity snapshot. So this is just from last year. So spring and fall, I picked 3 cities right in the middle. So Corpus Christi is on the south end of the southwest end of the Ingleside Sand Sheet, Rockport's right in the middle, and Port O'Connor is on that north end. So I got some decently high diversity going on here. I was expecting a pattern where you would see higher diversity in the spring versus fall, because that for some reason that's like an old wives' tale that's going around. Yeah, the spring is more diverse than the fall. Not sure about that, so I guess we will see, right? But here we can see that Port O'Connor has definitely the highest diversity, and in the spring as well, compared to Corpus Christi. And I have a feeling that ties back to management, people recreating around, and then, yeah, proximity to those disturbances. Powderhorn is pretty isolated. You have to get permission to go out there. They don't just let people walk in anytime. So I think that might have something to do with it. And then just some basic richness to report here. So Corpus Christi in the spring, I found 26 species at Pond 2, which is in Held Moran Nature Sanctuary, which is owned by the Audubon Club. And then in the fall, I had 33 species. Rockport in the spring, I had 62 species with 43 in the fall. And in Port O'Connor, there was 76 species and 68 in the fall, 76 in the spring. Some cool findings. So these are some rare plants I have found and have put on iNaturalist. The waxy thoroughwort there on the left, apparently that is like as south and like southeast as far as it goes. It is at, or southwest is in the very, very corner of its range. So much so that there was a lot of discourse back and forth on why it was there. And then the middle one there is a subspecies of sleeping daisy that apparently has only been documented, I want to say 3 times, and mine was the 3rd. And again, lots of discourse back and forth of like, wow, this is a really random, and of course I just was literally walking along and I was like, what's this little yellow flower? You know, what's this little aster, for those who know DYA. And then I also found the quillwort and have put that on. I think it's only on iNaturalist like 6 times, and I think 3 of those are mine. But yes, interestingly enough, there was a researcher who was curious as to what was going on here because the plants didn't look reproductive. Fun fact, she ended up coming down and actually looking at these quillworts with me, which is very, very cool. And okay, so in October of last year, 7 months after I found it for the first time at that pond, I found a ton of quillwort, like an absurd amount, where I didn't actually know what was going on. And I sent a manic email to the state botanist and was like, is this quillwort? Because if so, there's an unbelievable amount of it here. And I know there's a lot of points on that map, but just these red points here is where I mapped the quillwort, which I had originally only really found it right here in this kind of corner of the pond, but I was finding it almost all the way around the whole pond. And a researcher from Houston came down, Lisa George, the one who had commented on my iNaturalist observation, came down and confirmed, yes, this is quillwort, and there is an absolute ton of it. So we were able to get some samples, and we were able to press some plants and everything with her permit, and I'm going to be mailing some of that to Canada pretty soon here. In summary, the Ingleside sand sheep pond habitat is valuable, but we know that it is definitely under threat from various things: climate change, people, urbanization, etc. We need this floral diversity baseline data to answer any more questions about what's going on. I do expect differences across the space and seasons, just knowing floristic seasons differ, as well as possibly rainfall patterns differing from the west to the east. And I am expecting influences of the distance to structures and the management regime to have an impact on these floral communities. I'm assuming that impact will be, the closer to disturbance and different types of management, possibly the lower in quality these floral communities are. And I do want to thank Texas Parks and Wildlife. They funded this project and they made this all possible. So huge shout out to them. And with that, I will take questions.
Time for a few questions.
That's a great question. So the question was, when I found those rare plants, were they located in disturbed regions, burned regions? Are you asking about just various management of where these plants were? The answer is yes. No, it doesn't make much sense. The waxy thoroughwort was found in the middle of Aransas National Wildlife Refuge, not near any burns as far as I know. In that area, at least, there was no signs of recent burns. The sleeping daisy was found in a literal like a fire break that was like tilled and turned over at it was a North American Land Trust property in Rockport, and I was just walking back to my car and was like, oh, look at this one's maybe different. And sure enough, that's where that was, just in kind of a tilled area. And then the quillwort was in a pond that you have to drive very far to, and there were no cows. I found one small pig wallow in there, and that one is managed very tightly by the wildlife management area. So it didn't really make much sense. I do expect to see more of these rare plants out in the more remote areas, like in Aransas National Wildlife Refuge, where we have to go behind gates, where we have special access to get back there. It's not off the auto driving loop where everybody can go. So that is what I'm expecting more. But I would say that would make sense if they were, but no, there hasn't so far been much of a pattern with that. Mm-hmm. Yeah, so I am going to be looking at that. I'm looking at prescribed burning. I'm not going to necessarily look at herbicide, but it is something that we can do as far as, whether that has happened or not. The difference is that a lot of the other ponds that I've looked at are on like private property or other like conservation trusts, and they don't all do herbiciding or burning. But Aransas also does a lot of burning. Powderhorn does a lot of burning. Powderhorn has burned several of our places where sometimes we've also had to coordinate like getting our cameras out of there, etc. So yeah, I hope that answered the question. Thank you. Thank you so much.
Okay, we're going to take a 15-minute break. Just a reminder that the refreshments downstairs, if you have a poster to put up and you need to know the number of it, see me and I'll help you figure out where your poster goes. We'll meet back at 10:45. Okay, we're going to get started again. So if you'll take a seat. Hey, Russ. All right, we're going to get going again. So I have a number of people who are helping to judge the student posters. The poster session is tomorrow, but if you're a judge and you'd like to get the forms to go ahead and look at the posters today while they're up, see me. I have the judging forms, but we'll do most of the judging tomorrow, but you can take a look at those ahead of time if you would like. So our next presentation is by Russell Fowl, The Cow, the Spider, and the Bees, How Citizen Science Has Enhanced Our Knowledge of Biodiversity.
All right, thank you all. So what I want to do here today is give 3 case studies of projects that I've been involved in recently, that I'm currently involved in, that involve citizen science data. First, I'm going to start out with a quick survey. Raise your hand if you have an iNaturalist account and use it. Okay, so that looks like a little bit more than 50%, maybe around 60% of you. That's kind of what I was expecting to see. For those of you that might not be familiar with it, it's a online platform where individuals can make observations, usually in the form of a photograph, but it can be audio recording or something else, and post that. And then everybody else globally can see those observations and can participate in identifying them or discussing them if there's something unusual. And that contributes to biodiversity science. So all of those observations, once they're identified by a couple of folks, get uploaded into GBIF, the Global Biodiversity Information Facility, along with museum data, global museum data. So you can go to GBIF and access a tremendous amount of biodiversity data. These are the observations globally on iNaturalist as of around last week or so. So you can see the number of observations there at the top, over almost 225 million observations of 503 species. And there's over 400,000 people who have posted identifications of those organisms, and then about 3.5 million observers. This is my personal iNat experience. So I was nagged by a former student whose name is Sam. He nagged me for about a year to sign up for this, and I didn't really see much value in it or anything. So I finally caved in, got an account in fall of 2015, and I made 2 observations that year. The next year, I made about 600 observations. So now I'm up to about 19,000 observations, and I've identified things for other people for about 74,000. If that seems like a lot, it's not. There are a lot of people with way more observations and identifications than that. When I was putting this presentation together, I thought it would be sort of about serendipity, about the role of chance in discovering things, and that's true, as you'll see. But without iNaturalist, those chance encounters with nature would not have really led to anything, and none of this would have happened, I don't think. So I'm going to start out with the plant. In March of 2019, I was walking around my parents' 80-acre property up in the rolling plains, and I was just looking for things to photograph for iNaturalist, and I was really the only thing blooming then were the anemones, which are a little small wildflower that has a white or blue purplish flower. And I had seen several of them, and then I walked up on this one, and I was like, this one looks different than all of the other ones. And I was just now beginning to identify these. There's 5 species in the state. I was just now beginning to learn how to identify them myself. And so I took a picture of it, and I evidently thought it was significant enough that I'd take a plant and make an herbarium specimen. It's like the first herbarium specimen I made since I was a sophomore undergraduate student. But I promptly forgot about it. Never thought about it again until about the same time, 2020, when somebody posted an observation of an anemone out in Lubbock. And I was like, well, that looks different from all of the other ones I'm accustomed to. And then kind of light bulbs and connections went off, and I was like, that looks like the one I found, which is different from all of the others. By that time, I was much better at distinguishing the species. The species can be distinguished can be distinguished by shape of the leaf and the fuzziness of the stem, little minor characteristics, the sum total of which allow you to distinguish them. All right, so spoiler alert. This is, I'm pretty sure now, an undescribed species, and I'm not a botanist, so this is really weird for me to be discovering a new species here. Tentatively, we're calling it Anemone pilosa because it's the fuzziest of all of the species. Here's a couple of additional photos of it. It comes in a white form and a blue form. These are both on my parents' property, just side by side, so it's just a color polymorphism. This is iNaturalist observations of the 5 species that occur in Texas, and they're all in the subsection Carolini. The pink one here that's kind of widely distributed, if you have a yard, you probably have it in your yard. It's just a common yard weed. I'm going to remove the pink species to show you the ones that are hidden below those. So the green one here is endemic to the limestone outcrops, Saloma baconis escarpment there on the hill country. There's a eastern species and then a couple of species out west. The blue species there is found only in the El Paso area, Hueco Tank State Park. The purple species is the most recently described species. That species was described in mid-1990s. And by the end of that year, somebody had come along and said, no, that's not a new species. So there's been some controversy about that one. The anemone pilosa, we have, when I say we, I mean the citizen scientists, the global we, have found it in the yellow oval areas there. Just last week, a population was discovered here. And a person posted it on iNaturalist and contacted me, and I'm like, well, that's really weird. Way out there on the east side where I wasn't expecting it. And those are the observations that folks have made of the species. And these are mostly observations by people that really don't know anything about anemones at all, just ordinary citizens who ran across it on their property and posted a picture. And I'm like, yep, that's it. So I got to wondering, well, maybe people have been seeing this because it's not that uncommon. And so I dug into the online herbarium database and looked at some pressed specimens, and voila. So there it is, collected in 1936. To give you a historical little tidbit here, this was collected near Jericho Gap, which this is a stretch of Route 66 that had not yet been paved. People, right, so this was a long time ago. It was actually paved the year that this was collected. So this is a specimen at the New York Botanical Garden, and it was identified as Anemone caroliniana, and it's clearly not. And then I found several other specimens. This one was originally identified as a species that's no longer recognized. That's not a valid name anymore. And then it was updated in 2002 as Anemone burlandii, which it's clearly not. No offense to the person that identified it, all right? What I'm saying here is it's gone unrecognized for a long, long, long time. So I'm a geneticist, and I'm like, we got to see what the DNA says. And so me and a student have been sequencing DNA from all 5 species within this section. And sure enough, it's genetically distinct from all of the other 5 species. And the controversial species here is also distinct as well. So we'll be able to fix that taxonomic problem also. I was going to warn y'all, if you're afraid of spiders, you might want to close your eyes. So apologies that I forgot to do that. Surprise. So so second case study. This is male on the right, female on the left. And it's what we think is an undescribed species, which we're calling Hogna incognita because it's widely distributed. It's very common. If you have a yard with grass, chances are you probably have it on your property. It was discovered by an amateur naturalist, not a professional araneologist, in 2019. And I've been working with him to learn more about this and describe it as a new species. So here's one species on the right and one on the left. And it took me several months, I think, to where I really became comfortable enough that when I caught one of these, I could tell which one it was. So the new species is the one on the left. Very, very, very similar, which is probably why they've gone unnoticed. Flip them over and look behind their knees. The new species is black, and that one's tan. That's the most striking difference between those two species. So I'm a geneticist. I got to see what the DNA says. And so we sequenced DNA from both of the two species. And sure enough, the ones that we thought were the new species are genetically different from the others. So let's see. Third case study is bees. Prior to me getting involved in this study, bumblebees were large bees that were black and yellow, and that's all I knew. Through iNaturalist, I was able to figure out that there's more than one species, and it took me a while to figure out how to distinguish between these two. One's darker yellow and has a little yellow band there between the wings that the other one doesn't have. So I got interested in bumblebees through iNaturalist and helped start to identify a lot of them. There were a lot of misidentifications on iNaturalist at that time. And so I put together a guide that I shared with the iNaturalist community. And lots of folks started participating and identifying them correctly. And so we got a lot of that updated. And then I learned that some people think that these are just color variations of the same species. And I'm like, I don't think so. I'm seeing them both in my front yard on the flowers. So I'm like, I'm a geneticist. I can figure this out. And so I teamed up with Jessica Beckham and Jeff Johnson. And we just got this published this year, just a couple of months ago. And so we used mitochondrial DNA sequencing. That's what I did. And they did the nuclear genetic analysis. And I helped analyze those data. And then we combined that with also the color pattern, which you can see the distinct color pattern there. So our research question was, are the two forms distinct species? And they occur together, they overlap, their geographic distributions overlap across about 2/3 of the state of Texas. So it's a broad area of symmetry where they overlap. And so we were interested in, do they just grade from one into the other? Do they hybridize a lot? Do they hybridize occasionally or none at all? And so we were able to sort of use the biological species concept, since they do overlap and they could reproduce if they have the opportunity to reproduce with one another if they were capable of doing so. And so the iNaturalist part here is, well, where do the two species or forms actually occur? There was a publication in 2012 that put together the museum records and had a map of where the two species occur, but we knew that that was really limited. And it's a lot of work to go to museums nationwide to figure out where a species occurs. And you can just log on to iNaturalist, and bumblebees are a really hot item to be photographed. And so there's a lot of observations of them. In fact, well, there's the numbers there. So about 12,000 observations of Bombus pennsylvanicus in the state, and about 2,000 observations of sonorus in the state. And because we needed to look at the color patterns to make sure the color patterns corresponded with the genetics and that sort of thing, I personally looked at all 14,000 or so observations of those. You might think that takes a lot of time, but imagine going to every museum that has them in their collection and looking at them, right? So that's not a big effort at all, really. Just took a few months. So the nuclear rad-seq data show them to be very distinct. We see the Pennsylvanicus there. Each dot is an individual bumblebee that was sampled, and so they all grouped together, and pretty significant difference with the sonorous there on the other side. And so we looked at the mitochondrial data, and again, we see a very distinct difference. And in terms of the color morphology, they match up exactly as well. So the color morphology, the mitochondrial DNA and the nuclear DNA perfectly match. So again, that wouldn't have been possible. Well, it would have been possible, but it would have been very difficult to know where those species actually occur. And so we know now through unnaturalist data that right between Dallas and Fort Worth is their eastern limit for sonorous, which is in yellow, and then sort of the Balcones escarpment, which is really interesting because there's a lot of people observing in this area, and there's a very sharp line right on that Balcones escarpment limiting. So I'm not sure why. Why don't the bees want to? It must have something to do with habitat, but the habitat there is very different than anywhere else. So that's an open question there. We did find an error in one of the museum collections. They had documented Bombus sonorus up here, and I went over my time. I'm very sorry. All right, so anyway, that's the end of the presentation, actually. So I'm going to stop there.
Thank you very much, Russell. So if you have some questions, catch Russell at lunch. So our next presentation is by Kelsey Swind, Considering Landforms for Species, Mapping Big Tooth Maple Canyons.
All right, good morning, everyone. Again, my name is Kelsey Schwind. I am with the Landscape Ecology Program at Texas Parks and Wildlife Department. I have a spatial sciences background, and I'm excited to talk about how we've incorporated that into our Big Tooth Maple project. And before diving into that, I do want to talk a little bit about the program itself and kind of the context in which we play within the agency. So the Landscape Ecology Program is tasked with analyzing, providing, and disseminating spatial ecologically focused data at the landscape scale. So this helps the agency to make better informed data-driven conservation decisions, as well as other stakeholders. And what's really cool about our data too is it's also most of it publicly available. And some of our projects, the largest one is the Ecological Mapping Systems of Texas. So has anyone here accessed and used this data? Okay, so a few people here. I encourage you to go look this up. You can access this, anyone can. And this is ultimately a land cover classification map at 10-meter resolution for the entire state. So Texas being such a large and diverse state, we have over 400 classes right now. We are always trying to add to this. And for each class, you can read a description, and it talks about the different vegetation communities you can expect to see there. And you can also access information like elevation, canopy height for shrubs and forest types, as well as soils and geology data. And this data and project ties specifically into harvest limits determined by the agency, as well as plays a role in species suitability modeling. One of my colleagues has been working on the Conservation Opportunity Area project, which she has developed a spatial tool that identifies areas that are determined by Parks and Wildlife to have potentially high conservation value for land acquisition. So we want to make sure if the opportunity comes up for land acquisition, we're ensuring it is the best land possible, or trying our best to make sure it's of good conservation use. As well as grassland quality modeling. So not only do we want to know this is a grassland on the landscape, but we want to know, is this an exotic grassland? Is this a native prairie? Because that's all an important, or that's important to consider in conservation as well. So now moving into the Big Tooth Maple project that we've been working on. So big-tooth maple communities in the Texas Hill Country are very isolated, and we have very limited data on this species here. So the ultimate goal of this project is to locate more communities to hopefully inform conservation decisions, as well as develop a better understanding of this species in the hopes that we can direct fieldwork efforts, find those communities, and then by means of landowner incentives or land preservation, help to preserve them in the future. We'll start out with what we do know about this species. It is a deciduous multi-stem tree, very beautiful in the fall months. It's not very common in Texas, but as I mentioned, we see isolated communities in the Hill Country, specifically the Edwards or Plateau and Balcones Escarpment portion of that. And then we'll jump back to this, but based on current observations, we do know that it prefers deep limestone canyons. So I say we'll come back to that. So we know that it does prefer canyons. All observations that we do have, even though they're limited, point to this. And we do know that topography inherently affects landscape ecology, particularly when we're discussing vegetation communities, but this really is not incorporated very often into research or studies. So we wanted to ask ourselves, how can we incorporate canyons into this project to help better direct our field efforts to identify more communities, as well as how can we spatially map these features for not only this project, but other projects that we're working on as well. And what made this study both equally challenging and exciting is that I really tried hard to find canyon data of any kind early on. Geospatial canyon data, it just didn't exist. There was one other study I found, but where they automatically extracted them, it didn't really give much information. None of those were in the United States. So that made this project very exciting because we got to build it from scratch and create a novel workflow for this. So the study area, as I mentioned, our observations occur in the Edwards Plateau, specifically the Balcones Escarpment. This is an area of about 4.4 million acres in what we all know as the Texas Hill Country. This is a geologic normal fault zone. It's dominated by limestone. And I'm sure most of us are familiar with the Hill Country. So we have the canyons, rolling hills, a lot of those karst features because of the limestone. And then again, most of the observations are in this region, specifically Lost Maple State Natural Area. So that is one of the bigger limitations of this study is the vast majority of observations we do have is from that state natural area. And just a fun fact, so Georgetown is partially included. So not only does it lie on the eastern-western edge of the Balcones Escarpment, but also on the northern. So the southwestern portion of Georgetown is included within the study area. The first step of this was acquiring elevation data. So essentially what I needed for the entire study area was a seamless elevation model. For those of you who are not aware, the Texas Geographic Information Office is the agency tasked with compiling geospatial data for the state, and it provides this to the public. So if ever you need this data, there's LiDAR data, elevation data, there's a lot of data out there. But I specifically was interested in digital elevation models, or DEMs. These are derived from airborne LiDAR surveys at 1 meter resolution, so very high resolution when you're talking about the landscape scale. I was able to download those and again generate that seamless elevation model for the area, and then using that elevation model, create a slope map. So at this point, I have my elevation model shown up here and a slope map. And then moving on to identifying canyons. So I can easily look at a map and see where these canyons are, but we needed a way to automatically extract these. For an area of 4.4 million acres, it's just not possible to go through and digitize each of those canyons. So first question here was, what even qualifies as a canyon? And this was just a lot of visual interpretation. So looking at the maps, my supervisor and I decided that this would be a slope threshold of 30 degrees. So if the canyon walls were 30 degrees or higher, that was a canyon of interest for trying to identify maple communities, and it would therefore move forward in the study. And again, I'm looking at the maps. I can see what is a canyon, what's not, even using that slope map, but we needed a way to spatially in the software identify and mark each of these. And we did this through some pretty simple hydrologic modeling using GIS to generate stream networks. Now, these stream networks may or may not represent where there's actually a stream present, but what it does is consider if there's a precipitation event, where that would accumulate. And as you can see in the top image, it did a pretty good job of identifying the bottoms of those canyons for us. So that kind of gave us a starting place. And then from there, all we needed to do was just buffer and see that if within a certain buffer distance, the slope threshold was met, then we knew this is a canyon of interest, it meets that threshold, and it moved forward for the mapping. And at this point, we have our stream networks for potential canyons, but for the purposes of other projects as well, what we needed were polygons representing the entirety of the canyon. So we needed the entire canyon to the ridges. And this is a very interesting tool a colleague referred me to, which is called the GIS Geomorphon tool. This is available in ArcGIS Pro, or there's also a free version. There's a Grass GIS plugin for QGIS that you can use. And this is a really cool tool. What it does is you just feed it an elevation model, and it looks at how the elevation for each cell differs from one another and the patterns of how they shift, and it will output a landform feature map for you. So input an elevation model, and it's just going to output a landform feature map, as you can see here, of one of these different 10 types for the entire region. So flat, ridge, shoulder, slope, depression. And this is what essentially allowed us to not only have the streams representing canyons, but continue to capture all the features of interest up to the ridges. This is an example of what that looks like. So we just have a topographic map on the left side here of Kendall County, which falls within the study area for the project. And then on the right, this is what it looks like after I used that Geomorphon app or tool, sorry. And this is another visualization of that. So step 1, we have our stream. 2, in black, you see slope that is 30 degrees or higher. So we know that this is a canyon of interest. And then 3, 4, 5, we're just seeing the models pool those different geomorphons and features out to the ridges. So 6 represents kind of a cleaned up version of the final canyon. Our results here, over 10,000 canyons were extracted within that study area. And this represented an area of over 1.2 million acres. And just visually interpreting this, this is really all we have to go off of at this point. We're still trying to decide the best way to go ground truth this. But visual interpretation of the results indicate it seems very successful at capturing canyons within the study area. Challenges, the biggest challenge just being data, so much data. 1 meter resolution elevation model and a slope map for that entire 4.4 million acres. I have a lot of external hard drives, a lot of them. Multiple software were needed as well. And another challenge wasn't too big of an issue, but in urban environments especially, there was the extraction of things non-canyon, such as riverbeds that were highly eroded, industrial facilities, and this is because it's all based on that slope threshold. So if you have something with a high slope threshold, there's a chance it will be moved forward as a potential canyon. Additional work, so these canyons, in addition to other environmental variables that seem to be important for these maple communities, were used to model which were more likely to have maple communities. So we are going to have some ground truthing in the field, hopefully within this next year, to go out and see if we can actually identify some of those communities. And the hope being that we do find some, and that Parks and Wildlife biologists can work with their private landowners they have relationships with and see if anything can move forward in terms of conserving the species. And these canyons will also be used to update that ecological mapping systems of Texas map and provide some extra classes as well. And this is what the initial map or model output is for trying to predict where we might find other maple communities. You can see we do have observations there, both Lost Maples State Natural Area and the Kerr Wildlife Management Area. But we also have other regions that are showing they might have maple communities that we can go check out. And I want to leave with the question of, within your research or field of work, could incorporating the landscape or topography help in any way? So I know I've spoken with a biologist in the past, and he made the comment of maybe something like this could be used in West Texas to find ridges or other features where we might find new bat colonies. So with that, thank you. Here's my email. If you have any ideas on collaborating or just want to reach out with questions, please feel free to reach out.
Now, we have a few minutes for questions.
Yes. Yes, is the big tooth maple population Fort Hood included? Not that I am aware of, but I'm honestly not too sure. I didn't work on the predictive modeling side, so I can definitely find that out. Okay. Then I will leave that on our team. We have our lead ecologist. She's the one that was putting together the observations. But if you reach out, I'm happy to ask her that, and I would be interested myself. Thank you. Yes. Yes, I do believe we looked at solar insolation and it did not seem to be as important. Aspect does play a role, I'm pretty sure. And that's also something that we consider for the ecological mapping systems of Texas. So those are variables that we look into for these models.
Thank you very much.
Thank you.
So our next speaker is Kira Gangbin, using targeted poisoning of red imported fire ants to improve Texas horned lizard habitat.
All right, good morning, everybody. My name is Kyra Gangbin. I am a PhD candidate at TCU. But today I'm going to present to you the work I just did for my master's thesis at TCU. That involves some Texas horned lizards and red imported fire ants. So to start, I'd like to just introduce you to the Texas horned lizard. They are a desert, prairie, and scrubland dwelling species that used to occur through a lot of Texas. However, they've experienced a lot of population declines due to urbanization, agricultural development, and the invasion of the red imported fire ant. As a result, Texas Parks and Wildlife, Texas Zoos, and TCU have all partnered up to try and reintroduce Texas horned lizards into areas that they've disappeared from. So currently we are working on reintroducing horned lizards at Mason Mountain Wildlife Management Area, which is located in Central Texas and the Edwards Plateau vegetation region of the state. Since 2017, we've been able to release 2,066 hatchlings on site. And we've also been able to do a variety of studies here. One of the biggest and most, influential that we've done was a diet analysis on these horned lizards. We wanted to see what they were eating. And what we noticed is that adult horned lizards ate different things than the hatchlings. The adults would eat the large harvester ants, while the hatchlings would eat small native ants, as well as termites. In addition, neither adults nor hatchling lizards would eat fire ants. Not a good dietary substitute for these guys. So, I mentioned the red imported fire ant. I guess I should tell you a little bit about them. The red imported fire ant is an invasive species. They are a very aggressive ant. They have a very potent venom and they make very large colonies. Unfortunately, these guys do not mesh well with horned lizards. They directly impact horned lizards by directly predating upon them, but they also indirectly hurt horned lizards because they compete with the horned lizard's food source. These fire ants all those small native ants I was telling you about that are so important for hatchlings to eat, that's what these fire ants are competing with. They're pushing them off the landscape, leaving our horned lizards with nothing to eat, and then they end up starving. So we really want to find a way to poison fire ants. And some of you may be like, okay, Kyra, I get it. But I was at Home Depot the other day and I saw fire ant poison on the shelf. And you make a good point, but those manufacturers want you to think that those are only fire ant poisons, those poisons are actually just general ant poisons. So if I took that ant poison and I spread it all over my study site, I will kill some of those fire ants, but I'm also going to kill the small native ants that are important for those hatchlings horn lizards. So that brings in my research question. I wanted to design a targeted poisoning method that had two goals. One, I wanted to decrease the amount of fire ants on site, but two, I did not want it to harm those native ants, which I often refer to as hatchling prey, because that's what those hatchling horn lizards are eating. So the way I'm going to do this is I'm going to utilize a bait station poisoning strategy. So at 4 different plots at Mason Mountain, I'm going to put out 100 bait stations. At some of those sites, I'm just going to put out bait stations, no poison. Those are kind of our controls for the study. And at some of the other sites, which I consider my treated sites, I do put out poison. And I want to see if we can, in fact, reduce the amount of fire ants, but not hurt the native ants. So I keep talking about these bait stations, but I guess I should talk to you about what they look like. So, like I said, we have 100 bait stations at each plot. They are organized in a 10 by 10 grid with 5 meters spacing. At each point on this grid, what we do is we take an upside down deli cup lid and we put it on the ground and we put a piece of hot dog on it. And we leave it out in the environment for 30 minutes. What we're doing here is we're trying to get all of the ants that are underground to come show their faces. We want to look at them. So after that 30 minutes has elapsed, we check what kind of ants are there. Is there a harvester ant, a fire ant, or an other ant there? If at this point we are at a treated site and we notice that there are fire ants, what we will do then is apply a teaspoon of a granular ant poison called Amdro, and we sprinkle it directly on top of that hot dog chunk. This should mean that only fire ants should have access to that poison, because all of the bait stations that had native ants, we obviously did not give them poison. They just got a free hot dog lunch. After we apply the poison, we wait another 30 minutes, giving enough time for hopefully the ants to take the poison back to their colony, hopefully giving it to the queen ant. So you can kill the queen ant, you can kill a whole bunch of ants. After that, we come back with the actual deli cup lid itself, and we snap it onto that deli cup lid. This makes it so we can collect any poison that is left over, leaving no residual pesticides in the environment. So for this study, we did it once a month between the months of May and August, and we did this study in 2022 and 2023. This is how we applied poison to our sites, but we also wanted a way to assess how these ant communities are changing. So what we did is we put out pitfall traps. For those of you that aren't familiar, pitfall traps are a common method used in entomology where you take some kind of container and you bury it underground till the very edge of that container is flush with the soil surface. Inside this container, there's some kind of preservative, so ants or invertebrates that are crawling around, they fall into the container, they fall into a preservative, and I can ID what's inside. For this study, we have a 5 by 5 grid of pitfall traps, so 25 pitfall traps per plot, and they're 5 meters apart, located right in the middle of where we do our ant baitings. The container we use for our pitfall traps are test tubes. So we're burying these test tubes underground, and the reason we chose test tubes is because test tubes are big enough that ants will fall in, but they're small enough that my baby lizards will not. We leave these test tubes out in the environment for a total of 4 days, after which I go and collect them, and then I ID all of the ants that fell inside to genus. We do these pitfall traps also 4 times a year, and we do it 2 weeks after each ant baiting. So all of that pitfall trap data gets inputted into a generalized linear mixed model, and I know that sounds complicated, but all that allows me to do is to see how treatment type, so whether or not a site got poisoned or not, affects my data, and it also allows me to see how date or seasonality affects my data. For the purposes of this study, I ended up analyzing 2022 and 2023 data separately. And the reason I did that is because these 2 years were very different. 2022 was a lot drier and 2023 was a lot wetter. And that matters when you think about ant reproduction. Fire ants mate anytime it rains. So in 2023, there was just a lot more fire ants in the environment, and we didn't exactly know how that was going to affect the system. So we thought it was safer to make 2 separate models for each year. So starting with the 2022 pitfall trap data, what we found first for fire ants is that there were significantly less fire ants present at our treated sites. Essentially, anytime we put out poison, there were less fire ants. This is exactly what we wanted to see. It's suggesting that our poisoning was working. We also were curious about if these ants were experiencing some kind of seasonality. For fire ants in 2022, it looks like they don't. They seem to be equally active throughout the year. Next, we're going to look at hatchling prey, those important small native ants. And what we see for them in 2022 is there was no significant difference between their abundances between untreated sites and treated sites. What this means is that when we put out poison, it doesn't look like we hurt them in any way. That we have again met our second goal of that targeted poisoning method. We're not hurting those hatchling prey items. As far as seasonality goes, I know it's kind of hard to see, but at a p-value still of 0.01, these hatchling prey were actually more active earlier in the summer months versus later in the summer months. Next, we're going to talk about the 2023 data, what that pitfall trap data showed. We see a very similar story to what we saw before, or you'll see some graphs to what you saw before. First, when we look at fire ants, what we see is that there are significantly less fire ants at our treated sites versus our untreated sites, just like in 22, suggesting our poisoning efforts are working. We're reducing the number of fire ants on the landscape. As far as seasonality goes, what we see is that in this case, fire ants are more active earlier in the season versus later in the season. So we are seeing some kind of seasonality. For hatchling prey, what we see in 2023 was exactly what we were hoping not to see. What we see is that there's significantly less hatchling prey items at our treated sites. This means that it's possible that us putting out that poison did hurt their communities. I'll talk about this a little bit more in the next slide. But first, I want to just talk to you about the seasonality that these ants are experiencing. And for hatchling prey, again, we see that they are more active earlier in the summer months versus later in the summer months. So I realized I just showed you a bunch of really plain, boring graphs. So I wanted to summarize it in 3 quick bullet points. The first thing is it does seem like this targeting poisoning method reduces the abundance of fire ants. We saw that for both years. However, we saw conflicting results for hatchling prey, where we didn't hurt them in 2022, but we did in 2023. And so I think it's possible that either our poisoning method really did hurt them, or, like I said earlier, that since 2023 was so much more of a wet year, there were so many more fire ants present, and maybe just the presence of all those fire ants is what's causing that reduction. So we're actually going to continue the study in a couple more years, for a few more years, and we're going to see if this finding is consistent. But one other point I wanted to make is that ant abundance seems to decrease as the season progresses. So ants are more active earlier in the season. And this is important because if you have a limited amount of time, resources, and money, like we all do, what I would suggest to you is you should be poisoning for fire ants in those earlier summer months, because that's when more of the worker ants are going to be active and they'll be available to take the poison back to the colony. Conservation implications of this study is first, I do think that targeted baiting is a better method for poisoning than broadcast baiting, where you're putting poison everywhere because you're containerizing the baits. And I think this is going to be a lot more beneficial for your native ant communities. I can also see this type of poisoning being utilized around sensitive areas. For example, waterways, places we haven't necessarily been able to poison again. That's because it's like a pack in, pack out type of way of poisoning. You're not leaving any residual poison in the landscape. This type of poisoning also has a little bit lower of a cost because you're not putting pounds upon pounds of pesticides in the environment. Instead, you're only putting teaspoons of poison out at a time. But I will acknowledge it does have a bit more of a labor cost. And lastly, I do acknowledge that the ultimate test of these methods would be to see if this poisoning actually improves horned lizard survival. So that's what we're planning on doing. The study I just described was from 2022 to 2023. Now in the future, 2024 to 2025, we are going to be able to make those conclusions. We're releasing lizards in locations where we're doing these poisoning events. We're going to see how survival is different. And we've also improved the study in a lot of other ways. We've increased the number of plots, the plot sizes, and the amount of time that the poison's being left out. So with that, I just want to give a specific shout out to Texas Parks and Wildlife and the Fort Worth Zoo. They've been absolutely amazing collaborators, but I also have a lot of funding agencies and interns that have put their sweat into this project with me. Thank you, guys.
Question or two?
Yeah. So she asked in those pitfall traps if I was catching other invertebrates and if I saw any difference between treated and untreated sites. I did catch a lot of other invertebrates. We classified all other invertebrates to family, but we have not done any analyses yet to see if there's a difference, but we are looking to do that in the future. Okay, so what he asked is, we're trying to draw ants to a hot dog. If fire ants show up, will native ants show up afterwards, essentially, right? And so that usually doesn't happen. Since fire ants are so aggressive, if they get to the hot dog first, they will usually ward off all the other native ants and they stay there. There is some, I think it's like a 2% chance is what we calculated, or 2% of the times that it does happen, where hatchling prey items or native ants will come after fire ants, but it's really uncommon. Thank you.
So our next speaker is James Tracy. He's going to be talking about monarch butterfly roadkill reduction in Texas using TxDOT monarch flight diverters.
Thank you. Well, this is a project that's funded by TxDOT, and it's a collaboration between the Texas A&M Department of Entomology and the Texas A&M Transportation Institute. The background on this, probably a lot of you know the eastern monarch has been declining like 80% in the last 25 years or so, and it's about 6% per year, and that's an exponential decline curve, and doesn't look like it's slowing down. We've done surveys over 5 years from 2016 to 2020 in Texas that indicate that roadkill can represent up to 1 to 3% of the overwintering population, so that mortality. And it's only in certain years that we see this. It's about every other year when we see the 3% figure. The 3% is about half of the 6% annual decline, so that's a significant number there. Fish and Wildlife Service has just proposed the monarch for listing in December, and there's a 30-day or a 90-day comment period that ends in March, and then it'll be a 1-year determination whether it's going to be listed by 2025. This is some of the previous data that we did, a combination of funding from the Texas Controller's Office and TxDOT. And you see in these even numbered years, we were getting some very high roadkill, and not so much in the odd numbered years. We're not sure what's going on there, maybe some kind of biennial wind effect. But it can reach up to around 2.6, 2.5% of the population, overwintering population. And you see these big spots. These are hot spots that I'll talk more about that we started surveying along the coast in 2018, and we started picking up some there. The main hot spots are along Interstate 10 in West Texas, kind of between Sonora and Ozona. For several years, we've seen hot spots out there, and then along the coast, along the coastal flyways. What we think is going out in West Texas is they're following draws as they migrate through the arid areas, and where these cross the interstate is where we're seeing the hot spots. Along the coast, they follow causeways when they're crossing the bay, and that's where we see a lot of the hot spots there. And this is just another close-up here of these hot spots out in West Texas, and this Point Comfort and the Ozona Howard Draw hotspots are the main ones that I'm going to be talking about here. So what can be done about monarch roadkill? Well, there's actually been something successfully tried in Taiwan. They have a monarch, or it's a migratory butterfly. They're related to monarchs, called the purple crow butterfly. And they put up these nets. They migrate along this viaduct area, drainage area every year, and there's lots of numbers get killed on this bridge. So they put up these nets, and they actually counted the number of butterflies that made it across the bridge versus were roadkill, and they were able to reduce roadkill by 80 to 95%. So that was very successful there. So that's something that TxDOT then wanted to try. And so the goal of this study is to actually try using these flight diverters. They were installed at these two hotspot sites, one at Ozona and another one on Port Lavaca. And this is just kind of a concept figure of how this is supposed to work. So the Monarchs, they're flying, they hit that diverter, and they're diverted above the traffic, the height of the semis. And one of the things, of course, that we want to see is, does this actually work, or did they fly down, or did they stay up? That's one of the main things we're wanting to evaluate here. So with these flight diverters, there were 30% mesh tarps that we used. They're 30 feet from the roadway so that vehicles don't run into them. We actually had a vehicle crash into the netting at Port Lavaca, but they're breakaway poles, so the poles just broke away. And they're, I think what we have, 200 and 209 feet lengths at Howard Draw in West Texas. And then we had just one 316 feet one at Port Lavaca. They're 16 feet high. We did roadkill transects of 55 yards in front of the diverters and then beyond the diverters to try to see if we could detect any differences in roadkill. And we also stood around the diverters when we had good numbers of monarchs, and we were able to see what percentage were staying up above the roadway versus going down after they crossed over the diverters. And we correlated that with wind speed and direction. So this is the diverter at Howard Draw. One of the diverters, there's two there. And then this is a diverter at Port Lavaca on the state highway there. And this shows the placement of the two diverters at Howard Draw. So that's the draw there, and they're placed right on either side of the bridge that goes over the draw. And then we had these various transects that we set up around the diverter and beyond it. Same thing at Port Lavaca, we just had one diverter though. So at Howard Draw, the monarchs are usually flying north to south, so they're flying across this diverter here. Monarch flight in this area is more east to west. And so we put the diverter here so that when they flew across, they would go up over the road. That was the hope anyway. This is results from the first year. We have 2 years of data. We've really only analyzed the 2023 data, but I'll talk a little bit about the 2024. But we had very low roadkill in West Texas. Well, really in both years, it was kind of low roadkill in 55 yards. We maybe had one monarch in each roadkill transect. So we really couldn't tell much with those low numbers, versus the diverter and the non-diverter. Maybe it was a little lower around the diverter. We had the opposite at Port Lavaca. We also had low roadkill, a little bit higher though, up to 10, average of 10 monarchs in 55 yards. There was a little higher, possibly higher roadkill there, but it's not significant because the numbers are so low. Well, one thing we did notice is that if you had a high wind, sometimes the monarchs would get pinned to the diverter netting, but that was only temporary. They generally were able to, or they were always able to escape once the wind let up a little bit, but that might have weakened them a little bit. And we did see in 2023, some monarchs flew above the diverter, but we didn't really have large enough numbers to quantify much there. What we could quantify though was more how they were flying in relation to the wind. And so at Ozona, we saw the monarchs were flying where the wind sources were from the north to the south in relation to the diverter, which is good because the diverter is perpendicular to that wind. And so that's when the monarchs are going to be flying, following that wind to go south. Problem though at Port Lavaca was the wind directions were coming from the north-northeast, east-northeast, and they were parallel to the diverter. So that was, we'll see that's will cause a problem at this site because the monarch flight directions were mostly out of the north, as what you would expect in western Texas, and so they were crossing that diverter perpendicularly. Problem in Port Lavaca is they were flying east parallel to the diverter. So the diverter was not, could not be effective there because the monarchs were just flying along the road and they weren't encountering the diverter. So we had a problem with the wind there and the way things were at Port Lavaca. In 2024, we still analyzing this data. We did have a lot more monarchs at West Texas, but in Port Lavaca, we had a very unusual year where there was virtually no monarchs observed in the area, just a few stray ones here and there. We don't have any data from Port Lavaca this year, even though we had the nets up there. But we do have a lot of roadkill data from Howard Draw in 2024. There was much more monarchs. We found up to 53 roadkill monarchs in some of the transects. So that's one monarch per yard. And we also found high numbers of roosting. This is something that we had never seen before. Along the interstate, there were monarchs roosting in the trees, the little hackberry trees and juniper trees along the interstate beyond the draw, which presented a problem with that I'll talk about here. But they were up to 321 monarchs in some of these little trees, and they stayed for several weeks. It was unusual, not sure what was going on there. They may have been waiting for more favorable winds, which they never really got, so they eventually had to move on. And so the roadkill rates, what we noticed was that the roadkill rates were correlated with where these roosting trees were along the interstate, and the roosting trees were not where we had the nets. So that was a problem. There wasn't much roosting going on behind the nets, so we had a lot of roadkill away from the nets. Not much roadkill around the nets, but that really didn't tell us anything about the nets. But what we were able to tell, because we had such high numbers coming through, we were able to watch. One day we had, I think, 95% of the monarchs out of 21, when they topped that net, they stayed high and cleared 15 feet above the semis. But then another day, that was 16th of October, but another day on the 25th of October, only 42% of the monarchs stayed up high and cleared the semis. So what happened, we think, is as the day was favorable, they had light crosswinds, and so they were able to stay up. But when they had light headwinds, they went down. When they have headwinds, they will fly closer to the ground in order to progress better. So that's telling us that the flight diverters are only effective under certain wind conditions. And this is just showing some of the monarch roosting we saw along the interstate. This is in a juniper tree. Again, we would get up to, most of the roosts were maybe 50 or a dozen or so, but we had some over several hundred. This is a hackberry tree here that had a roost in there. And we also saw, what was interesting is along the interstate, there's this composite called brownfoot. There was patches of it. Monarchs were roosting in that. I can get this thing to just right along the interstate, and then as you progress off of it here, there was about 75 monarchs in this patch of brownfoot. It was about 9 in the morning. They came off of those roost trees and just started nectaring in mass. And about an hour later, they were all gone. They were replaced by other butterflies that covered it, fritillaries, Gulf fritillaries, and variegated fritillaries, and other things. And there was very few monarchs. So what we have is GPS points for all these areas where they were nectaring and roosting along the interstate. We're going to try to analyze that, try to figure out if there's something going on there. Maybe they're roosting around these nectaring sites. By the end of October, these plants had senesced to the point where they were not firing anymore, and then the monarchs moved on. So kind of an interesting year from that standpoint. Never seen anything like that along the interstate before. So I just want to conclude here that we had potentially lower roadkill in 2023 along the diverter at Ozona, but we definitely didn't see that in 2024. So we need to get some more data, see what's going on, especially with the monarchs and the flight directions. Right, but it looks like a real problem at Port Lavaca. The flight directions and the wind directions are not favorable for the diverter to be effective, but there is some potential effectiveness at Ozona. All right, any questions? Oh, I just wanted to acknowledge TxDOT on this. Yes. Okay, so he's asking whether we've seen roadkill, I think, with other organisms there. And yes, we get a lot of other, this year particularly at Ozona, we had a lot of snout butterflies and sleepy orange butterflies. There were a lot more of those than monarchs getting killed. And we record whenever we see a few birds and raccoons. Not sure that the net is obviously for the other butterflies. We don't think the net's probably helping those much. The monarchs are, because they tend to fly over the net and then drop down, where those monarchs probably stay up more, we think. No, we hadn't noticed anything. We have an area at the bottom of the net so the animals can get through, and the birds that we've just seen them perching sometimes on the poles, but that's about it.
Thank you very much. So our final talk this morning is by Joel Ledford, Species Limits of the Endangered Tooth Cave Spider in Central Texas.
Thank you for having me. My name is Joel Ledford. I'm from the University of California at Davis. And I should have forewarned you, I'm going to show you a few pictures of spiders. Too late. I want to start out by just telling you a little bit about the general biology of leptonetids. So these are small spiders. They're usually 2 to 4 millimeters in length. They build really fine, delicate sheet webs. Many species worldwide are closely associated with caves, and there's 2 listed species in Texas. I'll point out to you a couple of things here. One is they have kind of a peculiar eye arrangement here, and the males upon which most of the taxonomy has been based have kind of a distinctive swollen boxing glove-like look to their pedipalps. And if you're still unsure what a leptonetid is, you can just think of me, weird, short, pale skin, balding, with reduced eyesight, and you should have a good idea. My previous work on leptonetids was focused in large part on a revision of the taxonomy of them in Central Texas, and I did key in a lot on the listed species, and I used a combination of morphological data and DNA data from 4 different genes. And I discovered a lot of things. One of the things is I discovered that Myopica, which is the listed species, is sister to another species called Paracancina. I discovered that Paracancina not only occurs in caves, but more broadly on the surface. And then something that I kind of regret not emphasizing more in my original work was that there's a lot of morphological variation in these spiders. And so this is an example of the morphological variation in the listed species Myopica. And even to the untrained eye, you can see that some of them have more pigmentation than others. Some of them are clearly, super cave adapted like here, but if you compare it to here, it's very different look, right? You probably never knew that there are so many different shades of beige. But you get the idea. And this always kind of intrigued me, this variation. And so the questions that we've had over the past couple of years is, can we sort of go back and have a look at the taxonomy of Tationetta with specific focus on Myopica and Paracancina, and using some next generation sequencing, can we kind of tease apart and test the species boundaries between these 2? And so we do this in a very particular way. I'm not going to go through all of the nuts and bolts here, but we usually get specimens. We have to extract their DNA. We then send that DNA off to be sequenced off-site. And we work with these things called UCEs, or ultra conserved elements of DNA, which are highly conserved. And we pick the ones that have the best signal for the question that we're asking. We then use that data to build phylogenies so that we can infer evolutionary relationships among the specimens that we have. And then the other thing we do is we pull something called a single nucleotide polymorphism, or a SNP, out of the data that we collect. And you don't need to really know too much about what a SNP is, except that it's a really good marker to sort of track gene flow. And this is the same kind of data that if you have ever had your ancestry done, like a 23andMe, or if you ever had your dog, tested to find out what sort of mix it is, it's the same sort of data. Our sampling was largely based on previous work, but also included a number of new specimens. We did emphasize populations of Myopica and Paracancina, and we assembled 2 data sets. One of the data sets had a larger number of samples, but it was a little bit weaker, had more missing data. And then the second data set had fewer samples, but was more robust. And one of the things I'll point out is that my earlier work, I thought I was very fancy because I had 4 genes, but there's been a real revolution in the field, and now we routinely work between 800 to 1,200 different genes. So this is a really robust data. Our results right now, which I'm going to show you, this is part of the phylogeny that is based and focused on Myopica and Paracancina. I want to point out to you one really important thing, and that is that all of these sites are color-coded by county. So yellow is all Travis, and then the purple is Williamson. And you can see that we don't have quite as many samples in Williamson, but we do have samples that stretch most of the county, and we have some new samples from northern Williamson County, which I think are going to be very informative. Either way, additional sampling in Williamson County is going to be really important. And I'll also point out that we have a big gap in Bell County, obviously, as we approach Fort Cavazos, which is going to be important. So we kind of have 3 hypotheses that we're working with now to resolve taxonomy. The first one is the one I'm showing you here, where I've broken up Myopica and Paracancina largely along the lines that I had previously. I'll point out to you that the 2 red arrows are caves in Williamson County. So you have Myopica in Williamson County for sure. We even have a new site, which is Monarch Cave. Here, what I would say is that this is probably the most conservative approach to the taxonomy of Myopica and Paracancina right now, where both species are maintained, but there are not problems. There are some weird issues, one of which is Paracancina is not totally clustered together. We have the type locality for Paracancina, which falls outside of this group, which is on Fort Cavazos. And then we have a new record that is from way down south, this karst feature F65, that remains kind of a problem. But this could be resolved with some additional sampling. Now, another option that we've been playing with is that perhaps Paracancina is restricted only to those sites up in Bell County, and Myopica, therefore, is much broader ranging than previously expected. In order to test this a little bit more, we would definitely need more sampling in Williamson and particularly Bell County. And then the last hypothesis is one in which pretty much everything is Myopica. So this kind of turns everything on its head, where Myopica was previously only known from a few caves to now being a broad-ranging species that has lots of locally adapted cave populations. So what about the SNP data that I mentioned earlier? The thing I'll point out to you about this is that each colored dot represents a SNP here. And you don't really need to know too much about what's going on here, except that, well, the colors are not quite coming out, unfortunately. What these data are going to would show you is that if there are dots that are overlapping, then the dots overlapping reflect gene flow. If they're isolated, they reflect isolation and no gene flow. Unfortunately, these dots are not appearing very well here, but what I would tell you is that for a large data set that we have with 58 taxa and about 1,000 SNPs, there is gene flow between Myopica and Paracancina. And so using something like a biological species concept, you could therefore infer that because of recency of gene flow, there might be conspecific. If we remove things that have a lot of missing data, what we find instead is a little bit more isolation. But I would stress that this is probably an artifact of sampling. We have a lot of sampling gaps, and those things, once filled in, you might see evidence of more gene flow between all the populations. So in conclusion, a couple of things I think are important is one, incomplete sampling is probably having a negative effect on our results. And it would be great to get pretty much any samples from Williamson County, but especially up in Bell County, and additionally, new samples up in Fort Cavazos. I think that we've focused a lot of our sampling effort on caves, yet we know that these spiders live on the surface. And I think adapting and incorporating some surface sampling protocols, especially for cave monitoring and inventory efforts, is going to yield results not only for these spiders, but also for other cave invertebrates. Given the conflict that we're seeing right now, we're leaning more towards a conservative approach to the taxonomy, i.e. that hypothesis one that I'm telling you about, where we maintain both species. And last but not least, the troglodytic populations, we don't see any gene flow between them. So even if Myopa becomes a wide-ranging species, there are several populations which are in caves which are genetically distinct. And with that, I'll thank a lot of people that have given me a lot of help over the years, and happy to answer any questions that you may have.
We have time for some questions.
Kimball. Yeah, so the question is really, some of the other cave invertebrates, there are strong clusters, well-supported clusters of troglodytic species. In leptinetids, what do you see? Do you see something that's really broad-ranging and local adaptation to caves? And I would say that answer is yeah. We see the repeated evolution of troglodyty, and there is evidence for sure that there's gene flow between some caves, but I think with additional sampling, you would probably see gene flow between things that are even more on the surface or in less strongly, cave environments. And so I think that it's really a function of sampling. You already see this kind of a thing in other animals, like I know cavefish have shown some of these, some genetic exchange between surface and cave adapted populations. So I wouldn't be surprised if you see the same thing with spiders. Thank you for that question. right? Thank you.
Very much. So we're now going to take an hour break for lunch. There are lots of places to eat close by. We have a list back here of some places to eat. These are places that we've eaten at, that we know about, that we would recommend. So if you're looking for a place to eat, you might take a look at the list. We'll meet back at 1 o'clock.
So I will. Well, the good news is, that's, you don't know, but I love you, I know what.
You know, I got this.
That's right, that's right.
Excited. So yeah, so. Yeah, it really has gone.
Shoot my real much.
Yep, 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 we wouldn't be able to accommodate.
As cool as that, I think it was.
Yeah, that was always problematic.
I remember that. Say, yeah, that's what's up.
Yeah.
Well, it was good catching up. I'm gonna go grab a bite to eat and I'll see you a little later.
Travis?
Absolutely that company because it's not telling them to give in the narrative. And it was like this kind of fun. I was like, that is get people on it already together. I was like, what's the yeah, and it's like it's like it's it's more like not fitting it. Functional learning cases. Oh, God. Morning, yeah.
Your first thoughts. Okay, we can do your thing. Share your slide.
Thanks, sir. Yep.
And the, or the have they come on, Mary.
That's how that's probably done. Right. Yeah, that's what it might. All of these.
Lunch break. Yes, and you walk my fun.
I, I know for a fact there's
Not very far down.
right now.
So, I need to participate in the activity. Start and run there. About 30 minutes. Talk it about. Go. No chance. How bad it is happening. So you have.
Yeah.
Okay, folks, we're going to get started here. I'm going to try to keep on time. Please take a seat. So our first presenter this afternoon is David Ford, and he's going to be talking on status and distribution of 3 rare freshwater mussel species in Louisiana.
Hi, everyone. Hopefully you had a good lunch. As said, I am David Ford with Edge Engineering and Science, and I'm going to talk to you about a recent study we did in Louisiana. So as you're probably all aware by now, since mussels have kind of been a hot topic in Texas recently, freshwater mussels are a group of shelled aquatic invertebrates, which can be found nearly worldwide, really with just the exception of Antarctica. Otherwise, they're pretty much everywhere. They're a very, very diverse group. There's about 300 or so species here in North America. I'm actually trying to find and see them all before I end up dead eventually. That's one of my life goals. And they're really important for aquatic ecosystems. They actually alter the nutrients in a stream via their filter feeding and as they sequester nutrients both in their shells and in the substrate. They release nutrients as they move around, and then their burrows and their shells actually act as additional habitat for macroinvertebrates. So macroinvertebrate density and richness both increase. I think it's like 4 times as much when unionids are present. They also act as a food source for various organisms, but their main thing is really that they're the water filtration system of an aquatic system. They act just like the water plants that we built to basically replace these things. And like a lot of aquatic organisms, they're in trouble. About 70% of our North American species are considered to be imperiled to some degree. Partly this is due to their own complex life cycles, which involve using a fish host to reproduce. But invasive species like zebra mussels haven't helped, nor has our pollution of the various waterways or our alterations to it like dams and straightening rivers and whatnot. One of the biggest impacts that's more recurrent is climate change, as we all know. It's increasing the frequency and duration and strength of droughts. And when you're an aquatic organism and there's no water around, you're kind of in trouble. So that's going to get worse essentially as time goes on. Because of this, many states have started to instigate studies to basically see where their mussels are, determine how they're doing, and see how they've changed. And this includes both Texas and the state I'm going to talk about today, Louisiana. So Louisiana historically had somewhere around 65 or so mussel species, depending on which geneticist you're talking to that day. And about 40 of these are thought to still exist in Louisiana's waters. 33 of these have some kind of either state or federal protection, and quite a few of them that don't have any federal protections have been petitioned for those protections. Like Texas, large portions of Louisiana remain either very limitedly surveyed or completely unsurveyed, or the data is extremely old and not very useful anymore. And you'll see this here in a minute, there's a lot of older data that just isn't very useful. It's either just numbers with like a location, no, methods, none of that kind of stuff. So to help address this, we partnered with LDWF to essentially update the status on some of their rare mussels. We started with just a few. We did the Alabama hickory nut, the rayed creek shell, and the pyramid pigtoe, which are 3 that have been petitioned for federal protections. We had a couple of objectives with this. One was just, of course, to update the status and see if they were still present at some of those historical sites where they were found. We also wanted to find new locations where these species are And we wanted to compare our data from the historical sites with current data because a lot of it was pretty old. Finally, we just wanted to look at the health of the mussel assemblages that we ran into and just see how were mussels doing in Louisiana. So this is kind of a busy slide, but basically we got historical data from LDWF. We picked out 12 historical sites. Unfortunately, there wasn't a lot of historical data, so we actually ended up using one site for both Alabama hickory nut and Raid Creek shell, so we only had 11. And then we also went to 12 new sites that were either in the same system or had similar habitat. Now, what I want to point out here is a lot of this historical data is relatively old. You might be noticing there's like some 1970s, and even the newer stuff is like 20, 25 years old on its own. So a lot of this data is pretty out of date. And as much as the climate and environments are changing, as much as we're impacting it, data that old isn't super relevant or useful anymore. So we wanted to follow the same methods that those historical surveys had done to make them comparable. Unfortunately, most of what we got was, hey, here's the spot where they found them. We don't even know how many of them were found here. So we couldn't really do that. Instead, we followed Dinkins et al. 2022, which was a 2017 survey in Louisiana, and at least made it comparable with that. Basically, we'd go to a site, we'd measure out a 100-meter section, we'd spend 1 person hour surveying that bank to bank. After that first person hour, we would then figure out the location with the highest richness and density of mussels. And then we'd spend 3 additional person hours surveying there. That way we would ensure that we hopefully got all of the species and the most that we could find in that search time. We also did 12 quarter-meter quadrats just to get an idea of density. And we recorded some various habitat data like substrate and that kind of thing. Nothing too intense as far as our habitat. And then when we did get mussels, we'd count them, we'd measure their lengths, we'd record any dead shell species, and we'd record any invasive mollusks like zebras or corbicula. We calculated relative abundance, catch per unit effort, and density for all of our sites and overall, and then we compared those across sites and with our historical data where we could. We also calculated following Dinkins an abundance category, which basically just tells us how rare our species are. It's based off of person hours. And then for our target species, we calculated or created some length frequency histograms just to note recruitment cohorts essentially. And then we used, NMDS to, sorry about that, figure out if there are any species associations. Overall, we ended up spending 92 person hours at our 23 sites. They were all pretty similar. Substrate was basically just silt with a lot of trash and some gravel cobble in some places. Depths ranged considerably. We had to dive on some of them. We ended up with mussels at 18 of the 23 sites, and they varied considerably between sites, as you can see, 0 to almost a little over 700. And you might be noticing that both abundance and richness vary considerably across those. Our sites 1 through 8 are on the western side of the state, and then 9 through the rest of them are on the eastern side. And we're not entirely sure why they dropped so much. Habitat was somewhat different. It was kind of more loose gravel that's not usually suitable for freshwater mussels, but it wasn't that different. And we were relatively hitting the same kind of sites. So we're not entirely sure what's happening there. Overall, we ended up with about 4,800 animals of 33 species. Primarily, this consisted of like 3-ridge and bank climber, which were our dominant species. This is usually pretty typical in a mussel assemblage. You usually have 1 or 2 or 3 species that are dominant, and then everybody else is pretty rare and on the periphery in low numbers. And you can see from our abundance categories, that was kind of what occurred here. Most of our species were either rare or uncommon, and then we just had a few that were relatively abundant. We found pretty much everything in our time searches. That was more than 90% of our animals. And then in our quadrats, we did still get quite a few, which is really good because we had a good density. And if you've ever had to dig out quadrats for mussels, you know they get really boring when you don't find anything. It's really, really dull. So looking at our target species, we didn't find any of the Alabama hickory nut. We're not exactly sure why, because we went to those historical sites And we even went to new sites where they had recently been found in that same system. So like the Pearl River, for example. We also made sure to check the literature and went to the habitat types that are described for this species. So we don't really know what happened here. Maybe the initial historical records were misidentifications. We're not sure. Looking at pyramid pigtoe, we got 15 of these. Most of them came from one site in Bayou Macon, and then one was in the Tenhas. Both of these sites were historical sites. We did get a nice length range on these guys, so multiple cohorts. And then we got a couple of very small individuals, which would basically represent recruits. So they are, at least at Bayou Macon, doing relatively well. We found them, interestingly, both in our time searches and our quadrats, which was pretty cool. But what's kind of interesting is the length average for the quadrats was actually larger than that for the time searches. And usually for mussels, when you're digging quadrats, that you do that to find the smaller individuals. So it's kind of interesting that it's inverse here for some reason. There weren't really any species associations with them. They were just kind of with everybody in general. Looking at radiatus, we got 9 of these individuals from 3 sites, including one new site, which is good, but most of them came from one single historical site. I believe it was 6 from one site. They were found exclusively in silt during our time searches, and we got a pretty good length range again with these guys, indicating a couple of cohorts, but no recruits, unfortunately. They were also found with Velosa vibex, which is another sand-loving species as well. So in conclusion, we're going to continue this upcoming field season. We've got some additional species we're looking at. Overall, mussels appear to be doing okay. We did get them at most of our sites, both our historical and our new sites. And though it's difficult to really tell, especially since most of the historical data wasn't really useful, they seem to be doing okay, especially on that western side of the state. Again, we're not exactly sure why they were doing so much better over there. We were more targeting microhabitats specifically for Raid Creek shell and Alabama hickory nut on the eastern half, but I mean, it was still pretty similar, so I don't think that's really what it was. Another important thing was just that we saw signs of reproduction for most of our species. We got quite a few small individuals from various groups. Though we didn't find any federally listed species, we did get several state listed, and we found 2 of our 3 target species, which is really good. And though the abundances and our other variables for these species were relatively low, it was similar to other studies, and it may just be that these species have always been rare and been some of those periphery species like I mentioned earlier. Overall, though, our study basically just shows that despite losses and the difficulty in quantifying those losses, mussels are doing okay in Louisiana, and it highlights the importance of protecting those streams, especially on that western half of the state. So with that, I'd like to thank various individuals, and I will take any questions.
Okay, we're going to wait just a couple minutes, try to stay on schedule here. So we'll take about 2-minute break here before we start the next. Okay, our next speaker is Charles Lacray. He's going to be speaking on testing more individuals' hypothesis using freshwater mussel catch per unit effort among western Gulf slope drainages of Texas.
How's it going? My name's Colt. I'm a graduate student at Texas State University, and I'm in Dr. Bonner's lab. I want to start off by getting to just a little background. Prior to grad school, I had the opportunity to work with an environmental consulting firm known as BioWest for almost 2 years. And during my time there, I was involved in a lot of mussel work across the state of Texas. And one of the things that puzzled me the most was how abundant mussels are in East Texas compared to Central and West. While there are possible explanations that explain greater species richness in areas of Texas, there are limited research for greater abundances. Pieces of the puzzle started to come together when I came across a theory known as the more individuals hypothesis. This theory states that greater productivity allows for more individuals, while considering individuals and productivity are not homogeneously distributed. Caspery et al. 2000 found a positive relationship between net above ground productivity and abundance, as well as Evans et al. 2006 found a positive relationship between normalized difference vegetation index and migratory birds. Both net above ground productivity and normalized difference vegetation index are proxies of productivity. The more individuals hypothesis is a contemporary theory and has been supported across varying taxa, such as vertebrates and invertebrates. However, the theory has not been supported, and there are a few reasons why. The biggest caveat with the more individuals hypothesis is spatial scale. Spatial scale is dependent on the organism and their mobility. So for example, if you were to look at ants and their relationship with productivity, you would consider a smaller spatial scale. Whereas with birds, you would consider a larger spatial scale. Another caveat is trophic level diversity. It can cause predictions to not be met. Hulbert 2004 and Evans et al. 2005 used North American birds as their study organism and noticed that multi-trophic communities obscure the relationship between productivity and individuals. Lastly, ways in which you measure productivity can be challenging because the type of productivity is dependent on the trophic level of the organism that you're studying. Knowing these problems with the more individuals hypothesis, we can now ask the question, is the abundance of freshwater mussels related to productivity, and is this pattern consistent with the theory? By recognizing the caveats, we determine that freshwater mussels are a good study organism because they are widely distributed across Texas and they are on the same trophic level. So the purpose was to assess a relationship between freshwater mussel abundances and productivity. Our primary study objectives were to quantify catch per unit effort of freshwater mussels statewide and relate CPUE to a productivity gradient at the reach scale. We predicted that there is a relationship between CPUE and productivity, and that productivity will have the greater influence on freshwater mussel abundances than water quality and flow. Water quality and flow are known variables to have an impact on freshwater mussels, and so we predicted that there is a relationship between CPUE and water quality as well as CPUE and flow. That brings us to our methods. So we obtained mean CPUE of freshwater mussels from published and unpublished sources spanning from 2000 to 2023. That encompassed 9 basins, 660 sites, which combined into 42 reaches, and CPUE was calculated across all species for each reach of the 42 reaches. For productivity, this was definitely our biggest challenge, finding a measure of productivity that fits freshwater mussels. But freshwater productivity is primarily driven by nutrient enrichment from the surrounding land. So knowing that, we use the National Commodity Crop Productivity Index 3.0, which is a measure of soil fertility. The index considers the concentrations of 3 fundamental nutrients or elements, nitrogen, potassium, and phosphorus, and we calculated a productivity gradient or estimate for each of the 42 reaches. Water quality was pulled from the Texas Commission of Environmental Quality. We averaged 11 water quality variables for each of the 42 reaches. These variables came from Scheck et al. 2024, which was a recent publication on water quality and mussel tolerances. And using the United States Geological Services gauge stations, median flow was pulled for each reach. For our analyses, we used regression models with CPUE and productivity, as well as CPUE and flow. Furthermore, we used empirical principal component analysis with CPUE and water quality, and a principal component analysis with productivity, water quality, and flow. For our results, data compilation consisted of 49 species and almost 65,000 individuals. And right off the bat, we observed greater abundances of mussels in East Texas and fewer abundances central as you move from central to west. Sorry. We first wanted to look at productivity across the Western Gulf Slope drainages. And the way we set this up was from west to east. And we noticed that our Western rivers, Rio Grande, had lower productivity. And as we approached our Eastern rivers, Sabine and Red, it had more productivity. Moving forward, the next thing we wanted to do was look at productivity and our abundances. And we saw a positive linear relationship. The next thing we did was ran a PCA with our water quality and CPUE. PC1 explained 37% of total variation, and PC2 explained 21% of total variation. We didn't see a lot of associations with CPUE and our water quality. However, we did see some of our basins, our eastern rivers, pulling more towards total organic carbon, and our western rivers pulling more towards pH, alkalinity, and DO. For CPUE and flow, what we did is we took the varying median flows, our low flows and our high flows, and we combined them into categories, as you can see on the x-axis, ranging from less than 10 all the way up to greater than 6,500. And by doing this, I did see a positive relationship with abundance and flow. Our last model, it's kind of it's a lot, but we ran a PCA with just productivity, flow, and productivity, flow, and water quality, sorry. And what we noticed is that you can imagine on that PCA, our 42 reaches were spanned across productivity, TOC, and flow, as well as alkalinity and specific conductance. And so we wanted to see a clearer picture to better describe this. And so we took the PC1 scores, put that on an x-axis, and our y-axis we logged our abundance, and we saw a positive relationship with productivity. We see those reaches associating more with productivity and TOC and coming down into specific conductance and alkalinity. That brings us to our discussion. Study results supported some of our initial predictions, such as CPUE was positively related to productivity, as well as flow. However, there was no relationship detected between CPUE and water quality. So this diagram here just explains that, we see with greater productivity coming from more nutrients coming into the system, we see an increase of algal growth, planktonic abundances, sediment-based organisms, and benthic organic matter, which are all things which are the primary sources of food for freshwater mussels. We also see an increase in mussel abundances. For CPUE and flow, again, we saw a relationship, and we know that in lower median flow reaches and areas, it's a less favorable area for freshwater mussels because of lower dissolved oxygen, higher temperatures, whereas in higher median flow, it's a more favorable area because of increased space, increased habitat heterogeneity, increased feeding, and increased clearance rates. For CPUE and water quality, it was interesting because we did not detect a relationship, and this could primarily be that our 11 water quality variables didn't exceed the tolerances set noted by Scheck et al. You know, out of those 11 water qualities, 11 water quality parameters or variables, they didn't exceed the tolerances. And so that's maybe why we didn't see a relationship. So that brings me to study benefits. You know, this is the first study that looks at CPUE of freshwater mussels and productivity. So there's a lot for me to learn. We're still at the drawing board, but you know, this study could benefit support biomonitoring programs by establishing CPUE as a reliable indicator of ecosystem health, similar to aquatic invertebrates and fishes, as well as help predict environmental impacts by providing a framework to anticipate the effects of land use changes, climate change, and water management on the aquatic biodiversity. With that being said, I'd like to thank Dr. Bonner and Dr. Bonner's lab, present and past students, the folks at BioWest and Texas A&M Natural Resources Institute. Thanks.
Do we have some questions?
Yes, sir. I'm sorry? He asked if we looked at substrate, and no, sir, we didn't. We just took, you know, we just wanted to look at water quality, productivity, and kind of our abundances as well as flow. That is definitely something, though, we would like to look into because, you know, the impacts of stream power and stuff has, you know, an effect on mussels. So, yes, sir. Yeah, I think so, especially considering like the annual rainfall we get in East Texas compared to Central and West. So we're getting a lot more erosion into the river. And yeah, those three elements, potassium, phosphorus, and nitrogen, can definitely influence algal growth, you know, detritus, more things for the mussels to feed on. Any other questions?
So our next presenter is Somerley Swarm, and they're going to be talking on assessing acute ammonium toxicity of Texas uniated mussels.
Good afternoon. My name is Summerly. I'm with the U.S. Fish and Wildlife Service at the San Marcos Aquatic Resources Center. And today I'm going to talk about a collaborative project we did with the Guadalupe Blanco River Authority, Texas State University, and Texas Parks and Wildlife Department, where we're assessing acute ammonia toxicity of our Texas uniated mussel species. A little bit of background: ammonia is a chemical compound very common in wastewater discharge or agricultural runoff, like we talked about last presentation. Data showed in early 2007 that freshwater mussels are particularly sensitive to ammonia, even more so than the previous organisms used to regulate ammonia thresholds in the water. So much so that they had to lower the EPA guidance in 2013 to accommodate mussels and protect them. So the EPA releases this water quality criterion, which are guidelines that consist of the best available scientific data regarding concentrations of chemicals that might affect aquatic life or human health. So these are just guidelines, they are not regulations. Excuse me, I'm not a toxicologist, so these are some very loose definitions, but the document provides these final acute values, which are median lethal concentrations of a specific chemical that are derived from the best available data. So basically, they're lumping all the data that was collected on mussel toxicology to provide a value that is protective of mussels. And that value was 33.52 milligrams per liter of total ammonium nitrogen. They also provide an acute criterion maximum concentration. So this is the the highest concentration that they can be exposed to without experiencing adverse effects. This is kind of the bare minimum. This number is only supposed to be met for 1 hour every 3 years. Other than that, you will have damages to your populations. So that is 17 milligrams per liter of total ammonia nitrogen. And these are both at a pH of 7 and 20 degrees Celsius. So total ammonia nitrogen is both ammonia and ammonium. There are 2 species in there. And at higher, and the ratio in a solution varies based on the temperature and the pH. So at higher pH water, higher temperature, there is more ammonia in the water. And ammonia is more toxic than ammonium to aquatic animals. So important to note because Texas has high pH, high temperature water typically. So our study, there's a bunch of wonderful toxicology data out there on about 70 mussel species. Unfortunately, none of that data included any of our Texas mussel species. And now there are about 12 listed TNE species in Texas. We thought it would be important to provide those numbers so that regulatory guidance can be made. And it's up to the states to regulate those numbers. Again, those guidelines provided by the EPA are just guidance. So the GBRA developing their habitat conservation plan wanted numbers for some of those species represented as well as in the Sabine River. So our objective was to test Glycidia and juvenile mussels from Texas to determine those LC50 values. So it's been touched on a little bit, but uniated mussels are a family of mussels with this really cool life history. So a gravid female mussel broods her larvae in her gills. They're known as Glycidia. Those Glycidia are an obligate parasite on a host fish. They must make contact with a host fish to insist upon them and leach nutrients for a few weeks where they drop off as juvenile mussels, thus completing the life cycle. So in this study, we are only testing Glycidia and juvenile mussels. We just go and collect the brooding females from the wild. So their attraction strategies of getting the Glycidia to their host fish varies, and it's really interesting. Long-term brooding mussels have these very wide windows of gravidity. We can pretty much go find them gravid most months of the year. They're typically that luring species, which on that last slide, you can see the mantle tissue mimics the prey item, perhaps a percivorous fish. And so when the fish comes along, think it's going to get a bite of fish, instead it gets a face full of Glycidia. Surprise. So the gills of these mussels are very chocked full of Glycidia so that when that fish strikes, it's going to make contact. What makes working with these species really good is it's easy to do this in the lab. We can just use a needle and extract the Glycidia, making it available for our studies. The short-term brooders, they are a little bit different. So they only hold their brood for a period of a few weeks. It's very narrow windows that we can go find them gravid in the wild. Because they are, instead of luring a fish to them, they are actually spitting out conglutenids or Glycidia packages for a fish to come along and eat at their discretion. So we see lower fecundity a lot of the times. They can expel prematurely when we go out in the field and we're looking for them. If we just handle them wrong, they might spit them out. So it makes them a huge pain in the butt to work with in the lab. And there was not a lot of available literature on them. And now I know why. But we were able to get some data on these guys, so that's pretty novel and pretty exciting from our study. So how do you tell if a Glycidia or a juvenile mussel is alive? The Glycidia, we have to take a subsample. And so basically you'll quantify the number of open Glycidia and the number of closed Glycidia, and then you'll add a salt treatment. That salt treatment mimics the fish. So if a Glycidia is alive and it snaps shut, we can assume that it was alive because it was able to close down on the fish. So that gives us our viability percentages. Our juvenile mussels are a little easier. You literally just look and see if they're moving around. If they're moving around, they're alive. This is important because that is the end point for these toxicity tests, is knowing whether your animal is alive after exposure. Our guidelines followed the American Society for Testing and Materials. Pretty nice that they laid out all our methodology for us. It's good because it's standardized so we can compare our data to other species tested in other parts of the world. So there, we followed the 2022 revision. This is all pretty well laid out for us. Trials should be held at a 20 degrees Celsius and a pH of 8. Control viability needs to maintain above 90% for our test organisms. And we are typically aiming for above 3 brooding females to pool their glycidia together for these trials to introduce some variability. So this is what one of the trials would have looked like. It would be, for one trial, there would be 3 beakers of the same concentration. So there's replicates within each trial. The total ammonia nitrogen concentrations ranged from 32 down to 1 using a serial dilution, and then at 0 being our control, where there was no total ammonia nitrogen milligrams per liter. 500 glycidia per beaker. They would have been exposed for 24 hours. So we would do a test at the beginning of the study. So at 0 hours, do a sample of viability, and then at the end, do again that sample. For juveniles, the test concentrations and that layout looked the same, except there were only 5 juveniles per beaker. They're exposed for 96 hours. And so we would test at the beginning of the trial, again at 48 hours. At that 48-hour mark, we would also refresh the solution to maintain the total ammonia nitrogen because it can volatilize out. And then again at 96 hours, which would conclude the study. So here's our species that we ended up testing. As I mentioned, it's a mix of Guadalupe species and East Texas species. So we collected from the upper and mid ranges of the Guadalupe River and the Sabine River. And then there's one, the Lampsillus terraces from the Brazos. That was just while we were figuring out our methodology, but we included the data. So there's a mix of federally endangered mussels and common species, which was good. We wanted that data for those federally endangered species so we can inform our habitat conservation plans. You'll notice that the top 3 are the short-term breeders. We were only able to collect data on the glycidia for these species. And that is because, as I mentioned, they were really difficult to get enough glycidia to do these trials. So we didn't have enough to also infest fish and get the resulting juveniles to do those studies. So those last 2 columns show the number of trials we completed per species. For the long-term breeders, as mentioned, they're a lot easier to work with. We had a lot more success. We got juvenile and glycidia data for them. So here are our results. This is the lethal concentration of total ammonia nitrogen for each of those species. Pretty easy to read graph. I included those final acute values and the CMC values that the EPA set as the recommendation, and you can see that they are protective of our species for the most part. So it's good to know. EPA did a great job. And similar trends for our juveniles as well. As you can see, the juveniles are a lot more sensitive than the glycidia. And that's interesting and follows actually well along with the EPA data as well, that trend that juveniles being more sensitive. And so much so that the EPA is actually now recommending that the juveniles be used as the target organism for the regulation. So we're not going to hopefully move away from glycidia testing and only move towards juvenile testing because you want to protect the most sensitive life stage. So in conclusion, we determined the novel values for these species, which can now be utilized by regulatory agencies. We demonstrated that those guidelines are in fact protective of our species. And we'd like to continue this research doing chronic exposure. Although what's cool to know is that those 5-day juvenile trials can also be, what the EPA has shown is that chronic exposure and even those 5 days are pretty similar data. So the 5-day juvenile trials are actually probably our best bet for doing water quality testing in the future. And I'd also like to get some juvenile data for those short-term brooders, even though they were a pain in the butt. And I would love to thank my co-authors and colleagues on this, the Texas Parks and Wildlife for funding, the Guadalupe Blanco River Authority, as well as SMARC and Texas State. And with that, I'll take questions.
Some questions.
Well, Yeah, so she was asking if there was, yeah, I can go back to it too. Oh, never mind. So she asked if there was a difference in sensitivity from the short-term brooders to the long-term brooders. Obviously, we can only look at the glycidia data for this. There was no significance when we tested it. Yeah, they were not significantly more sensitive than the long-term brooders. In fact, they seem to be more tolerant, just observationally, but not statistically. Yeah, that's a good question. She asked if they might be more tolerant because they're exposed to the water longer, right? So it's possible. So we actually, because they're in a conglutinin package, it's suggested that the conglutinin package might be protective, but we did remove the glycidia from the protective package. So we would be removing that protective barrier for this study. So hopefully it made that, it eliminated that variable.
Thank you very much. Our next speaker is Alexander Zolmat, Population Genetic Structure in a Rare Freshwater Mussel.
All right, thank you. Thank you all for being here. I'm going to talk a little bit about some of my dissertation work that I've been doing with the Louisiana pigtoe, also known as Pleurobiema rodellii. So we'll get right into it. So Louisiana pigtoe is of the freshwater mussel family Unionidae. This particular species is rare throughout its range, which spans from the San Jacinto drainage in the west to the Pearl River in the east. And it is of conservation and management concern across its range, where it's particularly rare. It has been proposed as threatened by the U.S. Fish and Wildlife Service and is considered nearly threatened by the IUCN, and as such has been on the radar for resource managers throughout its range. There's a lack of data and information on this species. In particular, we know very little about its life history patterns and the population structuring in the system. We also know very little about its evolutionary history. And so our lab set out to rectify some of these gaps in the knowledge. And the data I'm going to show you today is part of a larger study. So I'm going to focus only on one of these study objectives, which is to characterize the distribution of genetic variation in the Louisiana pigtoe across the range. And so as I mentioned, this is part of a larger study that we're doing that involves a lot of other unionid species. And so we have a large collection of multiple species from the unionid family. Our collections for tissue samples span from 2018, and the last tissue sample in this data set is from 2022. And we took these tissue samples, extracted genomic DNA from them, and generated a restriction-associated digest DNA library, which we sent off to the UT Genomics Sequencing and Analysis Facility to generate sequence data. These sequences were then assembled de novo style, so we didn't use any fancy reference genomes for this, unfortunately. But we are able to leverage this data set to sort of confirm our IDs in the field. So anybody who works with freshwater mussels knows that these, especially in East Texas, identifying these things in the field can be problematic sometimes. So we always want to make sure that we are applying our analyses to genetically confirmed individuals. And so that's what we've done here. We use several methods to confirm these IDs. I'm showing you one of them. This is a PCA of genotypes, and our Pleurobiema rodellii are down here in the right corner, circled in blue. And so all downstream analyses involved these confirmed individuals. So everything I'm going to show you from here on out was done on these confirmed Louisiana pigtoes. And so with our confirmed pigtoes, we generated another assembly using just the species that we're interested in, the pigtoes. And this data set generated 12,000, around 12,000 single nucleotide polymorphism loci. We had 351 individuals in this data set from across the range, which included 7 major river basins. And here you can see our sampling localities, and the gray area outlines sort of the known recorded distribution for this species. And we took a 3-pronged approach to assess population structure. So we start with a principal component analysis of the genotypes. This is just a way to visualize genetic structure in sort of a 2-dimensional way. We then did something a little more rigorous with the entropy model. This is a model developed by Gompert et al. It basically it's a Bayesian model. It estimates several population genetic parameters that we're interested in, namely these two, Q, the ancestry proportion, which I'll talk about in a bit, and posterior genotype probabilities. And then finally, we applied the classical F statistic approach, which is a pairwise comparison method that generates a metric of genetic differentiation between 2 populations being compared. All right, so we'll start with the PCA. So I'll kind of explain what goes into the PCA. So we input our genotypes of our individuals across the sampled loci into the PCA. And what the PCA gives us is clustering of these individuals based on genotypic similarity across their genomes, across their sampled genomes. And what we found was, so on the right here, I have PC1 plotted against PC2 axis, and each point here is an individual. We colored them by their basin of origin, and we saw that they clustered mainly by basin. Interestingly, we found that PC1 really separated out our central geographic populations from our peripheral exterior populations that we sampled. And this pattern was consistent when we looked at finer scale patterns of genomic variation. So everything really kind of makes sense here given how muscles are distributed and dispersed. Next, we applied this entropy model. This is a hierarchical Bayesian model that estimates population genetic parameters. What's special about this model is it can account for uncertainty due to the sequencing process. So it takes into consideration the error rate of the sequencer. And what you give entropy is the genotype and the assumed number of populations. So you tell entropy to assume how many groups there are, and entropy will then try to assign each locus to one of these K groups. And so a way to think about this is, here's a bar plot. Each bar is an individual, and the y-axis is the proportion of that individual's genome that belongs to one of these groups. And so if you think there's 3 populations and there's high population structure, you might expect to see something like this, where each locality shows up as fully different. On the other end of the spectrum, you might expect to see something more like this, where there isn't a lot of population structure and it's more panmictic. And potentially the most interesting case is when you get some intermediate ancestry, where this is usually interpreted as gene flow, hybridization, some kind of connectivity between the 2 localities. So what did we find? Looking at K equals 2, so this is telling entropy to assume that there are 2 populations. The first thing that comes out is this big central portion of the group. I can't really see the names under this thing here. But basically, the San Jacinto is the furthest west population. Then we have the Neches, Sabine, and Calcasieu, which are all sort of the central portions of the range. And then again here, the external, peripheral portions of the range, including the Red, the Ouachita, and the Pearl rivers. So consistent with what we saw in the PCAs. Tell entropy to assume 3 populations, and we still are seeing that central geographic group sort of stay as one thing. What comes out here, I know you can't see it, this is the Red River populations. So we're differentiating the Red from the San Jacinto here. At K equals 4, we finally start to see and resolve some of that central geography. So here, again, you can't see the label here, but it's the Calcasieu River is coming out as this blue mass. And an interesting finding here is that the Sabine River shows some intermediate ancestry. This is potentially indicative of some gene flow between the pink group and the blue group, the Calcasieu and the Netches and Sabine. K equals 5 further separates out the Ouachita, and then K equals 6, we start to see the model sort of break down, and we're not adding any new information at this point. And so generally, we see those same patterns repeated in the entropy output. Finally, I applied the classic FST approach. So this is a classic measure, a pairwise measure of genetic differentiation between two populations. Larger measures of this metric mean that the two populations are very different genetically. Lower values indicate that they're more genetically similar. So what did we find? So I plotted this in a heat map. I think colors are better to look at than numbers. And darker values or darker colors mean larger values of FSD, higher differentiation. Lighter values are lower values of more genetic similarity. And so what did we see? Again, we're seeing those peripheral populations sort of pop out. They have the most dark red portions of this heat map. And the central populations are really similar to each other and really different from the peripheral populations. So this consistent pattern that we keep seeing throughout all of our analyses. So what does this all mean? Well, genetic variation appears to be structured by basin of collection. So these river basins are really structuring a lot of the genetic variation, and that makes sense given what we know about how mussels disperse, right? They need to attach to a host fish. These fish mainly stay in their basins, exceptions being when there's flooding or connectivity between these basins. We see the Neches, Sabine, and Calcasieu rivers share significant genotypic variation. And we saw that there is strong population structure that was most apparent on the peripherals of the range. The Ouachita and Pearl populations showed very heavy differentiation from most other basins, and especially in the FST analysis. Although we have to take this finding with a grain of salt, those two populations have extremely small sample sizes. But if we take these findings at face value, these findings are sort of consistent with a biogeographical range expansion and then a contraction, leaving those peripheral populations isolated and free to evolve on their own. And with that, I'd just like to say a couple thank yous to all the people that have helped me. This is a large collaborative project, so thank you to all my collaborators, and I will take any questions. Good deal. Thank you.
Okay, our next presenter is Tyler Bowling. He's going to be talking on species distribution modeling of the elusive Texas scarlet snake.
Hello. Sorry to ruin the muscle talk streak. So you got a sneak preview of when Dr. McCracken was speaking earlier about Texas scarlet snake. And this is a fairly recently described species. It was split from the now eastern scarlet snake, Stenophora coccinea, and this is Stenophora linneri. And we don't know very much about it. We can infer a little bit from its sister species life history, but there's less than 40 observations of this species so far. So it's very rare. We believe it's semi-fossorial. It mainly preys on reptile eggs. It may prefer sandy soils, possibly in oak mottes, likely nocturnal, though the range is obviously poorly defined given the limited number of observations. Currently, it is listed as critically imperiled at the state level, but the true status is unknown just because there's not enough information to really make these determinations. And so they're facing a lot of uphill battles, including urbanization and sea level rise. They're diminishing their habitat. And so there's this need to establish an accurate geographic range so we can create more studies for the species to learn more about its life history in general and to potentially protect core habitats. And so we set out with three main objectives. The first being to create a preliminary species distribution model to get an idea about where the species might occur. And this has been done, and I'm going to talk about this for a little bit. And then secondly, which we're actively in the process of doing, is conduct long-term surveys based on that species distribution model, trying to find more observations of this elusive critter. And then lastly, hopefully, we get observations of this snake, and then we can incorporate that into an improved model, which we can then pass along to the state. So for those of you who may not be familiar with a species distribution model or an SDM, it takes observations of a species and environmental variables about the habitats where they live, and it kind of fills in the gaps. It guesstimates similar areas where they might be found. And I made two different species distribution models for the scarlet snake, and I included false observation points as a balancing way of training the model, so it wasn't too biased. And so that way I could check if it was performing well. So if it was saying that it was located where it wasn't, I could check it a little easier. And I made two different versions. I made a random forest, which is an ensemble machine learning technique, and a maximum entropy model, which is a linear model. Here you can see the 35 observations that we know of. These are a combination of state herpetologist reports as well as museum specimens and iNaturalist observations. And there are still 3 iNaturalist observations that are obscured because this is a protected species. We've been trying to contact those users, but no luck yet. But you can see that they're all congregated around the south coast and the brushland. And so for gathering environmental data, I started with a large variety and tested them through the model and see which were the most strongly performing so I could limit them down. And these ended up being annual average temperature, shrubbery cover, percentage sand in the soil within the first 60 centimeters, soil pH, and soil density. And so you can see the comparison of these two models with the maximum entropy on your left and the random forest on your right, and they're largely in agreement. So probability of occurrence is on the y-axis, and then whatever the range of units for that particular variable is on the x. And so the broad trends are that in denser shrubs, there's less likely a chance to find them. They prefer higher annual temperatures, pretty steady soil pH, but at higher pHs, they kind of dip off in probability. They're more likely, greatly more likely to be found in sandy soils, and they prefer less dense soils, which makes sense. If they're burrowing around, they want to be able to push that earth around. And then I performed a Pearson correlation, which basically combines the two models and sees how much they agree with each other. And these are pretty close. They are correlated. A 0.76 out of a 1. Highest would be a 1, lowest would be a 0. So these are largely in agreement. So then I look at the actual performance of the models. And over here on the left, the max ent has extremely high AUC score, which is really good. It only goes to 1, and this is a 0.95. This is an extremely well-performing model. Unfortunately, our random forest did not perform as well. This is a very data-hungry model, which I was lacking in some areas, obviously, with so few observations. So it honestly got overwhelmed by sand and didn't train the model on the majority of the remaining variables. So we chose to proceed with the maximum entropy model. And this is what you see here. This is a laid out map of the species range, and you can see the different colors with the different predicted probabilities. So red being highest and purple being likely absent. And then you can see mapped out those existing observations, and which match up pretty closely. They're along the Ingleside sand sheet, the Texas coast, as well as the central brushland. And so now we're moving into phase 2, where we're trying to find more of these snakes based on this preliminary model. And you may be familiar with a drift fence. If you're not, it's simply a small fence that you put out in the environment. Animals naturally follow the barrier. There's usually a pitfall trap. And then you come back and collect them But these can have certain risks, including mortality from exposure or predation. And so we want these to be out on the landscape for a very long time. So we're using a modified technique called the AH drift fence, or the adaptive hunt drift fence. And so at either end is this overturned bucket with, openings on either side. So the animal moves along the fence, and inside the bucket is a game camera. And so that motion activates the camera, and so these can stay out for several months at a time, with the only really limiting agent being memory card space. Now, some things can trigger the camera, like shadows or leaves blowing around. Additionally, some of these are heat sensitive, and when you've got an organism that's ectothermic, they may not be triggering these as well. So we're working on implementing an update of a break beam sensor. And so essentially what that means is there'll be this beam, and when the animal interrupts the beam, it sends a signal to then take a picture. So something has to move through. And we've also are adding this little ramp that will, deter detritus from building up. So we should reduce our false triggers. We should increase our response time, and it should be compatible with any PIR's camera. So as of today, we actually have 24 of these fences deployed across the northern and central parts of the range. We plan to translocate these fences next fall into the southern range. But you can see that we have a mix of high probability locations, medium and low. Typically with these types of studies, you would only have high probabilities to maximize your chances. But given the extremely limited number of observations for this species, we're trying to cover all our bases and make sure that we aren't missing something by ignoring these low probability areas. And next week, a week from tomorrow, we'll actually be installing the 25th. So this is still an active project, and most of these have only been on the landscape for a little over a month. So it's very exciting to see what kind of data we're going to get soon, but we've already gotten a little bit. And as far as image analysis, you heard a little bit about this already, but Wildlife Insights is a online freely accessible AI. And so it does a pretty good job with mammals and telling you if the image is blank, and it'll show a bounding box in red to tell you if there's an animal. It doesn't do a great job with herps, but it does do a nice job of weeding out those blanks. And so we've gotten quite the variety of animals that we never really expected. I personally did not think we were going to get birds inside these buckets. But as you can see, reptiles is our top group that we've gotten so far, and this is across a staggered release everywhere from August through November. And we've gotten all kinds of stuff, different arthropods, mammals, and especially the birds are extremely surprising. And we expect some more as we'll be pulling SD cards next week. But like I said, the best group that we've gotten information for is reptiles, and among those reptiles is snakes. And you can see quite the variety of snakes here, but you will notice a distinct lack of scarlet snakes so far. As alluded to, it is technically possible that we may not find any, but I am forcing myself to believe that we will just find so many we won't know what to do with it. In addition to this, because images can sometimes be hard to identify, we've also been adding in video for easier identification within the buckets. And so, hopefully, probably regardless of whether we find the scarlet snake, but hopefully we do, we're going to be revising the model. We didn't have a good starting point of where to even look, so we just made a preliminary model to get started, and now we're going to be planning those next steps. And so we're going to incorporate any observations that we do have. We're going to be trying a couple different model types that we didn't originally try. We're going to include some different variables, including land use, some enhanced vegetation surveys. Even learned earlier this week that NOAA is doing a pretty intensive LiDAR land cover data set, making that available, looking at seasonality and whatnot. And we're also going to correct for some bias that we noticed in the model. For example, we want to randomly sample away from roads. A lot of the observations are of roadkill. So I don't think that they're naturally inclined to live on roads. So we're going to correct it for that. And then we're going to spatially filter the data so that clusters don't bias the model too much. And with that, I'd like to acknowledge the McCracken lab, especially our technician, Michael, who has gone out in the extreme heat and the cacti and the swamps and installed all these fences with me, and comptroller's office for funding, and all the wonderful folks who have given me access to their land. And thank you for listening.
Questions?
No? Okay. Thank you very much. Oh, I'm sorry. Did you have one? He asked when the most recent observation has been, and on iNaturalist, it is actually 2024. A woman's cat killed one. It was in her home. I try not to think that that was the last one. Any other questions?
Just a comment that the built some drip fences in the past. They're labor intensive for sure.
Mhm.
Seems like a lot of labor for perhaps a small return on.
We have really thought about like what is a better way to find this animal, and we're coming up short. I mean, we've discussed some eDNA techniques, but I think for what we're dealing with, this is probably the best we can put out there is these drip fences with these cameras to be long term on the landscape. Based on the sister species, the eastern scarlet snake, we believe that the mating season, and this kind of lines up with when the most iNaturalist observations are kind of like mid-spring around April. So we're really hoping that if we're going to get lucky, it'll be this spring when they're kind of moving around more, but we'll see. So she was bringing up that potentially the iNaturalist observations introduce another layer of bias because, people are going to be inside more. And that's absolutely, possible that may be influencing it. There is some kind of checks that we can do in the analysis to avoid that bias more, but it's hard to get around that given the extremely limited number of observations. So for folks online, she was suggesting adding a baffle or a little barrier to keep debris out. I don't know. We'd have to think about that. We don't want to deter them from entering the bucket in any way. It's at the end of the fence. And we just want it to be like a straight shot, like they can see the other side. But it is something worth considering. So thank you. Thank you.
Our next talk is entitled Needle in a Haystack Toward the Use of Machine Learning Models to Detect Texas Tortoises in Large Data Sets by Jacqueline Timat. I probably have mispronounced your last name, so Jackie, so you'll have to tell us how to pronounce your name. We're going to take a minute to get the presentation up. Tell us how to pronounce your last name.
Yeah, I'll go ahead and introduce myself. Hi, my name is Jackie Talimat. I am a PhD candidate at A&M Corpus Christi. And yeah, we'll just give it a second. I accidentally made my PowerPoint too big and that caused some issues. Womp womp. I was requested to tell you guys a tortoise fact while I'm up here, and I have you all captive anyways. So I should mention, I drive around my Kia Rio that I refashioned into the tortoise taxi so people can text me for information about tortoises. So I'll share details like Texas tortoises can detect changes in barometric pressure, so they will sense when there's going to be rainfall and gather to areas that have depression. Sometimes that'll be around roads. So people know when they're driving around during rains to look out for tortoises on the road and avoid hitting them. Information like the fact that Texas tortoises have really small home range sizes and really high, they have a homing sense. So if people see them, they shouldn't pick them up and move them somewhere else because it can really decrease their chance of survival. So just trying to connect other people with tortoises since down in South Texas, they're kind of really common to come across, but people don't always know too much about them, which has created some issues with them. But yeah. Yeah, so if you see one in the road, I always tell people that take a note of what direction they're moving and move them that direction to the other side to safety. Otherwise, they're gonna just try and go back again. You could be like, oh, okay, they're moving to the left. Well, the nearest side is to the right, so I'm gonna move them there. That tortoise is just gonna try and go back to the left again. It knows where it wants to go. So if you can safely pull over and move them, that's what I always try and tell people to do and just keep them going where they wanna go. Yeah.
So we have a break planned right after Jackie's talk. So we're going to go ahead and take the break now. Please come back in 15 minutes and then we'll have her talk. And then we're going to have the BAT presentations and the BAT panel talks. So be back here in 15 minutes. 2:30. Okay, we're going to return to our talks. And the first talk, already introduced, Jackie Kalimat, needle in a haystack toward the use of machine learning models to detect Texas tortoises in large databases.
The problem child has returned. Okay, so I'm really excited to talk to you guys about something that I didn't necessarily expect to be working on when I started my work with Texas tortoises. So for a brief introduction for anyone who's not familiar with this adorable little animal, the Texas tortoise lives in South Texas, primarily south of San Antonio, all the way following kind of the San Antonio River, reaching as far north typically as Rockport, but these days the most northern part is in about Sinton. And then they range all the way down into Mexico. They're kind of an iconic poster child of the Tamaulipan biosphere, is what it's called. Now, what I wanted to talk about today was survey methods. These species can be kind of underrepresented because they can be a pain in the butt to find. The typical methods used for detecting these species are either A, road cruising surveys, or B, trans-sector area and search surveys. Now, road cruising surveys can be limited because you're stuck to a road, and a lot of properties there might not be accessible roads or not a whole awful lot to get to, or they might be highways, which are not necessarily very safe if you're going to collect tortoises off the road to process. The other one is doing transect surveys, which if you know anything about Tamaulipan thorn scrub, you know a lot of time when you're trying to walk through it, it's more of a crawl through a lot of really spiny, pokey stuff like acacia that's going to rip open your clothes as you're crawling and desperately searching for a tortoise. So these can be some limiting factors in detecting them kind of ubiquitously across an area instead of being limited to areas that humans can reasonably access. Another part that makes it difficult, when tortoises are not moving, they do this thing known as palleting. It's different from burrowing because instead of creating this nice big hole in the substrate, they kind of dig up 2 or 3 inches of dirt and settle there. Now, I want someone to be able to tell me exactly, if you can look at these images and tell me where a tortoise is, you win a grand prize because they are a pain in the butt to find. And there are 3 Texas tortoises on the screen right now. So I'll give everyone a minute to look. A little longer, speculating. So you got one here, one there, and one there. So if you're just walking around in their habitat, it is very unlikely to find them. You might even come across them by hearing a thunk as you accidentally step on one. So because of that, we've decided to look at camera traps as a way to detect them across the landscape because, as you all are aware, they can be out for a much longer period of time than humans can be on the landscape, and you only have to go in some of that really painful habitat a couple of times instead of continuously to go through the same area over and over again. Camera traps have really been developing in popularity and how they're utilized. So instead of just traditional putting them on trees, to target mammals, they've been moved up in the canopy. They've been implementing AI to detect birds. They've even been used for moths and for herps with time-lapse modes that are available. And they've also been used for gopher tortoises. So a lot of what they're being used for with gopher tortoises is seeing how many different species utilize their burrows. And the nice thing with gopher tortoises, they have that really recognizable burrow, which Texas tortoises do not have to target, which is why, again, we wanted to try some different methods of using these camera traps. And the reason this is important is because Texas tortoises, again, they're hard to detect. And because of that, that can lead to some areas not detecting them for almost decades. Like with the San Antonio Missions National Park, they detected a Texas tortoise 2 years ago for the first time in 13 years. Now that could have been a tortoise that was transplanted there, or it could have been that there is a very small population that's just in some very thick habitat and rarely encountered, which is some of the cases with some of the properties I've done surveys with for other parts of my dissertations is people are like, well, I see them maybe once every couple of years, which means there might be a need to change the methods we survey. So originally, I was just going to test out different, grid methods, camera deployment methods, and whatnot, seeing if seasonality, habitat type changed any of this, and use that as a recommendation for people. I very quickly gathered a lot of data. In fact, at this point, it's close to 11 terabytes of data. that's not something I'm going to tell someone to do who's just doing this as a side mission. I wanted to make an easier way to sort through the large sets of data that often occur when you're using time-lapse data, which is necessary with Texas tortoises because they are ectotherms and they won't trigger the motion sensors that are often using infrared to detect the reptiles on the landscape. So I decided to go out on a very far side quest from my usual stuff and learn how to make a machine learning model to detect Texas tortoises on the landscape and compare it to pre-existing models, which is still in the works. So I'm just going to focus on the first objective for today. So this is a general pipeline of how data gets processed for machine learning. And I'm going to break down each of these steps for you. So I hope you're all ready to learn about machine learning because I am so excited to at least get some of you to understand a little bit of it. Okay, so first is data collection. Now, as you saw that map earlier, this is pointing out the 3 different sites. I was trying to get representatives from as many different habitat types as I could. So the first site was at Chaparral Wildlife Management Area, which represents more open kind of brushland, much easier to walk around in. Then the other 2, there's Las Palomas, which is Tamaulipan thorn scrub and an agricultural matrix. And then last was Cactus Creek Ranch and Laguna Atascosa National Wildlife Refuge. So those kind of represent your Loma, Tamaulipan thorn scrub, and coastal grassland matrices. So those 3 to, again, help train the machine learning model to be as robust as possible and be able to recognize tortoises in as many different habitat types as possible. Next was processing. So the files that the cameras we got process these in were TLS or videos that I would have to watch through and sort through for tortoises. So it is very difficult to process them. A lot of times you're staring at nothing, grass, waving, plants. But sometimes you very excitingly get tortoises copulating in front of you and you're like, oh, okay, there we go. Now I want to point out the timestamp, 6:39 p.m. 2 minutes later, different side of the screen, did not trigger. We had it set to a mode where it would also detect any movement, did not trigger the motion sensor, just to show that you really need to use time lapse for these animals. So alongside using that, so I would notate the time and any sort of additional activity that was going on when I was going through these images or videos for Texas tortoises. And these were going to be used for training the machine learning model. On top of that, I gathered some additional data from iNaturalist and A&M's Kingsville. They've been doing some underpass surveys. They have a lot of Texas tortoises because they're in Cameron County, and they were kind enough to offer me their Texas tortoise images to, again, get a wider array of tortoises on the landscape. Next was annotation. Now, this was a really painful part. I went through, and because I'm a little bit of a perfectionist, instead of just using bounding boxes, which give a general area of where a tortoise is, I drew polygons. So each single circle you see here is a dot I placed manually zooming in, so it instead learns every single pixel within that polygon is a tortoise. Every single pixel outside is not a tortoise. So it is much more intensive and much more specific to learning tortoises or whatever object you're looking at in a picture. Now, next, what I did, ChatGPT helped me with this part to create the architecture and didn't know the environmental impacts at the time. Highly recommend reading some articles, learning more about that, but it helped me to create the model. And I used what's known as transfer learning. For any of you interested in machine learning for species specific, transfer learning is the way to go because it is able to learn off of a very small data set. So it takes pre-existing models. So the models I used are MaskRCNN and FastRCNN. If you want more details on that, I can give that to you later, but I won't go into that now. Essentially, these models are already trained to recognize objects that use the same type of annotation within a context of an image. And so with that data set, I take that model, it already knows like, okay, this is how I learn images. This is how I learn these pixels. It takes my data set and it transfers that learning skill to learn the new data that I am giving to it. So that's the very basics. I use a pre-existing model and use it to train a brand new model. So along with that, I subsetted my big annotation data set into several different experiments. I would change how many times the model would run through the entire data set to learn from it, what types, if it would include blanks, non-tortoises, augmented data, all of those sorts of things to get the very best possible model. Once I had run all of these experiments, I did model evaluation. Now I'll break this down as simply as I can. To do evaluation of a model, you're essentially looking often at two parameters. Precision, which means it looks at the image, it calculates what's known as intersection over union. So essentially, we have a tortoise here. This blue box, that is the ground truth annotation. Then with every time the model is guessing what a tortoise is, it takes how much that box overlaps the main annotation. And if it's really overlapping it, like it's almost dead on perfect, you're gonna have a high precision. If it's hardly overlapping it, very low precision. The next is recall. That just means the model tells you yes or no, a tortoise is there when it actually is there. If there's a tortoise, the model doesn't detect it, it's a 0. If there's a tortoise and the model detects it, it's a 1. And it creates these nice curves of what is called precision and recall. So this one is showing at a really high confidence threshold. The recall and precision isn't necessarily too high, but when you move to 90%, it's doing pretty well. It's got a recall of about 70% and precision of about 55%, which means it's often knowing when a tortoise is in the image properly, but not necessarily getting it dead on perfect. What has been really exciting for me is using my current best performing model, and it's actually doing a really good job at finding tortoises that would be a lot trickier to find. So there's this one here, and the model is 96% confident that there's a tortoise there, and it's perfectly outlining a tortoise, which is really great for knowing the metrics of how well it's learning a tortoise. And this is an image it's never been trained on before, so just new data, what's known as the test set. Along with that, I have another one that it's 100% confident in, and it's even working on other North American tortoises like this gopher tortoise. Now, it was also guessing that this little rock here was a tortoise, but it was only 7% confident in that. So pretty easy to filter that out. So what I'm hoping is that this model will be used by others in areas where tortoises maybe haven't been detected in a while. So cameras can be put out and people can kind of go through this model and check if tortoises are present. So there's a lot of people that have helped me with that I would like to thank, especially my University A&M Corpus for helping fund this project. And with that, I also wanted to mention I put a GitHub repository together. So if you guys want to check it out, I don't have the model currently uploaded, but I have the code you can use to really easily, you don't need to be a computer scientist. There will be specific instructions, and you can actually use the predictor to go through your own data sets and find Texas tortoises. So I just recommend following it for now because it's not completely put together yet. But yeah, with that, I'll take any questions.
Questions?
Yes. Yeah, so a question was asked, what is the lifespan of a Texas tortoise and how many eggs do they lay? So currently it is thought that they live up to 30 to 50 years. We're not totally sure on that because previous aging metrics counting, the rings on the scoots is not super accurate, we know anymore based on diet and whatnot. And then how many eggs? It's typically about 2 per clutch, and they typically only have 1 clutch per year.
Thank you very much.
Yeah.
Okay, for the remainder of the afternoon, we have a special session on bat conservation. We're going to have 6 oral presentations, and then following that, we're going to have a panel discussion. Panel is going to address some questions that have been generated, and then you'll also have an opportunity to ask some questions. So we're going to begin with the first oral presentation by Madison Nadler, Seasonal and Spatial Variation of Bat Communities in Texas.
Good afternoon. I am Madison Nadler. I'm a research associate at Texas State University, and I will be starting the bat information session off on telling you about the bat communities across Texas. Bats are a diverse taxon. Of the 47 species found in the United States, 33 have been detected in Texas. And these species have many physical differences. They have different body sizes, wing morphologies, and nose and ear shapes. They also differ in their diet and echolocation characteristics. Bats also live in diverse habitats and utilize different types of roosts, potentially due to niche partitioning. And bats also behave differently, with some migrating or hibernating and others remaining active year-round. Bats are also contribute and also provide many ecosystem services, such as agricultural pest suppression and pollination. They help farmers save millions of dollars each year in pesticide use by eating crop damaging pests, and they also pollinate important crops. In addition, bats contribute to local economies by attracting tourists to nightly roost emergences. With their large bat diversity, their large spatial range, and their sensitivity to environmental change, this makes bats ideal indicator species. As bat communities change, changes in those communities can be representative of changes in the environment or environmental characteristics. And so it's important to know how these bat communities are changing due to different changes in their environment. While many states are diverse in their habitats and weather patterns, Texas is unique in that it hosts the greatest number of bats. It also has the greatest bat richness, and it's also the largest state in the continental United States, along with having a lot of diverse habitats and weather patterns. This makes Texas an ideal study system for studying the effects of land cover and weather on bat communities. Texas also has many threats to bats. Texas is the largest wind-producing state in the US, and bats are affected not only by the wind turbines themselves, but also through habitat destruction for their construction. Other reasons for habitat destruction in Texas include growing urbanization and increased agriculture. And while bats help farmers by eating crop pests, they still require suitable habitat for roosting. Bats are also affected by disease. In 2017, PD, the fungus responsible for white nose syndrome, was detected in Texas. And the first bat mortality due to white nose syndrome occurred in 2020 on the cave myotis, which was also a first for that species in the country. And last year was detected in tricolored bats. So our objective was to determine the seasonal and spatial variation in bat communities across the state, as our study aligned with the emergence of white nose syndrome in Texas and with the growing development of wind energy facilities. Due to the many threats faced by bats, scientists developed the North American Monitoring Program in 2015 to standardize bat research across the continent. We used these methods to monitor 49 sites across Texas for 3 years. Each site had 1 to 2 acoustic detectors, and we used a combination of manual and automatic identification of those recordings. We used that bat echolocation data to compare the acoustically active bat communities across Texas using Shannon diversity and richness indices and Permanova and Semper analyses. We combined some species into species groups, red bats, yellow bats, and all myotis species except for the cave myotis due to difficulties in differentiating these species acoustically. We also measured land cover variables at 3 scales around each detector for use in some of our analyses. First analysis is the diversity and richness between seasons. Diversity was greatest in summer, but the richness was relatively even across all 4 seasons. However, there was greater variation between ecoregions, and the greatest diversity occurred in the piney woods, followed by the Trans-Pecos and Oak Prairie. And the least diversity was in the cross timbers and South Texas plains. However, richness was greatest in the high plains and panhandle, Trans-Pecos, and hill country areas, and it was lowest in the post oak savanna and Oak Prairie ecoregions. Next, we compared the bat communities between seasons. And while visually it looks like the bat communities might not differ because those ellipses all overlap, both the Permanova and post hoc analysis revealed that the winter bat communities differed from the spring and summer communities. And so this makes sense if you know bats, because some bats migrate in fall, some hibernate in winter, and others remain active to some degree year-round. I also want to point out and remind you again that this data is based on acoustic recordings, and so these communities represent the acoustically active bat communities. Hibernating bats are still present in winter, they are just not as active. 4 species, the resilient free-tailed, red bats, silver-haired, and evening bats, contributed to over 75% of the dissimilarity between seasons in the bat communities, and 5 additional species contributed to over 90% of the seasonal communities of bats. However, despite the low differences between seasons, ecoregions had much more difference in their bat communities. The map in the top right is colored to correspond to the same colors of the ecoregions in the MDS plot, so you can visualize where these bat communities are differing or similar to one another. The resilient free-tailed and red bats were the most important species in differentiating the bat communities between ecoregions, but silver-haired evening and yellow bats were also big contributors to differences between ecoregions. Canyon bat and all myotis species set apart the Trans-Pecos from the other ecoregions. The cave myotis was important in the High Plains Panhandle and South Texas Plains ecoregions, and the tricolored bat helped set apart the piney woods, Trans-Pecos, and the Oak Prairie ecoregions from the bat communities in the other ecoregions. Lastly, we compared Permanova models with land cover at 3 different scales to determine which scale was the most important for determining bat communities. And so we did this by comparing the R-squared, or the unexplained variance of the Permanova models, and selected the Permanova model with the least unexplained variance as the best fit. So each bar is one model with colors corresponding to land cover types, and the percentages at the top are the unexplained variance of that model. The 25-kilometer model had the least unexplained variance of the 3 scales, and so we use land cover at the 25-kilometer scale for the remaining analyses. We wanted to add ecoregion because we know land cover varies across the state, and then we also added season because bat communities vary between seasons. In that final model, season and ecoregion explain the most variance, and of the land cover variables, urban explained the most, followed by forest, wetland, water, barren land, grassland, and crop with the least. Some of the reasons for these seasonal and spatial differences we observed in the bat communities across Texas include weather and land cover. Weather varies throughout the season throughout the year, as you would expect, and the large size of Texas makes sense because the weather is going to vary across the state, and land cover is also going to vary. Roost availability is important to bats because they are not going to be found in areas without appropriate roosts, and some species can even utilize different types of roosts. For example, cave roosting bats are generally found in caves, but they can also utilize bridges or culverts for roosting. Bat behavior also likely contributes to the observed differences in bat communities, as some bats migrate in fall and spring, some hibernate in winter, and others remain active year-round. However, we're likely going to see changes in bat communities as the ecosystem changes. Specifically, climate change is likely going to lead to range shifts due to changes in land use, such as urbanization, increased agriculture, or development of wind turbine facilities, and the increased occurrence of extreme weather events such as flooding, drought, or extreme heat and cold. The figure in the bottom right is a bridge in Houston, home to Brazilian free-tailed bats, before and after extreme flooding forced the bats to relocate. These environmental changes are likely going to lead to range expansion, shifts, or contractions, and has already been reported in several studies. This figure to the right is from a 1996 study predicting changes in bat species richness of tree roosting bats in Texas under 2 climate change models. So the top map was the species richness under the current climate model, and you can see even without seeing those little numbers, you can tell that the lines and everything changed under both climate models, and this was back in 1996. With advancing science and research, especially with genetics, previous range maps of bat species may be due to misidentification and lead to false ideas of range changes. A recent study detected western red bats in central Texas due to genetic analysis, where it was previously thought only eastern red bats occurred. But it's unknown if this recent finding is due to a true range shift of western red bats or a previous misidentification. Also, bat communities are going to be representative of changes, altered ecosystems or ecosystem function. And so the ecosystem may not respond to changes that we see in bat communities if bats arrive to take the place of bats that leave. So it's important to note that while we may see changes in species ranges, we may still have a healthy and functioning ecosystem. I also want to point out that there is bias in this study, as well as with many others, especially in relation to the scale at which we collect or record variables. Bats are volant animals. They can traverse large areas within a single night. And it's unsure like at what scale habitat variables are going to be important to bats. Weather also affects bat activity at an hourly scale, but also seasonally, as evidenced by migrating and hibernating species. Bat communities also likely vary temporally, and the patterns in which they differ are going to depend on the scale at which you're measuring them. So comparing bat communities from one day to the next day are likely not going to reveal many changes in the community, whereas seasonal comparisons are much more likely to show changes in the bat communities. There's also bias with the method of collecting the data. So acoustic monitoring is typically less field intensive than live trapping, but there are differences in the species detection probabilities between monitoring methods and between species. In addition, there are discrepancies in automatic identification programs, and it's generally recommended to have identifications manually verified by an expert, especially for difficult to distinguish species. In conclusion, the bat communities differed seasonally and spatially across Texas, and they will continue to do so as the land use changes and extreme weather events continue to occur. However, this study can serve as a baseline for targeted studies focusing on specific species, monitoring or treatment of white nose syndrome, and wind energy development. So by knowing where specific species can be found, future research can be focused on specific areas that are already known to have those target species present. I want to thank Texas State University, Vesper Bats, and Texas Parks and Wildlife for funding, and everyone who has helped on this project.
We have a few minutes for questions. Bit about the accuracy of the auto detection, the acoustic detection, identifying species. Yeah, so you would ask if I could talk a little bit about the automatic identification success and identifying species. So I can talk a little bit about that. I might push it off to some others who know a little bit more, but I know we use Sonobat to automatically identify a species, but some species that are very similar in their acoustic like characteristics can be difficult for that program to differentiate between, and it might just group it together like, oh, this is just a Myota species. And that might differ from that program's learning to other automatic identification programs that you use. And so some of those you might say, oh, this looks like it is a cave myotis, but someone who manually looks through all of those might look at it and say, nope, that's a misidentification. That's actually a different species. Other questions? So our next speaker is Jared Haney. He's going to be talking on mapping potential habitat for the endangered tricolored bat in Central Texas.
Go up. All right. Hello. As he said, I'm Jared Haney. I'm a biologist with ICF, and I'm going to be talking to you about our efforts to map potential habitat for the tricolored bat in Central Texas. So the tricolored bat was proposed for endangered status in 2022 due to declines from white nose syndrome, habitat loss, and climate change. Myself, some of my colleagues at ICF, and the good folks at Cambrian are helping to develop the Belcour Regional Habitat Conservation Plan. It's in Bell and Coryell counties, just north of here. And I've been told that everyone knows what an HCP is, so I probably don't need to go over that anymore. But the tricolored bat is one of 10 species that we're looking to cover for the Belcour plan. And this area, Bell and Coryell County, is very understudied for the species, and most of the species that we're looking at for the plan, there's very limited occurrence data. And so we needed a way to identify areas of habitat in Bell and Coryell County without utilizing occurrence data. So what do we know about tricolored bat habitat? Well, we know that they roost in trees, near constant flow water bodies. they show some preference for oaks, elms, hickories, red cedar, and maples. They're very small bats, and they tend to roost in tree splits underneath leaf litter or in Spanish moss, vines. And they forage at the interface of the forest edge and the constant flow water bodies. That includes rivers, streams, ponds, and reservoirs. We also know that they use karst caverns for their winter roosting sites. However, Central Texas and our plan area is considered a year-round active zone for the species, so they do not hibernate in our plan area. And unlike some other bats you're probably more familiar with, these bats are sort of solitary in nature. They tend to roost alone or in very small groups, both during the summer and the winter. So our project goal was to map suitable habitat for the tricolored bat within the Belcourt RHCP plan area. We do this for a few reasons, the most important of which is to support our impact analysis. We need to understand how we're going to impact these species and where impacts are likely to occur, and also to inform our conservation strategy so that we can mitigate for any impacts that are going to occur. We derived our habitat parameters from the literature, and we use the National Land Cover Database for our land cover. We created 3 maps, one for summer roosting habitat, one for winter roosting habitat, and one for foraging habitat. This paper is where we pulled our habitat parameters from, Cable and Wilcox 2024. They monitored the summer roosting and foraging locations of tag bats in Tennessee. They exclusively targeted pregnant females, and you can see it's a pretty small sample size, only 4 bats in the study. But they found that the bats exclusively roosted in deciduous trees. Those trees were on average 19 meters tall. And at the landscape scale, roosting habitat was dependent on proximity to water. That's within 30 to 338 meters of rivers and streams. And also upon proximity to major roads. Sorry, I look sideways from here. So proximity to major roads, they were at least 2,400 meters away from major roads and up to 10,000 meters away. And then elevation, slope, and aspect were also important. For foraging habitat, they found that the maximum nightly distance traveled for these tricolored bats was up to 9,000 meters away. That's about 5.5 miles, with a median of 2,232 meters. And that the foraging landscape included areas with a higher percentage of water, less development, and less shrub and early successional cover. So our first map here is the summer habitat map. We took the parameters provided by Cable and Wilcox. So we selected for trees that were at least 19 meters tall and within 338 meters of water features. We then excluded those trees that were within 2,400 meters of major roads. Those 3 major roads are I-35, SH-190, and SH-84. And because they didn't seem to affect how our map looked all that much, we discarded elevation, slope, and aspect. With the summer roosting habitat map, we created a foraging habitat map. So essentially, we just buffered the summer roosting habitat map by 500 meters and considered this core foraging habitat. You remember I just said that the bats can travel at least 9,000 meters away and probably further on a nightly basis. But if we buffered our roosting habitat by that much, this entire plan area would turn out blue. So in this map, you can see the pink or purple summer roosting habitat buffered by the blue foraging buffer. And then this one was pretty simple. The winter habitat map, we just mapped exposed karst. Kimball from Cambrian helped us stitch together the map. And yeah. And so this is our final product. This has all 3 maps laid on top of one another. So you have the green, that's the summer roosting habitat. The beige is the winter roosting habitat, and the blue is foraging habitat. So results. Summer roosting habitat was found mostly along major water corridors, which isn't really a surprise since that was one of the inputs in our models. But you can see notable concentrations around the reservoirs. You got Belton Lake and Stillhouse Hollow Lake, as well as in Fort Cavazos and spread up in northern Coryell County along the rivers up there. Winter roosting habitat was concentrated in western Bell County and scattered around the karst regions in Coryell County. And foraging habitat was also focused around Belton Lake, Stillhouse Hollow Lake, and then also scattered around the plan area in the numerous rivers, creeks, and the hundreds of ponds that you can see dotting the entire area. Some of the assumptions we use for this model. So we assume that the habitat parameters from Cable and Wilcox were both accurate and applicable in Central Texas. Accurate because they only had a sample size of 4. So we're assuming that these habitat parameters apply more generally to the tricolored bat. And then applicable in Central Texas, they did their work in Tennessee. We're in Central Texas, so we're assuming that that transfers over as well. Absence of occurrence data does not equal absence of species. We know that tricolored bats occur in Bell and Coryell County. We do have a couple occurrence points for them, but these bats are cryptic, they are solitary, and they can be kind of hard to locate. And then the core foraging distance buffer, again, we used a 500-meter buffer instead of the 9,000-meter buffer. If we had applied that, it would drastically overrepresent foraging habitat. Every great study has its limitations. Of course, these bats are also known to utilize artificial roosting structures. You can see here on your right, this is a tricolored bat roosting in some sort of pipe or culvert or something. Again, exclusion of larger buffer areas for nightly movement. And the Cable and Wilcox sample size of 4, that kind of underscores the need for more survey data in this area. Species behavior, bats are highly mobile, as you're probably aware. And they have generalist habitat tendencies. They can roost in trees, they can roost in cars, they can roost in man-made structures, which can really complicate defining habitat boundaries. Ongoing improvements, of course, having more occurrence data, more surveys on these species in Central Texas in general and in the Belcour plan area would help refine our model drastically. In conclusion, our comprehensive modeling approach helped lay the foundation for conservation planning in the Belcour plan area. These results highlight not only areas for projected impacts to the species, but also highlight key areas for conservation. And we like to think that this model is pretty simple and can be easily applied to other areas in Central Texas. And the last thing I'll say, species occurrence data, as we've heard a few times today, is limited on a lot of species of concern. And so habitat mapping is going to be an effective way to continue conservation planning. And that's it. Yeah, so your question was for the roost tree, the height of the roost tree that we were looking at. He was asking if the lower limit was 7 meters and we used a 19-meter average. We have not looked at what it looks like if we lower that tree height. I've been thinking about that because I knew a question like that was going to come up. But I'll have to work with our GIS team to take a deeper look. Mhm. So her question was, we're using data from a small sample size where they targeted pregnant females. There's the opportunity to look at other demographic groups in the tricolored bat within the species. And that's probably a good idea. This is the only paper that's tracked bats and given us habitat parameters. And they targeted pregnant females because of their increased energy requirements and how critical it is to understand their roosting and foraging habitat. But we're happy to expand. We just require the data. Yes. Mm-hmm. Yeah, that was another question that I anticipated. So her question is, we have the winter forage or winter roosting habitat. It's just the exposed karst in the plant area. And that was kind of an afterthought after we produced the other models. And we brought that in just to show some wintering habitat. We could refine that by limiting the distance away from roosting trees. It's probably a good idea. I'm open to any suggestions you have, so please. Anyone else? You.
Our next speaker is Julia Wilson. She's going to be speaking on new records of tricolored bats, maternity roost, and silviculture landscapes.
Nothing like a microphone to make you feel really short. Also, sorry to break your hearts. Julia is sick and can't be here today, so you guys are stuck with me. My name is Halen Hollabaugh, and I'm a biologist with SWCA Environmental Consultants, and today I'm going to be talking with you guys about some new records of maternity roosts in silviculture landscapes. We got a little intro already, but I'm going to give you some more fun facts anyways. The tricolored bat is our tiniest bat that we have here in the eastern United States, weighing in as little as 0.16 ounces. They get their name from their distinctive 3 colors of their fur. They are right now proposed endangered under the Endangered Species Act, hopefully with a listing coming up soon, a final decision. Who knows? A lot of further research, as we've heard like 5 times now, is needed for these species. They are such habitat generalists, and we really don't have a lot of baseline data. So they are known to occur in 39 states, and they are active year-round in 10 of those. Okay, cool. So this hashed line right here is all of their hibernating range, and these solid colors down here in the blue and orange are where they are year-round active. They do exhibit some different behaviors in their different areas in year-round or hibernation ranges. So in their hibernation range, they will more likely roost in caves or culverts and bridges in the winter, and then in the summer, more likely to roost in trees. However, they kind of do whatever they want. But in year-round, they will use both of those habitats a little more equally throughout all of the seasons. Their average home range, again, guesstimated around 1.5 miles from their roost tree, and that will be where they're more likely to commute and forage and roost within that area. So right now, the 2024 guidelines list roosting habitat as woody and forest areas. They will roost in live and dead leaf clusters, Spanish moss, and beard lichen. The trees they typically will roost in are at least 4 dBH, most commonly in oaks. They will also sometimes roost in pines and spruce, cedar, but most commonly they're going to be found in some type of oak tree or that dead foliage. They will also roost in human-made structures such as bridge, culverts, barns, anything with an open bottom that they can squeeze into to try to help regulate their temperature throughout the seasons. In 2023, the guidelines actually did not include industrial pine plantations as roosting habitat. It was labeled as unsuitable. They did amend it later in 2023 and just kind of took that out. However, still not really explicitly listed as habitat. So during our surveys, when we learned a little more about what they're roosting in, from our data during our presence and absence surveys, we were able to determine that they are actually roosting in pine plantations, but I'll talk a little more about that in a couple slides. So current research is mainly based in riparian hardwood bottomland forests. That's where most of the papers and research have been focused. Literature on managed pines is kind of scarce. There is some occupancy data, so there is presence there, but not a whole lot on if they are actually roosting there. We were only really able to find 3 papers that had occurrences in managed pine areas, and the general consensus we gathered from what we read was hardwoods are preferred, and there's not a strong correlation to pine plantations, or they're just generally avoided in altogether. So we performed some presence absence surveys in 2 industrial pine plantations last season, and we came up with new finds. So we did 1 survey in Oklahoma, and we did 1 survey in Louisiana. So for our methods, we followed U.S. Fish and Wildlife guidelines. We did 1 site per every 123 acres. So for Oklahoma, we had 10 sites and 100 net nights. And Louisiana, we had 14 sites with about 84 net sites. We also did a habitat analysis comparison after we located the roost trees to check the land cover in the 1.5-mile radius of the home range and NWI water resources. We also ran a 5x5 kilometer analysis because that's something that's used in clearing thresholds. So we wanted to check out both of those. So for our results, we captured 3 tricolored bats at the pond site. For both sites, they were captured at a pond site, so shows that correlation that they have with the water features. They were, in Oklahoma, all juveniles, all females. In Louisiana, they were all pregnant females. We were able to track at least 1 bat back to a roost. And seen multiple bats emerge, 2 to 3 per site. So our roost specifications that we found these guys in, who can find the cute little guy hiding up there in the leaves. What we found interesting about the Oklahoma site was the DBH of this tree was only 3. It was really tiny and only 12 feet tall, so it was kind of unusual. The Louisiana roost tree actually had more of a normal. We found him roosting, or her. We found her roosting in mid canopy of a very tall pine tree and a cluster of dead pine needles. So these are our roost trees. This teeny tiny little itty bitty thing that looks so sad and small is where we found 2 of them roosting. We were actually able to walk underneath the tree and see them with the naked eye, just curled up in little leaves there. The one in Louisiana, couldn't see her, but she was up there somewhere in the mid canopy. For our land cover comparison, we considered our suitable habitat to be evergreen forest, mixed forest, woody wetlands, and deciduous forest. We use the evergreen forest kind of as our proxy for silviculture. However, it is imperfect. There could be some native pines mixed in there, but it was the best we can do. And If you look up at our 1.5-mile home range, the habitat considered suitable from those 4 categories is over 70%. But if we take out that silviculture, it drops all the way down to 10%. So we're losing about 60% of what could be considered habitat. You can also see the percentages of water features that were within the home range. And with our grid analysis, we see a similar trend to the home range where we have 80% habitat cover, but when we remove the silviculture, it significantly drops again, like over 60%. So it just goes to show that if we don't include these pine plantations as suitable habitat, we actually could be missing a lot of bats and a lot of roosts. Also, for the 5x5 grid analysis, we use that for clearing thresholds. So if we're missing that much habitat, the thresholds for clearing could change drastically and could actually be a little skewed in one way or another. So it's good to keep looking at this data and compiling it all. This is just showing a visual map of what I was saying. The roost tree is that red dot in the middle, and the blue star is our capture site. The roost trees were all within a mile of where we actually captured them, near a water feature. And all of that dark green is the silviculture habitat. So the light green grassland and herbaceous is actually recently cleared pine. It's not really any type of natural grassland at all. So numbers could be even more before that was cleared. Our 5x5 grid analysis shows the same depiction. And I don't think bats care too much about human activity either, because our capture site for the one in Oklahoma was right next to an active sawmill, and it was very loud at night. So in conclusion, we still need so much research done on this species. We are learning new things every day about them with each new season that comes in. But we captured them adjacent to a water feature. So in silviculture, this is probably a good area for foraging. Roots were not adjacent to water, but they were within the home range. Reproductive bats do roost in industrial pine plantations. We were able to capture 5 of them in total. Seasonality-wise, we caught pregnant females in late May and juvies in June. And studies suggest that, again, they're extremely general, and we may have valuable insight to inform regulatory compliances on that silviculture may need to be considered. And we expect to learn a lot more to come in future surveys this season. All right, any questions?
Questions?
So two-part question. The first part was about, oh gosh, I already forgot the first part. So we'll start with second part. Second part is it better to kind of use a more open area or a closed area when you're misnetting? Then the first one was proximity to water, correct? Okay, awesome. So what we have noticed from our methods is that tricolors utilize water more, and they're kind of a high flying species. So in Texas, a lot of our trees just kind of are a goalpost. They don't really make this nice canopy. So by putting nets closer to water, we're able to utilize when they dip down and try to get a drink. So that has worked better for us. So when we miss net, we're a lot closer to water than we would be deploying our acoustic detectors. And then same for foraging. We've actually found that tricolors prefer a more open area or open flyways compared to like a northern long-eared bat, which actually would prefer more butter, I guess you can say.
Question back here.
Yeah. I can touch a little bit on that. We didn't do much with bridges, but I have seen bats roosting in larger bridges, especially underwater bodies with a good riparian area next to it. We didn't find any in any bridges because we weren't really looking for them. But they definitely use them. We've found them in trees and bridges, and they kind of utilize everything all year round down in this year-round range. I think my best guess, because I don't know for sure, would probably be temperature regulation. So easier access to water and resources and maybe able to regulate their temperature better than they would in the other. But I don't know for sure. Any other questions?
So our next speaker is Samantha Levers. She's going to talk on tricolored bats and white nose syndrome in Texas.
Hello, good afternoon, everybody. I hope you're all still feeling nice and awake after having lunch. So my name is Samantha Levers. I'm the State Bat Specialist for Texas Parks and Wildlife. And I'm going to be talking to you guys today about tricolored bats, shocking, and white nose syndrome here in Texas. So some of you may be familiar with white nose syndrome, but for those of you who are not, white nose syndrome is a disease that affects hibernating bats, and it's caused by a fungus called Pseudogymnoascus destructans. Say that 5 times in a row. And we just call it PD. So PD is a cold-loving fungus, and it likes moist places. So it likes dark, damp, cold places. So shockingly, in places like caves. Now, when hibernating bats go into hibernation, they go to a state of torpor. This is where their body temperature drops to sort of ambient temperature. We see a lowering in heart rate, lowering in breathing rate, and it actually makes the bats themselves a good host for this fungus to grow on. And that's what you can see in this picture here. So this is a little brown bat. This isn't actually a species that we get here, but it's a really good picture where you can actually see where the fungus has grown on the muzzle of the bat and also on the wings. So because of this infection, the bats arouse out of torpor much more often than they usually would during winter. And this uses up a lot of energy reserves that they need to be able to survive the winter. So when we see this fungus on the nose and we see this odd awakening and active behavior in winter, that is when we call it white nose syndrome. So it's important to note that a bat can carry PD and not have white nose syndrome, but you can't get a bat that has white nose syndrome that doesn't have PD. So we first found, that was supposed to be a nice time lapse for you. There you go. We first found PD in a cave in Albany back in 2006, where a large number of dead and dying bats were found in a cave. It has since spread, as you can see, south into the west, and it is now in almost all states. I believe about, 40 states as well as several provinces in Canada. And PD was actually first recognized here. And when it was, there was more research done and it was found that it was also actually in Europe and Asia. But the bats don't seem to get as sick there. And we think that the strain that came over here was in fact from Europe. So these fungal spores, they're quite hardy. They can actually last quite a long time. And that's how we believe it came over. So talking about the history of white nose syndrome in Texas, we first detected PD in the Panhandle in 2017, and we found this on cave myotis as well as tricolor bats, and I believe Townsend's big-eared bats as well. PD, the fungus continued to spread. We were finding it in central Texas, over into parts of East Texas, and also West Texas. In 2020, we had a mass die-off of cave myotis. This is the first time we've seen a die-off of the species due to this disease. And it's been hard to track the decline because we have little access or knowledge of high vernacular in central Texas. But the work that we've been doing, actually doing maternity emergence counts, has shown very significant declines. In fact, one of our maternity roosts in central Texas has gone from 18,000 cave myotis in the summer to 0. So we're looking at very, very significant declines. So like I said, white nose syndrome affects hibernating bat species, and we have ones that have had white nose syndrome, but we also have bats that can carry the fungus but not develop white nose syndrome. So for instance, our beloved Brazilian free-tail bat, luckily they don't suffer from white nose syndrome, but they can carry the fungus. And this can cause a problem as well because they will often co-roost with species that are susceptible to disease, such as cave myotis and tricolor bat. And we're going to be talking about our delightful tricolor bats some more. So I'm going to skip through some of this. Tricolor pelage, small bat in the US, it's found in the eastern half of the US, and it's found throughout most of Texas. So really it's just that kind of far western area where we're not finding them. They hibernate in caves and cave-like structures. So this can include culverts. They have been known to roost in bridges as well. We actually don't know a lot about their use of bridges here in Texas. So that is an area of research that we would really like to get some more information on. And then in summer, as you've seen, they primarily roost in foliage or in trees, pretty much anything by the looks of it. So where we stand right now with white nose syndrome, last winter, we found 3 tricolored bats. This was some work with Bat Conservation International and TPWD. We found 3 tricolored bats that were discovered with visible fungus. 2 of these were collected and sent off for analyses, and they both tested positive for PD, and 1 histologically confirmed as having white nose syndrome. So this is the first case of white nose syndrome in tricolor bats in Texas, and it's the southernmost detection of the disease in the species. So white nose syndrome is now confirmed in tricolor bats in central Texas as of last winter. So what do we do? Well, we haven't all just been sitting idly by, and we've been trying to see what we can do to help our bats. And there's lots of different options, lots of different treatments that are being trialed. And generally these can be used in the high vernacular themselves, they can be used on the bats, or it can be a combination of the 2. So for instance, we have polyethylene glycol, which is something that can be sprayed on the substrate of the roost, and it makes it a less hospitable environment for the fungus to grow. Steam cleaning can also kill the fungus. Like I said, it's cold loving. If you expose it to high enough temperatures for long enough, it will die. There's also the use of UVC light. So they use low levels of UVC light, and this disrupts the DNA of the fungus and eradicates it. For bat treatments, we have vaccines. We also have probiotics, which are a mixture of fungi and bacteria that are naturally found on the wings of the bat. So you kind of increase these loads, and this seems to provide some level of protection against PD. And then we can also use volatile organic compounds. So there's a particular one called B3, which is a mix of naturally produced antimicrobial volatile organic compounds, and you can essentially aerolyze both the bats and the roost with this to help kill the PD. So what about treatment options in Texas? Well, when we found PD in Texas, we knew that it was going to spread. So one of the first things we did was figure out how can we protect our vulnerable bats. Now, in East Texas, over 3,000 tricolor bats have been found overwintering in 13 culverts. These are very, very large numbers. We were told before that these bats do not cluster. They roost singularly, and this is a very rare event where you can actually see them clustering in this culvert together. So they're a very, very important population. And as of 2018, we had one positive PD detection at these sites. So we ran a project where we trialed 2 different treatment options at 6 of the culverts and left the 7 other ones as controls. So we tried the polyethylene glycol, the PEG, and also volatile organic compounds, so VOC. And the cool thing with culverts is you don't have to worry about delicate cave ecosystems. It's not like we're going in there spraying stuff and we're going to kill some endangered karst invertebrate. There are more options open to us. So we wanted to see how logistically feasible it was. We wanted to get started with treatment before PD arrived to see if we could even stop it from actually invading these roost sites. So here's some of the great fun we had. The PEG, we would apply that in the summer. You can see there's a picture of me there sitting in the middle with a big backpack sprayer on. I would literally get dragged through the culvert by a winch and spray the PEG back and forth, which is as tiring as it sounds in 100 degrees. And we would do this when there weren't bats present. So they're not at these culverts in summer. The VOC you can see is below. So this was a machine that was passed through and then we winched it back and it aerosolized. This was done during winter, so it actually aerosolized both the roost and the bats as well. One of the really cool things about culverts is you've got all the dimensions, so you can calculate the amount of volume that you need to be able to fill and make sure you're getting all your calculations correct. But that's done by people much smarter than me. So we continued PD monitoring throughout all of this. 2019, negative. 2020, negative. 2021, oh no. We have positive PD swabs, both from bats and substrate in 8 of the culverts. Unfortunately, there was a bit of a delay with the lab that year, and we weren't able to get those results until way, way later. So in 2022, we were ready. We swabbed beforehand, before we did all the treatments, and we were ready to go in and swab again. So we were prepared. We knew PD was going to be there, maybe even white nose. We were wrong. The PD was gone. And I thought, fantastic, the treatments are working. But the PD was gone from the control culverts too, where we hadn't done any treatment whatsoever. So what was going on? Some of you may be familiar with the disease triangle. So essentially, with any disease, you need a conducive environment, a susceptible host, and a pathogen. So we wanted to know, because we had preliminary stuff suggesting that our culverts were within the temperature range for this fungus to grow, what was happening? Why weren't we seeing the development of the fungus? Why had we had these failed invasions? So we conducted some work, and I do have notes for this one because this isn't my one. So Dr. Katie Hess and her student Leah Crowley from APSU measured the temperature and humidity and then determined the mean % of winter that culverts and other hibernacula were in the optimal range for the proliferation of the fungus. And as you can see, the culverts are not in the optimal range for more than about a quarter of the time, and much less so than caves and tunnels as well. So we used this data and we took information from another study to calculate the amount of fungal growth required over the course of winter to result in tricolour bat mortality. So essentially, Maguire et al. Found that white nose related mortality occurred in little brown bats when 8.4% of one wing was covered in PD. So we essentially determined what the proportion of growth would be needed on the wing of a tricolour bat to see potentially white nose related mortality. And we used growth rate equations using both temperature and humidity to do this. And what you can see is we found a threshold there. So essentially, PD would need to be on a wing at about 1.55 centimeters squared in order for it to potentially result in white nose syndrome mortality. And as you can see, with our culverts, only about 25% of them would allow for that level of growth during a winter. So there's actually a good chance that the microclimate itself is not conducive to the growth of the fungus in most of our culverts. But what about the susceptibility of the host? Well, in the same study, we put temperature sensitive telemetry on some of our tricolour bats, and we put antennas up throughout the culvert, which is just as much hard work as you would imagine it would be. And for this reason, we were able to look at bouts of torpor, so torpor length, activity, and we found that these guys are really active. So they're not going into a deep, deep torpor. They're not going into that very, very cold state that the fungus wants in order to be able to thrive. I also did a dietary study, a winter dietary study with the tricolour bats at this site, and they are eating quite happily. So there is every chance that even if they are aroused due to any kind of infection, then they're arousing naturally anyway, because remember, this is taken at times when we don't have PD. So this isn't behavior and this isn't eating behavior that's based around a PD infection. This is baseline data without PD. They are naturally arousing, active, and hunting throughout winter. So what's the future of tricolour bats and white nose? Well, there seems to be generally a delayed initial progress of PD across the state. So like we said, we found it in 2017 in the Panhandle, but we didn't have a confirmed case that we know of in tricolour bats until this winter just gone. Now, there is some evidence of steep declines in some of the Central Texas caves that Bat Conservation International have been looking at. But it looks like there is a good chance that the die-offs that we do see in Texas are cryptic. In the Northeast, people would go into caves, they would see dead and dying bats. We don't have any of that. And it could be because of, frankly, the sheer size of Texas and being able to get to all these sites, and also just getting access to the sites as well. We're heavily privately, land is very heavily privately owned. So it could be that we are just not able to see these die-offs as they have been seen in other states. It doesn't necessarily mean they're not happening. Oh, I keep clicking, sorry. We also have milder, shorter winters. We usually have almost year-round insect activity here in Texas. We've got shorter torpor bouts, at least in East Texas. We know that they're eating over there. So there's no real reason to expect that we wouldn't see something similar in Central Texas. But that's very easy to say because we expected that with cave myotis also. And cave myotis died off in Central Texas, not in the Panhandle, which is where we thought it would happen. So essentially, we are on the leading edge of PD expansion into the southwestern United States, and we're seeing abnormal pathogen dynamics based on things that we've seen in the Northeast for the fungus. And it doesn't follow our modeling prediction efforts either. So we're learning more, but right now we actually can't be confident in what we're going to be expecting for this species. And it's going to be a case of being very different depending where we are in Texas. So we are funding and collaborating with research efforts across Texas and the Southwest to try and understand these variations that we're seeing in white nose susceptibility, not just in tricolour bats, but in other species, and why we have this variation both between and within species and with different roost types and latitudes. And there's a lovely picture of some furry chicken nuggets for you.
Time for questions.
And yes, I am petitioning to have their name changed to furry chicken nuggets. Oh, wait. So the question was, are all treatments preventative, or are they something you can apply after white nose syndrome? So I think the best course of action is always prevention. And usually if you get to the point where you're actually seeing the fungal growth, that bat is kind of beyond the stage of really being able to help it. Like I said, the bat can still have the fungus without actually having developed the disease yet. So there's, you know, if a bat has a fungus, you can still apply treatments such as the probiotics or similar to try and help them fight it, basically. But yeah, by the time you're seeing a bat that's in the stage where it has like really prominent fungal growth, there's not really much you can do to come back from that. So she was asking how effective the treatments were and whether we would recommend them and when those recommendations would be available. And as you could see from it, we weren't actually able to test the efficacy because it seemed to kind of sort itself out, frankly. It seemed like there was a failed invasion. And for that reason, that's why we went into looking at all the microclimate options, looking at hibernation behavior and stuff. Logistically and monetarily, I would say that the VOC was the better option rather than the PEG. The PEG required a huge amount of physical effort, but the VOC does require quite a lot more of specialized knowledge. So I think it is something that could be suitable for culverts or other sort of hibernacular that don't necessarily have like a very delicate kind of ecosystem involved with it. You know, I don't think volatile organic compounds are necessarily going to be suitable to use in like a cave environment, for instance. But like the enclosed area of the culvert and all the known dimensions made it much, much easier to actually treat in there. And I think it would be a suitable option should we see white nose develop in some of these, you know, few culverts where it looks like it might be able to. I'm not sure how to repeat that. Okay, so the question was really, do survivors shed the fungus when they leave the hibernacular, and what is the concern around that? They do seem to shed the fungus as they leave. Now, some of the issues that we have is even if a bat survives, the PD can still cause significant damage to the wings. And I think there is a good chance of like a secondary cause of mortality through that wing damage as well. So I think that's something that we need to consider. Generally, the thought process is more that the fungus is staying on the hibernacular substrate. And, you know, we do have some work that we're doing with Bat Conservation International looking at the pattern of, you know, PD growth in the substrate. And indeed, in some years, we've actually seen PD increase during the summer. So the idea is more that it's going to be contracting the fungus again once it's getting back to its hibernacular. Thank you.
So our next speaker is Sarah Fritz. She will talk on the green dilemma, reducing bat fatalities from wind energy production.
Okay. Yes, hello, I'm Sarah Fritz, as you mentioned. I'm the director of the Wildlife Ecology Program and an associate professor at Texas State. So I'm gonna switch gears just a little bit, still the really cool taxa of bats, but now we're gonna talk about another issue and how Texas is somewhat unique in this issue. And this is a green dilemma that's not only affecting Texas, obviously, but this is a worldwide societal issue that wind turbines have the unintended consequence of killing bats. So we need wind turbines, right, as a mitigator for climate change, because climate change also is killing bats. Just like with all the taxa we work with, there are direct and indirect effects of climate change. One of the most shocking, I think, direct effects was a heat wave that hit Australia several years ago, and it killed 1/3 of one of their species, the spectacled flying fox, and they were literally just falling out of trees because their critical maximum temperature was hit. But droughts and aridity also are affecting bats, as many species are closely rely on water sources. But it's not only the heat and the drought. So all of us in here probably went through Snowmageddon several years ago, and we survived. Unfortunately, many of the bats did not. So a lot of the Brazilian free-tail bats that typically would roost in caves, and caves have a more thermally stable environment, now roost in a lot of culverts and bridges, and a lot of those died. I was actually at the Wimberley Bridge when this was occurring, and these bats were literally just falling from the bridge. They were not dead yet, but they were completely unable to fly. Even with sustenance and food, they just could not get rehabbed. So hundreds of thousands of bats were estimated to have died during that. So that's another indirect effect of climate change, or direct effect through the cold snap. So renewable energy, we need it. And we are proponents for wind and solar and all of the things, but all of them have some types of unintended consequence. For wind energy, first, it's increasing. And in fact, in 2024, it surpassed coal. This was not the first time, but the first time kind of more longer term. And you can see that in the blue line here with coal in the gray. And in 2023, it comprised about over 10% of our electricity consumption in the US. And that's great for climate change. And a lot of the wind energy is coming from Texas. Texas is a leading producer of wind in the US. Probably many of you have been driving down the road and been cut off by wind turbine blades and the large trucks towing those and trying to make right-hand turns, which doesn't work very well. But you've seen how big they are. And if Texas was its own country, it'd actually be the 5th largest wind producer in the world. So you can see from this heat map of where wind turbines are in the US, heavily focused here in Texas. So great for us, good paying jobs, great for our socioeconomics, bad for bats. Texas is also the leading, as you heard earlier, leads the states in species richness and also just population sizes. We're home to the densest colony of any mammal anywhere in the world. And that's where this picture was taken, which is Bracken Cave, about 2 hours south of here, just north of San Antonio. And this cave, now, okay, little alert, spoiler alert here, this species is not super heavily impacted by wind turbines. Any bat species can be killed. And I'll get to that in a minute. But I do need to nerd out just a little bit on these bats because they're very cool and very important here in Texas, not only for tourism, but also for agriculture. So these bats eat a lot. Just this one cave, which is a maternity colony, again, Brazilian freetails, about 18 to 20 million bats. They eat about 140 tons of insects every single night. Every single night. So who in here can think of what 140 tons of insects looks like? No? I'll show you in a minute. First, I want to show you, this is Doppler radar of those bats leaving that cave. So this is taken out of your weather app every single night throughout spring and summer. And this is caves, mostly Brazilian freetails, from leaving caves and culverts in central Texas. And that red circle is that one cave that I showed you a minute ago, Bracken Cave, north of San Antonio. Okay, so ready for the 140 tons of insects? Some of you might have seen this before, but it's still the best analogy. The weight of 70 F-150s. Here in Texas, we all know what an F-150 weighs or looks like, not what it weighs, but we know what it looks like. Most of us have ridden in those. So it's the same weight, and that's just one of the caves. Still don't understand 280,000 Whataburgers. I mean, you all have held a Whataburger, right? So, and one of the main, or two of the main food items for this species are two of the main crop pests for cotton. So they are saving cotton farmers here in Texas millions of dollars. Okay, back to wind. First, bats and birds differ in a lot of ways on how they are affected by wind energy. First, birds are more opportunistically hit, it's thought. So if you were doing carcass surveys under wind turbines, you could find a lot of different bird species. Whereas interestingly, there's only really, well, any bat can be hit, and they are, but there's 3 species that comprise about 75% of all known fatalities in the US. So that's a weird pattern. There's some other weird patterns that I'm going to show you, and all of these together have led to the hypothesis that perhaps bats are actually attracted to wind turbines. That's problematic. So the 3 species that comprise most of the fatalities are the hoary bat, the silver-haired bat, and the red bat, the eastern red. So different than our cave-dwelling species, which are often the ones I just showed you, very gregarious, obviously, these are solitary tree-roosting bats. These bats' mating season is in the fall. And you can see, this is a paper we have in review right now. This is from carcasses from wind energy developments across the US of the timing of fatalities throughout a year. Now, this isn't a new trend that we just found. This is supporting a trend that we've seen for a long time, which is that most bats are dying from July to October. And this coincides somewhat with these species' fall migration. But migration is a two-way movement, right? They're not being killed in the spring. So this plot is showing you the number of fatalities, and it's split by species, with the first one being eastern red bats, and the second one being hoary bats, 2 of the biggest bats that are being killed, or not biggest, but comprise most of the fatalities. And it's also worth noting that there's still a lot of unknowns because a lot of the wind farms are on private lands. And so we don't get access to that data. Even though a lot of places do post-construction monitoring, that data is not shared with us. That's problematic because as biologists, we're trying to find solutions, but we don't even know the actual problem and how big it expands at this point. But anyway, these two things, so most fatalities are 3 species. They're happening during the fall. Weird, right? Okay, some other things. This is another study that my lab conducted. This is from South Texas. The U.S. Fish and Wildlife Service has a voluntary tiered approach to siting for wind farms, and that includes monitoring bats with Met. Well, they're monitoring weather at Met towers, meteorological towers, and that's the towers you can see here. And on those, they place microphones, and these microphones can pick up bat echolocation, and that's the common way to monitor bats pre-siting for a wind farm. And those echolocation calls can then sometimes be used to identify species, not always, and also just give us a form or an estimate of activity. So one thing that's also weird in this pattern is that post-construction fatalities are not estimated by pre-construction activity. So in fact, there's a lot more fatalities that are occurring than is estimated using these metrics with the microphone before a wind farm is constructed. So another thing that's pointing to are bats attracted to wind turbines. Okay, so this study, we supplemented microphones with thermal cameras. Now, this isn't always done. This was just something that we did for our study at a couple Met towers in South Texas, and we found some weird stuff. So the microphones, obviously, we can't tell where the bat is in relationship to the Met tower, but with the thermal cameras, we can. And with the microphones, we can't tell how many bats are flying around, but with thermal cameras, we can. So what you're looking at is overlays of 5 minutes from the thermal camera imaging. And the top photos are 3 consecutive nights. And you can see this pattern where they're actually changing their flight, like where their flight focus is on the Met tower. So when we started looking at that, we zoomed in and realized they're coming from downwind. And we can tell that from the weather vanes that are on the Met tower. So they're approaching from the leeward side. So that's interesting. And then we found this other phenomenon that when there's one bat in the flight space, they're kind of flying, doing loop-de-loops all over, and then a second bat enters the flight area, and they all of a sudden start targeting their flight again to like one specific spot. So we started thinking, are they scent marking these Met towers? We don't know. And if bats are attracted to wind turbines, they're probably using a variety of proximate and ultimate cues, depending on how far away they are. But it's also worth noting that the tree bats, the 3 species I mentioned earlier, these often will find the largest tree on the landscape to set up mating areas. So that's a possibility too, that they're mistaking these wind turbines for trees. Lots of people have been trying to figure this out, but we still don't have an answer. So a couple of other weird things here. We've known for many years that bats might not always be echolocating in flight. It was thought that, for a while, anytime they're out to be able to discern their surroundings when it's dark, that they're echolocating. But that's not always true. So again, that's been known for a while, but at that same study that I just showed you, we were also comparing the detections with thermal cameras versus acoustics. So here's 2 patterns we saw, both temporal, but one's on a nightly basis and one's on a yearly basis. And what we saw here, as you can see in the bottom plot, is that thermals and acoustics, so what you're seeing in the black line is thermals, and there's a dotted line. It's a little hard to see on this screen, but the dotted gray line is acoustic detections. And where it's gray is where the detection or the activity of detections overlap. It's the same. But you can see at about 2 a.m., there's a lot of thermal detections and not nearly as many acoustics. So often we see this trend in acoustics that bats are echolocating in a bimodal distribution at night. So they're flying out of the roost, they're hungry, they eat. Maybe they're out just hanging around, hanging out, quite literally. And then later in the night, they go and eat again. But with thermal cameras, that's not true. They're just not echolocating during that time, at least at the study sites. And then seasonally, or throughout the year, we kind of saw something interesting as well. So we have thermal camera detections, or a metric of that in the orange triangles and acoustic detections in blue circles. And they're not matching during those time frames that bats are being killed. So through July through October, you can see that the thermal detections were way greater than the acoustic detections. So for some reason, they're not echolocating, potentially not echolocating during this time frame. We can't say that for sure. It could be something else that's going on. Who knows? Maybe they sometimes do micro calls, so they can do pretty small calls, quiet calls. But either way, our microphones are not picking them up. So regardless if they're attracted to wind turbines or not, this method that the U.S. Fish and Wildlife Service is using right now, which is a great method for a lot of things, but it might not be working to estimate bat activity for pre-construction for a wind turbine farm. So why is this important? Well, for a lot of reasons, right? For the bats, but also just for wind energy. We're clearing a lot of land and putting in these wind farms. Sometimes they're shutting down, and this is actually occurring quite often. This is the first wind farm that had a really big shutdown. This was a few years ago in Missouri, and they killed 3 Indiana bats, and that was a protected species. We don't have that species here in Texas, but because they killed those, they voluntarily shut down for 6 months, all night, every night. So that's great for the bats, not so good for climate change because it reverts the use of electricity back potentially to fossil fuels. Not good for socioeconomics or the good paying jobs that wind energy creates as well. And here in Texas, as you've heard the last few talks, the tricolor bat maybe will become listed, and that range overlaps with a lot of our wind farms here. So that could happen here in Texas, and probably down the road, as more bats are succumbing to both wind energy, white nose syndrome. It is typically different species groups that are being affected, but still, no matter what, if a species is protected, then incidental take is an issue, right? So solutions or potential solutions. So the first one, curtailment. So that's when a wind turbine shuts down, often during specific environmental conditions or different times of the year. So there are voluntary guidelines that the U.S. Fish and Wildlife Service has put out for tricolor bats and other species that are protected or about to be protected. But this is the big one for here in Texas. And basically, certain times of the year, reproductive timing at certain wind speeds, like low wind speeds, and times we think bats might be really active, they curtail, they shut down. Again, great for the bat, not so great for the climate change mitigation measure of renewable energy. So another possible solution are acoustic deterrents. And so deterrents have been manufactured to emit ultrasonic frequencies that are very similar to bats' echolocation. And it's thought that those either jam bats' echolocation or at least in some way make the airspace unattractive. Maybe they're just, it's like being at a heavy metal concert and it's really loud in your ear. And they've been shown to work. In fact, an in situ study in South Texas showed that they decreased fatalities of some species 50%, 76%, but not all species. So they're working for some. They're a little bit expensive to put on wind turbines, but in the long run, the profits can continue, wind energy development can continue. So we've been testing some deterrents in my lab. We have a flight cage where we actually test deterrents, and we saw some interesting patterns there as well. There were some species differences as expected, but there were also seasonal, sex, and sex within season differences. So for example, one of the deterrents that we worked with, we saw that red bats, during the fall, the deterrents didn't work at all. The bats were not moving further away from it in the flight cage. But during spring, those males were out of there. So whether it's some type of reproductive behavior that's happening, we don't know. So one of the things about deterrents, though, in the past is they are typically mounted on the nacelle of a wind turbine blade because they need power. So they're powered through the nacelle. And this is problematic because wind turbine blades are getting longer, longer, and longer. And so just the basic physics of sound, these ultrasonic noises do not travel that far in space. So it's not covering the tips of the wind turbine blades. So in my lab, I have 2 students here that have been testing 2 other deterrents. One that they're trying to get to be a passive deterrent, and one is a passive deterrent. It was just developed at Iowa State University, and this was the first field trial at some ponds in San Marcos. where it could be actually installed on a wind turbine blade, and as the blade spins, air is moving through it like a whistle. Now's the hard part of maybe getting the manufacturers to actually do that without voiding their warranty. And going back to, revert the old wind turbine blades into these, or is it just going to be something they put on new ones? So there's a lot of moving parts to this issue. Another really interesting thing is that most of these in the studies that have been done, they were working pretty well, but they've been on top of the wind turbines because it was thought that's where most bat activity is occurring. But now through some of the really cool thermal imaging and FLIR imaging detections that they're doing at wind farms, this is one of my colleagues from Wildlife Imaging Systems, we're seeing that a lot of the bat activity is actually really low. This is all under the wind turbine. It's kind of hard to see the scale of that, but it's happening lower to the ground. So perhaps just the placement of deterrents lower to the ground, or maybe on the on its own stalk, or just on the sides of the wind turbines can help as well. So we still have a lot to figure out. And again, Texas is somewhat unique as the largest producer of wind, and we have a lot of the bats here that are being killed, but there's a lot of people working together, thankfully, to try to figure this out. And I can take questions. I think. Yes. Yeah, so the gentleman asked if it was possible that the lights could be attracting invertebrate prey that could be attracting bats. So a lot of studies have been done. So this has been studied for a few decades now. Studies with lights, the noise, just the movement of wind turbines as foraging grounds. Nothing is pointing to an absolute solution or knowledge right now of what's causing it. So no, it does not seem like it's lights. Yes, Jared. He asked if all deterrents were acoustic. Up until recently, yes. Recently, I heard they're trying to develop some type of light deterrent, even though lights on the turbines right now are not attracting bats. It doesn't seem. So I don't know where they're at in that, but they are trying to have other types of deterrents as well. But so far, all acoustics. Interestingly, they form these on the basis of tiger moths, which tiger moths as a bat prey item emit their own echolocation, and it's thought that it jams bats' echolocation, so to deter bats from being able to capture them as they're like triangulating in flight and homing in on a prey item. So that was what was used to develop the ultrasonic deterrents, because if it can jam their echolocation, make it a non-attractive breeding ground, and just kind of make them disoriented that they leave. Okay, thank you. Yeah, sure. So he's asking if it's the bats that are actually being struck, or if there's what's like a barometric pressure is one of the issues. Mostly struck. There have been studies with barotrauma, and that does possibly account for some. There's a little bit of competing thought on that, but likely some could have barotrauma, but a lot are being struck. And you have to remember they're moving.
So our last speaker before the panel is Tracy White. She's going to talk on exclusion of bat roosting habitat in TxDOT bridges, lessons learned.
So many lessons, y'all. It's been a lot, but that's okay. So today, let me think about what. Like I said, we've learned a lot today. You've made it to the last presentation. Congratulations. And you're lucky for you. I'm going to pull the curtain aside, and you're going to look into the bowels of TxDOT. And what we're doing, I'm just kidding. What we're doing for these bridges that are in our project areas that have bat colonies. And we're really the first district in the state that is doing a large scale bat exclusion. And it's been a lot. But I will tell you the good thing is that Austin is a bat town. And so it makes it a lot easier for people to be looking at the bats and watching out for the bats and having that input. And so it's nice to be in the Austin district, but it has been very challenging. And the project that we've learned a lot on is the Capital Express North project. I can't walk away. And so that essentially goes from SH 45 North down to US 290 East. And within that project area, we've got two bridges, Howard Lane and Wells Branch Parkway, that have Mexican freetail colonies. And they're not small colonies. And I feel like there's 4 billion bats there, but really we're looking at about 10,000, 20,000 bats. They are maternal colonies. And these two bridges are box beam bridges. That's why the bats are there. So I don't know if everybody knows what a box beam bridge is, but it's essentially kind of elongated concrete. You lay them down next to each other, spans the bridge, and it creates these gaps that are perfectly suitable and great thermal kind of areas for these bats to roost in. The third bridge within the project area that I want to mention is the Walnut Creek Bridge down at the southern extent of the project. That would be a great place to put in box beam bridges, but the span is too long. We've talked to the engineers. We can't just put in box beam bridges. So it's currently an I-beam bridge. It's going to be rebuilt as an I-beam bridge. But we have put a lot of alternate roosting habitat there. We put in bat boxes, which I'll talk more about in a minute. But the two bridges, Howard Lane and Wells Branch Parkway, are box beam bridges, and they're going to be built back as box beam bridges. So the first hurdle is dealing with engineers. And, you know, things show up and it's like all these boxes on paper and you're like, oh my God, what's happening? And so lucky for me, some of these engineers, I mean, they're Austinites. They appreciate the bats. So they show up and they're like, yeah, bats, this is great. They eat insects. And so the other engineers that are kind of not in the bat camp yet, you can kind of bring them over into the bat camp. And so by the end, they're making bat boxes. They're really happy to design them and talk about them. And we end up with a really great plan, a really great bridge model, an approach to things. And it's great. So if the engineers didn't start off in bat camp, we're hoping that they come over and they stay in bat camp. And so in the future, when these bats, well, these engineers, these bat colonies and bridges show up in future projects, I'm hoping that the engineers kind of keep this in mind and start incorporating more bat habitat into their design. It's not this foreign concept of like, why do we care about the bats? And so they carry that forward, I'm hoping. So what starts off, you know, boxes, ends up to a really great plan and everybody working together and moving forward. Like I said, Austin is really a great bat town. We understand bats. We have a bat festival. I mean, McNeil Bridge has its own website, like a Facebook site, I guess. But we get bats here. So once we get that plan from the engineers, this is when we invite the bat stakeholder groups. We get Bat Conservation International, Merlin Tuttle. We get Texas Parks and Wildlife to come in. And so we get all these different groups to come in. We give them a presentation. We present what is going on. And a lot of times these projects are years out, years in advance. And so we don't have, we kind of have I don't want to say a concept of a plan, but we have a plan going forward. And so we present that information, but these groups are very important because we want their feedback. And what's really great about these bat groups is that the public comes and talks to them. So as long as we keep them informed about what's going on, you know, they can help us get the word out to the public if the public has any questions. It's also nice, and I'll talk about the contractors here in a second, but it's nice to have an extra pair of eyes out there and have people watching the contractors and what they're doing. And so it's really, really nice to talk to the stakeholders, get their input, make them aware of what's going on, and they can kind of be the eyes out there and help spread information. All right. So this is the hardest part. Dealing with the contractor is the hardest part of this whole situation. So usually we have a plan, you know, we get done with the design,
we get done with engineers, we present the information to the stakeholders, we kind of have a plan going forward of how they're going to approach construction. And then we talk to the contractor and they come on board, and TxDOT is very notorious for means and methods. We don't want to like tell the contractor what to do, but we kind of have to when it comes to these bat bridges. And so the contractor will come in, we have a plan, we know when they're going to demo, or we kind of think how they're going to proceed with demolition of these bridges. And then they come in and turn everything on its head. And so it requires me, it requires us and my community of helpers and emotional support group really to be flexible and creative. And so the contractor will come in and we'll have a meeting, you know, we'll kick off, everything's rosy at the beginning, and you have a kickoff meeting and you talk to them, you're like, hey, there's bats. You're gonna have to have some restrictions. Like you can't just go and do whatever, how fast you wanna go. You're gonna have to kind of pump the brakes on some things and think about some stuff and plan ahead. And so it's really hard for contractors to really hear that at the beginning of the project because they're just excited about getting going and whatever. But then months later, they'll actually kind of read the fine print and the epics and the general notes, and they're like, oh my gosh, we got to do stuff. And so it's tough. And then you have like, you know, they come 3 months before they want a demo, or you know, they want to do exclusion in a weird time of year, and you're just like, oh my gosh. So it's really about managing expectations, and the contractor kind of giving very conservative dates and just being really communicative with them about what's going on. And so because that contractor, they're going to want to go as fast or as slow as they want to go. They don't want anything to get in the way. And apparently I just pee all over everybody's plans. So, but it's really hard. But you know, I've got a good group around me, a good community that kind of helps me stay strong. And you know, I try to be nice, but I don't have a problem telling them to stop and no. So, but the contractor, that's a whole thing, but we're doing okay. But this is where I've learned the most. Totally learned a lot and how we're going to proceed with future projects. Something that helps with my anxiety for these projects is to have a really great bat consultant that is going to help with the exclusion and just brainstorming how we're going to approach things. Because I love the bats. It's nice to have a bat consultant. As long as I know that they love the bats as much as I love the bats, then we're good. And so that takes a lot of stress off my shoulders, and I really appreciate it. So for those bat consultants, we love creative solutions. We love flexibility. We've learned a lot, and we've learned what we're not going to do the next time. But bat consultants help with stress a lot. Now, prior to construction and demo of any of the bridges, we like to have or put in place alternate roosting habitat. And so when I pointed out Walnut Creek Bridge, we put in 36 bat boxes down on the northbound frontage road. And we know the bats fly off to the east, the Mexican free-tailed bats fly off to the east, and so we put it on the northbound frontage road so that there wouldn't be car strikes. That's something to really, we have that issue with those braided ramps at McNeil. We put in that chain link fence to get the bats to kind of funnel up and fly over traffic. But in this case, we're just trying to avoid that at all costs. And just, we put everything on the easternmost. And so, as you guys know, the bats fly off to the east, go eat lots of bugs. Now, unfortunately, the contractor was moving fast. We wanted to have these bat boxes in place for a year before they were going to demo anything. But it turns out it was just a couple of months. But, you know, they were okay. We've got McNeil Bridge, which can absorb a lot of the bat population. And, you know, even if we exclude like half the bridge, there's plenty of places where the bats can go. Now, for other projects, other Capital Express projects like CapEx Central, CapEx South, we're going to put some bat boxes in there. There's smaller populations in downtown Austin at the Airport Boulevard Bridge. We didn't know they were there, but we took, you know, the general public let us know the bats were there. Unfortunately, they're going to be tunneling down, and so we can't put bat boxes on anything. So I'm hoping we can do, I don't want to touch this, I'm scared. There it is, bat condo. maybe we can put a bat condo, because I feel like these bats, if they took the time to notice them, they're going to want them to stay in the neighborhood. And why wouldn't you? Exclusion. This is the next step. And there's a lot of factors that go on. The lesson I think that we primarily learned from CapEx North and trying to do spring exclusion is that it's really bad. Typically, if you are in an environment where there's traffic or like cars driving, you don't want stuff falling down. You want a more permanent exclusion material in the cracks. Or if you have a lot of pedestrian, people who walk around, you don't want them to pull down the pool noodles. I mean, why? It's just so, we put in the spray foam, and the problem with the spray foam is that it has to be at 70 degrees Fahrenheit for that to set properly and not be tacky. And so in the spring, we're like, yeah, we can do spring exclusion. No, we can't. It's too cold at night. And then the bats don't want to leave. And then, so spring exclusion, if anything I've learned, spring exclusion is kind of a no-go now. But we really, each bridge is different. So here going forward, we just have to see what the situation is at the bridge, what the environment's like, what's going on, and then we can kind of choose what kind of exclusion devices we need. So it's either usually spray foam long-term, don't want things to fall out, or pool noodles with one-way exits. But we're very mindful of disease prevention with the white nose syndrome, you know, and the timing of the year, it's phased. It's not like we do exclusion all in one night. It has to be phased. Something that was really cool was that when they were doing exclusion, if you stick a pool noodle up to kind of push the bats out of the way, they don't really respond. But if you wiggle it like a snake, they just exit, just drop. It's so amazing. You're just like, oh. So if you guys are trying to get rid of a bat, whatever. Anyways, this is what we went in prior to, like at the start when we were talking to the engineers and the plan design, and we were trying to figure out when you shouldn't do exclusion and when you shouldn't do demolition. This is what we came up with. And the front end is really good. We're not going to do exclusion in the spring anymore. The ladies are pregnant, they're tired, it's cold, they don't want to go, it's hard to get them out. So fall is really the time. This was the original. Now we can push it up probably mid-September. The key thing that you want to have in the fall is to make sure everybody can fly. We want the bats to be able to go, because demolition is pretty loud and pretty disruptive. So I'm thinking like mid-September, we can kind of move things up. But knowing this now in future projects, we are going to put those limitations onto the contractor. We're only going to do fall exclusion, and we have to wait until, for whatever reason, I don't know if it's the first freeze or whatever, all those bats need to leave. Like that first flush of cold air, and everybody exits, because I know there's only a couple thousands, but it feels like a billion of those little bats, those Mexican free-tail bats in there. So it makes life a lot easier if most of them get the idea to fly south. That helps a lot. So demolition in itself, this is again a means and methods. So you don't really know what they're going to do until they do it. And so I spent one night out at Wells Branch Parkway, and they hammer the edges of the beams until it just falls. And so they did this demolition September, well, they did it in, I think it was late September at Wells Branch Parkway. And it was very disruptive. And every time a beam would fall down, the bats would fly out. And then they chip at the beams that are down on the ground, and then they haul that material out. Now, the good thing is they do this in one night. So it's quick. So I like that. But it's anything adjacent that, because the way we approach demolition and exclusion is we excluded habitat and a little bit on the northbound main lane and a little bit into the southbound main lane. And so they demoed all of that, and they're going to build that back up, and that's going to be habitat. And then we're going to exclude southbound main lane and then demo that. But the proximity of the new habitat to the old habitat that they're going to demo now, it's too close. So I have that lovely conversation upcoming with the contractor that really the only time we're going to let you demo is probably in the fall. Or we'll see. I don't know. We have to make sure that everybody can fly. I'm not gonna have pups falling out of there. A good thing to have though is the back pocket is all of y'all's eyeballs in the news station and be like, people are watching. You don't want to do this. Do you want to be on the news? I've got friends of friends that can call. I'm just kidding. I'm just kidding. I wouldn't do that, but I would probably. Anyways, so demo, we have to be sensitive. You want to make sure you don't want to do it on a cold night. So if you scare the adjacent bats, they can't fly. You have to be very considerate of that. But you try to be as reasonable with the contractor and their schedule as possible, because the last thing we need to do at TxDOT is hold up a project. And y'all have been on I-35 and we just need that to hurry up and go. But then the bridge construction. So even after demo, my job's not done. I have to go back out with the contractor as they're putting those new box beams in, and I have to measure the gaps and make sure the gaps are appropriate weight or size. And that is 3/4 of an inch to 1.5 inches for Mexican free-tail bats. And I'm gonna make sure every one of those beams is put back at the right space, because it just has to be. So, but then again, it kind of goes full circle. We come back, that new bridge construction, let's just make sure we put them in places where there's not going to be any bat strikes or anything like that. So let's see. Oh, that's it. I thought I had more to tell y'all, but that's it. But anyways, Josh, thanks for having me. But y'all, I'm serious. If you see anything funny going on with those construction people, you just call us or let your bat people know, Texas Parks and Wildlife Bat Conservation International, Timberland Tuttle, whoever. Just let anybody know, because TxDOT, we're trying to take care of our bats now. Well, we're just trying to take care of our bats. We need to take care of them. So thank you.
Time for a question or two for the panel.
Purple. Or blue, purple? There's not. I think that's an I-beam bridge, but we are going to put, when I-35, I'm sorry, is gonna get built back over Lady Bird Lake, on the pedestrian bridge, we're going to put bat boxes in. Green. There's bats occupying condos at Walnut. Oh, how are they? Have they yet occupied? As far as we know, so they were installed in March. 18 were installed in March, 18 were installed in June. We don't know yet if they're occupied. But probably I anticipate this spring, we're going to be taking a look at them and seeing. Yep. Yes. They're too high. So the bat houses, yes, they're very high up. There will be a trail that will run under there eventually. The city of Austin wants to put a trail in, but everything's going to be very high. There is some concern about the vegetation climbing up the pillars and snakes getting into the bat houses, but I just don't. I don't think that's going to happen. So, yes. Oh, go ahead. Mm-hmm. So the question is, the bat boxes that were installed at Walnut Creek Bridge, will that be the same amount of habitat as one of the bridges? And that is correct. So we calculated kind of the linear feet of occupied habitat under Howard Lane Bridge or Walnut Creek Bridge, and we created that many bat boxes that replicates that amount of linear feet. Well, it already has. So northbound Main Lane on Howard Lane and Wells Branch Parkway, those have already been excluded and demoed. And so eventually southbound Main Lane will be excluded and demoed. But in the meantime, they'll have new fresh habitat. And also one more thing, snowpocalypse with the bats. We were doing our study at the time and it was very heartbreaking. But it's good that we've documented that study. But I can tell you the bats are doing great and they're back. So they're everywhere. There's so many.
Thank you.
Thank you.
So now we're going to have a panel discussion on bat conservation. The panel discussion is going to be led by Darren Propay, And the panelists will be Amanda Adams, Sarah Fritz, Samantha Liebers, and Tracy White. So if you guys want to come up to the table, and I'll turn this over to Darren.
All right, great. Thank you very much. All right, so let's go ahead and get started. I know that you have met three of the panelists through the last three talks, but we have one additional panelist. So Amanda, I was wondering if you could take just a moment and introduce yourself since we haven't heard from you yet.
I know I got lucky. That hear me okay? Whoa. Okay.
Oh, it went off.
Hi, that's better. I'm Amanda Adams. I'm the Director of Research Coordination at Bat Conservation International and a Texas resident. So I get the fun job of working with our Texas bats and these amazing women here.
Awesome, thank you so much. All right, well, to get us started, I'm Darren Propay. I work at Texas Parks and Wildlife, and I just have to say that while I work with a lot of different species, I am not a bat expert. So we will leave all the answers to this crowd here, but I have put together a list of 147 questions that we're going to go through here today. So we'll go ahead and get started. These are more general questions. I feel like we got a lot into some of the technical details, and now we're going to ask some of these broader scaled questions. So my first question, you know, we hear about all these problems and challenges. Are bats doomed? Why or why not? And this is anybody can take this.
Yes, we give up.
Okay, moving on to the next question.
I'm more of a glass half full. I mean, I'm not going to say I don't get discouraged. Joking, but you know, we're trying, and it's just trying to keep positive. And if we can create more habitat, and if I was in charge of TxDOT, we'd have bat stuff, habitat everywhere. But it's gradual, and we're getting people aware, and the concern is there, and it's starting. But I don't think bats are doomed. I get sad a lot, but.
In case you don't realize, I'm British and I'm very sarcastic, so I was joking there. No, bats are not doomed, and I think a lot of the stuff that we have talked about here today shows the amount of effort, energy, expertise, funding that is going into trying to combat threats to bats, whether that be disease, energy, transport. There's a lot of really great people doing a lot of really great work, and we're continually expanding on the knowledge that we have and the practices that we're able to implement.
Yeah, so I'll just add a little bit to that. I agree that bats are not doomed. And if you look at just bat research in the past 10 years versus the 50 years before that, I mean, and even just the proportion for like, say, terrestrial vertebrate research or something, it is increasing a lot. They are really starting to get noticed for their importance, again, through agriculture, tourism. You know, $53 billion a year, the USDA is now saying that they save for agricultural products or the agricultural system. So we can see that attitudes are changing. You know, it's not that most people are still kind of scared of bats. And I'm not saying if there's a bat on the ground to go grab it, please don't. Call one of us. But I think attitudes are changing, and we see that a lot in Texas. And when it comes to white nose syndrome in the Northeast, some of those populations are starting to rebound. They had their big flow of white nose again, 2006, 2007. And we are starting to see some of those rebounds. So there seems to be like, even though there's like 90% population declines in some of the species, the few that survive are starting to repopulate. And so what we don't know, or I don't know, she might know, is are they now immune to white nose? Are those more immune? I don't think anyone knows. It's not just here. No one knows yet, but it's being worked on. You know, okay. Okay. So there's a lot of things that are improving and a lot of notice that's happening. So hopefully not.
Just to round that out, thinking about bats, we just had some research come out and an awesome report that you guys should check out at stateofthebats.org. Very pretty. But we have found through expert elicitation that 53% of our bat species just in North America are at conservation risk or at risk. So it is scary. I mean, I think we're all in ecology and conservation because we think we can make a difference. But it does get depressing when you work on trying to find a solution to, a invasive fungus that has come in, and it's been, over 10 years, 20 years of research, and we still don't have a solution. Same thing with wind fatalities, or yeah, it can be depressing. So it's really helpful when we have a new group to talk to about what we can do to conserve them, and we can all do to make a difference on the landscape.
Awesome, thank you all. I think it's important to establish that there are things that can be done and that are being done. And we saw that already in the talks, that there's action in a positive direction on many of these issues. So that's going to lead me into another question, because I would imagine some are asking, what can we do in the audience, in the public in general? So what would you say are some things that a landowner or someone of the public could do if they had bats on their property, or if they have some control over bat activity? What can they do to help bat populations?
So I know all of us sitting at this table have some favorite projects that we like to share that people can do in their own backyards, on their properties, on their big ranches, under their bridges. So I'm just going to share a couple that I really like sharing. One is just in your backyard is planting native. So we have a Gardening for Bats program, and it's really in line with what so many other taxa need, pollinators, and it's supporting that native ecosystem so that we are having a healthy habitat and insect base to help those bats eat well at night, and then they can go on and thrive. And then just another really very specific thing for those with larger tracts of land and tanks and whatnot on their ranches in Texas. We have this they call them wildlife ramps. So putting some little ramps, because bats can be water limited, and they'll take advantage of those tanks like many other wildlife. But sometimes they get trapped and they are not good swimmers. So giving them a little exit ramp out of the water so they don't drown is just one easy thing that landowners can do tomorrow.
Thanks. Something that just popped into my head that could be interesting for people is there are, so we've talked a lot about, or a few of us have talked about acoustics and using acoustics to survey bats. There's actually these tiny little acoustic recorders that you can plug into your smartphone, and they're $150, and they try, there's some issues, but they try to identify the species in real time. So you could literally walk around your property, see what species is there, and then find out what does that species need if you want to be a little bit more tailored to certain species. So that would be something that's really cool. And then I think taking kids to go to some of these locations. Bracken, if you sign up to be a member or a contributor to Bat Conservation International, $20 a year, you can go to Bracken for free one time, and you can schedule that. It is, owned by the Nature Conservancy and BCI, so it's private. But the Congress Bridge, or the bridge in Austin, is another great place. I think just education at the Congress Bridge is typically one of the, they were on your slide, I think, Austin Bat Refuge, Lee and Diane, and they have little bats there. And so the kids, I just took some kids there this summer, they get to go see those bats up close. And the little kid that I took, who's 6, now has a bats in their room. I was in their room this past weekend. Not real, no. Big, like stuffies hanging from their ceiling because they saw the bats in person a few months ago. And they're really interested. And so that was really cool to watch.
I think something that always comes to mind for me as someone who tries really hard to facilitate research and survey efforts. One of the best things that landowners can do if, they have suspicions that they have, a bridge that has bats under it, or they have a small cave or something like that, allowing researchers the access to actually monitor these populations is extremely important. Now we have white nose here. We've had a lot of people say like, well, we think that this is a loss, but we didn't really count the bats before now. Getting that data so that we can continue to, look at the impacts of white nose syndrome and other threats on bat populations, and then have the ability to actually see, hopefully, recovery is an extremely helpful way to help with bat conservation. Also, for those who, have kind of past features or something, many people will want to gate off of a cave property or something. They don't want random people going into their caves. Well, there are bat-friendly caves, and there are bat-friendly gates, and there are non-bat-friendly gates. And there are people out there who know how to put in bat-friendly gates. So that's another area where it's a really, really easy way to make sure that you're just doing it the right way to actually help the bats.
Put up bat boxes in your yard. I love it, and I feel bad that the neighbor had holes in their like roof, but I love walking by it, and there's bats. There was bats in like their house, which was bad, but put up a bat box. Put up a bat box. But something I want to share with y'all that y'all may not understand is that TxDOT funds a lot of mitigation for our impacts of our projects. And so a lot of that mitigation consists of excavation out of caves, out of karst features on preserves. And so it does a lot of good to the karst, little bugs in there, little insects in there, and salamanders and recharge, but it also provides more habitat, natural habitat for these bats to come and live. So it's not that you need to excavate a cave in your backyard because whatever, but we do that. And so like everybody else, we're just trying a little bit that we can to help with these bats.
So if I scale it up a little bit, and you can tell me your answers are the same if they are, but if I say we have a number of HCPs in the room, we have some governing bodies in the room, are there things that could be done at that level where governing organizations, governing bodies could implement some positive actions for bats?
What do you think? Oh, yeah.
For online folks, did we get that?
No.
Okay.
So I think we're struggling right now. We would like to do a program. I feel like Texas, the southeastern states are a little bit further ahead with kind of HC programmatics and figuring out kind of not mitigation, but just things that we can do to help mitigate our impacts to bats. And so instead of having to consult on everything, we want a giant document that if we do A, B, C, and D, we can help take care of the bats, and it's more streamlined, and we get a lot of traction. But we're still learning all that information. Tricolor bat, I mean, you guys, I don't know if you got the big picture. We don't know a whole lot. In Texas, it's rough. They're everywhere. They're nowhere. We don't know what we can do to help them other than just make sure they're not there, but then they're cute and they're curled up in the little leaves, and then like there could be in every tree. And so we're trying. And if we had more guys, we knew what to do, and it's okay that we don't. Hopefully we'll get there. And we'll be able to help do the right things to help take care of the bat better, more so.
All right, so, go ahead. The question was, are there things that governing bodies, HEPs, and so on could implement for bat conservation?
I just wanted to put in a plug for making sure that we're using data-driven, like science-driven conservation actions, and making sure that then we're supporting those efforts to find that data, and making sure that we're really like asking those questions and working together I think it's having enough time to do that, bringing everyone together before like the big project has to happen, right? Like, so it's having those conversations early and continuously. And so it's just really fostering those partnerships at the end.
Obviously, education. But, you know, on a small scale, depending on the regulating body, this is not HCP, this is a very different scale, I guess. But, you know, one thing that I think we had a little talk about this earlier is making sure that like our pest control people know what they're doing, because that often is not, they do not. And having like bat-friendly exclusions instead of bat killing. You know, certain things like that are policies that local governments could put into place, education wise for those types of people as well, not just kids education. But I think the more people learn, and it's amazing when you see people see these caves and see bats up close for the first time. People are mesmerized. A lot of people. Now, I don't know. These are also wildlife students often, but people are pretty mesmerized. And so, you know, having people come in to do some of those programs, I think would be great.
Awesome. So far I've focused some of these questions on action items, right? What are the things that we can do? And that's certainly going to be a key component of this. But we know that the research, as some of you all described already, is also a key component, filling in those gaps that help us to know where that action needs to take place. What would you say are some of the big research gaps where, you know, maybe if we could fill some of these gaps, we could move that needle forward on conservation?
Everything. Yeah, right. So everything. So as was mentioned, we don't know much about bats. So the study that Madison mentioned before, you know, we were capturing eastern red bats for a year, and then we're like, let's do genetics on these. They were all western reds. But we don't know what they were before we did the genetics on 15 of them. We don't know where they are. We don't know when they're there. We don't know so much about bats. So really, I think as they have become more popular, what's really cool to watch is the engineers, not necessarily the people building bridges. I don't know about your engineers, but the engineers that are doing a lot of the technology for tracking, also the video tracking, like wildlife imaging system. That person did not start off in bats. The person at Iowa State that's making the deterrent is an engineer, didn't start off with bats, but then like saw this issue and took what they knew, their expertise, to create bat deterrents. And so these types of people coming together and having meetings like the Wind Wildlife Research meeting, where people come from all sects to kind of work together, see what the issues are, is really important, because all the knowledge gaps exist with bats.
I always say it's really difficult having the word specialist in your name, because everybody expects me to know everything about bats. And the fact is, we don't know everything about bats. For me, what I would absolutely love to see, and I know what every single one of us would love to see, is a way to track individual bats in real time. You know, I think in the wildlife field, we kind of take it as a given now. We think of like GPS collars on like coyotes or bighorn sheep or something like that. Well, you can't put that on a bat. You know, and there are certain things that we have in place now. So the modus tracking system is an option, but again, it's only an option for some bats, and it's quite crude movement. So I would absolutely love to find a way for us to be able to track individual bats in real time and finally get a real idea of, you know, when they're arriving, when they're leaving, actual migration routes, stopover routes. And that would make my job a lot easier, actually.
Just to add on to that, because I know there's a lot of different disciplines sitting in the room. That's actually one of the coolest things, going back to the action items, is just finding ways to leverage our different efforts. So, you know, putting people who are looking at studying bird migration, like at nocturnal bird migration and that work, doing more synergistic work. I think there's still, like, we're still a little siloed, but, you know, like, especially with this MODIS work and doing this tracking, like putting up those towers is pretty expensive, but then once they're up, they can benefit multiple taxa. So if you're writing a grant to put up a MODIS tower, think about the other groups in your area that would also want to tag some animals, and then you guys can all be using it and work on siting. That'll be mutually beneficial. So I think there's some of that
synergism that we need to do more. So as you're out there doing your work, think about how you can include bats in your research, because obviously we need some help.
Great. So there's a bunch of Texans in the room here, I assume, right? Safely so. And Texans, at least our image sometimes is like we like to lead, we like to be at the forefront, right? In front of everything. So are there parts of the bat conservation research where we can brag a little bit, maybe say, these are areas that in Texas, we're really doing a good job sort of getting on the front of this. And then maybe conversely, are there some areas we'd say, we're lagging a little bit and then we need to really pick up speed?
So I think Sam is going to have one of the best answers for this. I'm hoping she'll dive into it with some of our white nose work that's been done in the state. But I did want to, like, some of the talks emphasized just in terms of how special Texas is, being the most diverse state for bats. You know, we have this huge suite of, you know, Western species, Eastern species, like they all come into the middle. We just like, there's a lot of really, really cool things going on here. So it is, and it's probably like the most, like we have Austin, the city that loves bats the most. You know, I just think in terms of Texas and being proud of bats, Texas is pretty special. And we have done some leading in the charge in white nose work and wind work, say. I'll let you guys talk. Well, obviously, clearly as a native Texan born and bred, I would agree with Amanda. I think some of the white nose work that we have been doing and are continuing to do right now is going to be extremely informative. Like I said in my talk, Texas is really now very much on the leading edge of the expansion of white nose syndrome, and we are learning more and more, and it's beginning to take shape. But you know, we need more research, and that is what we're getting the funding to do. And not just in Texas, like across the Southwest, even down into Mexico. So we're taking part in a really prominent area of research with that right now. And I would also agree, just, you know, leading the way, it is wonderful to see the attitude of Texans towards their bats. And I would say 9 times out of 10, when I tell people my job, they go, "That's awesome." And there's only maybe like 1 out of 10 who's like, "Ew, bat." So it's really phenomenal to see the level of support for bats in Texas. And I think that that's something that we can be really, really proud of.
I think the fact that we have a state bat biologist is pretty cool. So you know, applause to Texas Parks and Wildlife. Not a lot of states do that. Texas Parks and Wildlife and other state agencies, including the Comptroller, also fund bat research. Yay! That helps me out and helps the bats out. And in fact, there was even a Pittman-Robertson grant that was used to fund a bat research. And typically that money, it is for any mammal or bird, but typically has gone to game species. But several years ago, that was earmarked for bats. And so I think Texas Parks and Wildlife has a lot of interest. So thanks to your agencies, all of y'all's agencies. I mean, the fact that we have people really cared about that for TxDOT as well. And BCI was founded here. So we have a lot of people, we have a lot of experts, we have a lot of things going on. We just need more.
So I think, you know, I've only been at TxDOT for about 5 years, and so I don't really know the history and the approach. But in speaking to senior former TxDOT people, 10 years ago, I don't think TxDOT would have taken the approach that we're trying to preserve this habitat and exclude that habitat and take care of our bats, because these Mexican free-tailed bats, they're not federally protected. There's, you know, yeah. So we're being proactive. We're taking care of them because, and I do think, again, it's because we are in Austin. That's part of it. And hopefully that will spread across the other districts in the state. And so hoping that'll happen. I think we're headed that way.
Awesome. Well, let's have a little fun with this next question. So if I gave you 20 seconds, I'm not going to time you, but we have to save it 20 seconds on world news tonight. What is your priority message or request? Anybody can go when you're ready.
Bats are super important and provide increasingly important ecosystem services. Please give us more money.
I think that's it too. There's a lot of really cool things about bats that people don't know. So, people are very afraid of rabies, and they are a rabies vector, but only 0.1% of bats in the wild carry rabies. Bats can see very well. They don't fly into your hair. They don't drink your blood. There's only 3 vampire bat species in the world, and they all live a little south of the border, and only one has been documented to feed on humans. Baby bats, which are pups, have been documented to babble, just like human babies. So I know, so.
You just got a bonus extra 20 seconds for that one.
And the thought behind that for humans is that babies will like say the same syllable, da, 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 because it gets the parents' attention without annoying them, but pups do the same thing. Our Brazilian free-tail bats that we've talked about have at least 30 different types of vocalizations. They have individual recognition. In Bracken Cave, one sheet of paper, 8 by 10, has like 300, can fit like 300 bat pups, and the moms fly out and find their bat pup by olfaction, and is it 20 seconds? So really cool facts, right? And I think if people start seeing that, then they will care more.
I haven't workshopped this, but I think it would be coming back to that concept of One Health, in that we really need, for us to be healthy, we need healthy ecosystems and we need healthy wildlife. So by spending time like protecting our ecosystems and important wildlife, especially bats, we're going to be happier and healthier, and we're going to have these amazing animals flying around, at least making the 4 of us smile.
You're good. All right. Thank you all. I have one last question. I always think it's good to end with some cool stories, some hopeful insight when we think about bats or really any conservation topic. And you've already done a little bit of this, but is there a couple stories that you could offer in our closing time here that are fun and give us some hope and excitement about bats and what's going on?
I didn't get to finish, so it kind of so it falls actually under this, I guess, like stories. I mean, I don't know if you mean for exactly what we've done or for bats, but another cool story I have. It wasn't me. It was a former student colleague of mine when I was an undergrad at Georgia who documented bats leaving a roost, and the mothers were dropping off their pups in a bush, so they have little daycares. And some of the other mothers like hang out and watch. And when I heard that, and I was an undergrad at the time, he was a master's student, and I was just like, wait, what? I mean, bats have not always comprised all my research, but I was pretty mesmerized and wanted to know more right then and there. So that was a really cool story that changed my perception of bats when I was younger.
The first thing that really comes to mind, the week before Christmas, I did my particularly large white nose syndrome monitoring trip that I do. And I surveyed 19 hibernacula in 7 days. And even though it was the beginning of winter, so we can't say too much, touch wood, I saw healthy tricolored bats within the normal kind of numbers that I've been finding the last several years, and no evidence of white fungus on noses or wings, including in the cave where we had white nose positive bats last year. So I'm hopeful. I'm going to be checking, towards the end of winter and seeing how we're doing there, but I was very pleased to see healthy tricolored bats this year.
So I'm an invertebrate zoologist, y'all. I don't like things with backbones, and I love bats. I don't know if you heard me over every time a cute bat picture came on, you're just like, they're great. And so I think there's just a general love for bats. But like I touched on when I was talking, it was really sad to see the snowpocalypse and the impacts. It was crushing. But I will tell you, they're back, and there's so many. And then just think about that little wiggle in the pool noodle. I just think about that all the time. pool noodle don't work, but you wiggle it and they freak out. It's magic. So it's pretty amazing, but they're very resilient and they're squeaky and they're wonderful. And I'm a convert now.
I'm going to zoom out a little bit, and sometimes it can feel like I was saying, depressing when we keep working on conservation and we don't have wins. But we do have very tangible wins in our bat conservation efforts with some species that have had real efforts, and federal listings made a difference in their conservation. So the lesser longnose bat, Leptonicterus nivalis, is one that was listed in the U.S. and Mexico, and through the efforts of many, many people, agave restoration across in the US and Mexico along their migration routes around their maternity colonies, like they are doing much better. They are delisted. They're doing great. So we have a species success. Gray bats in the Midwest. Our most recent counts from our national or North American Bat Monitoring Program data is showing that they're increasing, and they're still federally listed, but their population trends are going up. They are not decreasing. So we have a lot of hope that these efforts are making a difference, and our time and financial investments are worthwhile. So I think also, so don't give up hope on the bats, but also on your own conservation projects. It's all worth it.
Awesome. Thanks, y'all. We'll close our conversation with that. I appreciate all of you being on the panel here today. I will turn it back over.
Yeah, join me in thanking Darren and all the members of the panel. This was a really great way to end our first day. We'll return tomorrow at 9 a.m. Josh, do you want to say anything about the reception tonight? We have a reception starting at 5:30.
Yeah, so it will be downtown from 5:30 to 7:30. We'll have some folks there. We'll have drink tickets. So if you don't have one, which none of you do, there will be at least a handful of people with them handing them out. There is going to be some light food there also. So it's probably not dinner, but there is something to eat. So yeah, we will certainly see you there.
Right.